Is now with us. So, we welcome you to all this webinar, Role of Biodiversity Conservation in Ecosystem Functioning. Respected dignitaries, Honorable Director, Zoological Survey of India, Dr. Thursday Banerjee, Honorable Vice Chancellor, Professor Nilima Gupta, Director, Dean, School of Biological Sciences, and Head, Department of Zoology, Professor Subodh Zen. Coordinator from Zoological Survey of India, Dr. Anzum Rizvi. We are thankful for your valuable time and your kind presence in this event. Before proceeding to the webinar, I would like to tell about the MOU signed between both organizations, Zoological Survey of India and Dr. Harisingh Vishwavidyalaya. We had signed the MOU this year, and this is our first activity. I hope this webinar will kick off our academic activities in a long way. In this webinar, we have enrolled 349 participants and 26 from the faculty and 37% from research scholars, 24% from PG student. And out of them, 66% participants have registered themselves for gathering information to boost their research activity, and 31% have registered to obtain the information for their further studies. Fortunately, the webinar has received a wide attention across the country. We have 60 participants from central India. From northern region, we have 90 participants, and southern part of the country, 34 participants. And from west and eastern region, 87 and 57 participants respectively. We have 6 speakers in this webinar. One of the speakers will speak on role of aquatic insects in wetland biodiversity conservation. Another one will speak on plant diversity in northeastern region. One of the speaker will throw light on conservation strategy, and another one will speak on nematode diversity and invasive reverse co-evolution from Allahabad University. And one of the speakers from Agriculture Research Institute will speak on crop breeding strategy in changing climatic scenario. And the last one will throw light on the top of the animal kingdom, mammals, in particular, role of mammals in ecosystem functioning. We have speakers from Zoological Survey of India, Allahabad University, Indian Agriculture Research Institute, and Botanical Survey of India. The deliberation will be placed jointly by Dr. Harisingh Vishwavidyalaya and Zoological Survey of India by coordinator Dr. Anzum Rizvi along with me in the last of the conference. At 4 o'clock, we will place a link for the feedback in the chat box. After completing the desired information, the participants will able to download their e-certificates. The link will be open for 2 hours, including 1 hour after the completion of the webinar. Since the webinar has received wide attention, we have enrolled sizable number of the participants. It was not feasible to accommodate all of these participants over Google Meet. So we have streamlined the webinar on YouTube channel. And the information about the YouTube channel and the Google Meet has already sent to the participant, and for the ready reference, it has been pasted in the chat box. So for those participants are not able to see over the Google Meet, can watch it over the YouTube channel. Further, I hope that webinar will be useful not only for the ZSI and the Dr. Harisingh Vishwavidyalaya, for all participants, those are here, and it will open a new venture for the research in this thirst area of the webinar. Now, I would like to hand over it to our Dean School of Biological Sciences, Head Department of Zoology, Dr. Subodh Zain. Now it is over to him.
Thank you very much. Good morning, Honorable Dr. Dhriti Banerjee, Director of Zoological Survey of India, Kolkata, Honorable Vice Chancellor, Professor Nilima Gupta, Dr. Anjum Rijvi from ZSI Kolkata, Professor Sweta Yadav, convener of the webinar, resource persons, invited speakers, fellow colleagues, media persons, participants and friends. On behalf of the Department of Zoology, Dr. Harisingh Gaur Vishwavidyalaya Sagar, and my own behalf, I welcome you all in the webinar on the role of biodiversity conservation in ecosystem functioning. Dr. Harisingh Gaur, Central University Sagar, and Zoological Survey of India, Kolkata, signed MOU on May 25th this year with the sole effort of our Honorable Vice Chancellor, Professor Nilima Gupta, with the objectives to exchange students, scholars, and scientific staff, exchange of academic information and materials with ZSI Kolkata and any of the 16 regional centers for undertaking doctoral and postdoctoral program by this university, joint research activities, publications, organization of conference, seminar, lectures, etc., so that participants of both the institutions may be benefited. In order to fulfill the goal of the MOU, Dr. Jasmine P. from ZSI Kolkata and Professor Sweta Yadav from our university are designated as coordinators of the program. And as you can see, today we are organizing this webinar on biodiversity conservation, for which Professor Sweta Yadav worked very hard to discuss various issues related to the role of biodiversity conservation in ecosystem functioning with the participants, distinguished scientists from all over the country, Dr. K. Subramanian, ZSI, Southern Regional Center, Chennai, Dr. Umesh Tiwari, Botanical Survey of India, Kolkata, Dr. Umar Sharif, Botanical Survey of India, Southern Regional Center, Coimbatore, Professor S. K. Malhotra, Allahabad University, Prayagraj, Dr. Rajendra Kumar, Indian Institute of Agricultural Research, New Delhi, Dr. Kamla Kanan, M. ZSI Western Ghat Regional Center, Kerala, Dr. Anjum Rijvi, ZSI Kolkata, have very kindly given their consent to deliver lecture and interact with the participants. I welcome all the distinguished scientists and you all. Thank you very much.
Thank you, Professor Subodh Jain. Now I would request Honorable Director Dr. Dhriti Banerjee to deliver her inaugural address to the webinar. Now it is over to Dr. Banerjee.
Thank you, Dr. Shweta. Am I visible and audible?
Yes, you are. You are perfect.
Yes, ma'am.
Okay, okay. So thank you so very much. Good morning, everyone. And today it's a very bright morning in Kolkata. I don't know what it is there around you, but I think it's a very sunny morning in all of you because it's cold, it's pleasant, it's winter, and it's a privilege to welcome you all to the webinar, which is being jointly organized by the Department of Zoology, Dr. Harisingh Gaur Vishwavidyalaya, Sagar MP, and the Zoological Survey of India, Kolkata. It's an honor to share the platform with honorable delegates like Professor Nilima Gupta, the Honorable Vice Chancellor of Dr. Harisingh Gaur Vishwavidyalaya, Sagar MP, Professor S.K. Malhotra, Allahabad University, Dr. Umar Sharif, Dr. Umesh Tiwari, Dr. Rajendra Kumar, Head of the Department of Zoology, Professor Sandeep Malhotra, as well as my colleagues in Zoological Survey of India. So, as the head of the Department of Zoology, Dr. Subodh Jain has said that we have signed an MOU with the Zoological Survey of India, with the Harisingh Gaur University, ZSI and Harisingh Gour University have signed an MOU, and as a first step in making this MOU viable, we thought it would be an interesting thing. The effort was mainly on part of the university, that it would be an important step for us to have a joint webinar with both the university as well as with the university professors and the ZSI scientists. So, and I think it will be a resounding success considering the number of participants who have already joined the webinar. So it is, and I would like to thank Dr. Anjum Rizvi, scientist of Zoological Survey of India, for taking the initiative, as well as Professor Shweta Yadav, Department of Zoology, Harisingh Gour Vishwavidyalaya, for jointly taking the initiative and basically harping on me, madam time dena hai, madam time dena hai, kab baithna hai, madam ek webinar kar lete hai. So thank you for making this possible. So today we'll be speaking on role of biodiversity conservation in ecosystem functioning, and it's my pleasure and my privilege to address in this inaugural session. So, as a motto of the Ministry of Environment, Forest and Climate Change, we know that it is Prakriti Rakshati Rakshita, or nature protects when she is protected. And protection of and conservation of the ecosystem is an integral part of nature, and it needs to be protected for the betterment of our world. The term ecosystem functioning refers to a general concept that incorporates individually or in combination different ecosystem processes like biogeochemical cycles, properties like pools of organic matters, goods derived from the ecosystem, foods, medicines, special services like regulation of climate or cleansing of air or water, or temporal resistance or resilience of these factors over a time or response to disturbance. So in brief, it includes total processes of ecosystems and individual ecosystem components which are involved in them. From the mid-1990s, a huge number of high-impact articles have been published regarding the relationship between species diversity richness and the stability and functioning of the ecosystems. These scientific articles assert a relationship exists between the species richness and ecosystem functioning, and that this relationship can be used to argue for conservation of biodiversity. According to the research done, if ecosystem functions are sustained at relatively low species richness, then arguing for conservation of ecosystem function, no matter how important in its own right, it does not strongly argue for conservation of the species. For this to be a strong conservation argument, the link between species diversity and ecosystem functions of value to the human community must be very clear to the researchers. Now, to use a function and relationship between an ecosystem function and diversity as a foundation for advocating for conservation of biodiversity, a few criteria must be met. Like firstly, the maintenance of ecosystem processes must depend on a broad array of species such that the conservation of function embraces the conservation of most or all diversity within the ecosystem. Secondly, ecosystem function or the products of its function ought to depend on the native species. If exotic species that dominate ecosystems are redundant to those that they displace, then the conservation of function can be maintained by conservation of non-native diversity. But this would not provide strong support for the conservation of the native species diversity. In most of the famous studies done by Schwartz somewhat around 2000, it was shown that communities are characterized by strong dominance and that a few species provide the vast majority of community biomass. Rapid turnover of the species can rescue the concept that diversity leads to maximum function and stability of a set community. Now, the process for conservation of a community as a whole, the factors that are detrimental to the species in question must be removed or worked upon. For this purpose, in our country, many government and non-profit organizations are working tirelessly and relentlessly. The species that are vulnerable or endangered are flagged and are used as a marker for conservation of the area they are in, creating a better living situation for them and protecting the complete community as a whole. Now, for the conservation of biodiversity, our institution, the Zoological Survey of India, has been working relentlessly on several platforms in association with international and national systems like the CBD, that is the Convention of Biological Diversity, the Excess Benefit Sharing, the Wildlife Protection Act, the CITES, as well as under the umbrella of the Ministry of Environment, Forest and Climate Change. Now, the Convention of Biological Diversity, as you know, is an international legally binding treaty which was adopted in Rio de Janeiro in June 1992. ZSI provides national level data to faunal resources of India for the country report to CBD, along with supporting implementation of the National Biodiversity Action Plan and achieving national and Aichi biodiversity targets. ZSI experts are relentlessly and tirelessly contributing to various assessments under the multidisciplinary expert panels to implement work program under intergovernmental platform on biodiversity and ecosystem services, or the IPBES, a science policy interface on biodiversity. Access and benefit sharing, or ABS, is one of the key provisions of the Biological Diversity Act 2002, and ZSI has been part of the expert committee of the National Biodiversity Authority that guides and advises to the issues related to ABS. Our institution also supports the state biodiversity boards, the BMCs, in developing and validating the People's Biodiversity Register. So we are a premier organization, as you all know, on faunal resources of Ministry of Environment, Forest and Climate Change. We have prepared the list of faunal species under different schedules of the Wildlife Protection Act and previously, and we are now supporting Ministry of Environment, Forest and Climate Change in revising the Wildlife Protection Act and its schedules and providing guidance for amending the different sections of the WPA. ZSI is a leading organization which is identified and supports state governments in notifying the faunal species to be listed in the threatened species under the Section 38 of the Biodiversity Act. Most recently, under the guidance of the MOFCC, we have developed a strategy for designating the species as threatened for conservation and access and benefit sharing as per Section 38 of the Biodiversity Act of 2002. As a preliminary step for conservation of the species is assessing its role in community they belong to. For conservation of an ecosystem, the assessment of its functional indicator traits needs to be prepared. Information on each gathered species of the ecosystem to be restored or conserved is studied by splitting the traits into categories. For example, in marine ecosystem, the traits can be categorized into feeding methods, which again be subcategorized into producer, filter feeder, deposit feeder, etc. So generation of ideas about how the approach need to be the guide to conservation and management of a certain ecosystem, the assessment of the effects of anthropogenic activities, and subsequent management strategies are always taken into account, and predictions are done accordingly for future changes. Trait analysis provides more information on functioning than species-based methods, and our institution is now trying to thread together these two aspects, which can be a supreme factor in the coming days for conservation of biodiversity in many areas of our country. So conservation biology has been called a crisis discipline by Soule in 1986, which requires timely answers to complex problems. The complexity of linkage between ecosystem function and species richness limits our ability to precisely predict the relationship between them. The amount of ongoing work on this topic is high, and we expect to get some conclusive results in the near future regarding the proximity and dependence of these two. From the conservation point of view, it is the need of the hour, and the research on the relationship between the biodiversity and ecosystem function will provide us access to get some great ideas and policies for conservation in the future. As a famous author, Joel Salatin, had said, that the average person is still under the aberrant delusion that food should be somebody else's responsibility until he is ready to eat it. It is our duty to awake the common people and start the awareness about nature's degradation and how conservation is a need of the hour before it is too late. Thank you again, Harisingh Gour Vishwavidyalaya, for giving us this opportunity to host this webinar jointly along with you. A very warm welcome to all the participants. Thank you very much. Namaskar and Jai Hind.
Thank you, Dr. Banerjee. You have given valuable information regarding the species diversity and the functional ecosystem. And we totally agree to your worriness about the invasive species. And you have given light over the wide spreading of the exotic species that are certainly affecting the biodiversity and playing adverse role in ecosystem functioning. So now it is the time to hand over this webinar to our Honorable Vice Chancellor, Professor Nilima Gupta, Madam, for her presidential address. Now, Madam.
Thank you very much, Professor Shweta Yadav. It was wonderful to listen right now to the very lively address by Dr. Dhriti Banerjee, who is the director of the Zoological Survey of India. And let me tell you, the morning always becomes bright when we see bright people around us. So it's indeed a very wonderful morning when we have with us not only the director of ZSI, but we also have a number of very eminent speakers with us, like Dr. K.A. Subramaniam, who is from the Zoological Survey of India. We have Dr. Umesh Tiwari from the Botanical Survey of India. I call them sisters, ZSI and BSI. So from their sister concern, Dr. Omar Sharif from, again, Botanical Survey of India. Professor S.K. Malhotra from Allahabad University, Prayagraj, who is a pioneer on nematode study. So I think he'll be talking about nematode biodiversity. He's also with us. We have another very prominent figure with us, and that is Dr. Rajendra Kumar, who has been the Director General of UP Council of Agricultural Research, and he is right now at IARI New Delhi. And since this is a webinar on biodiversity, he has an expertise on crop breeding strategies, especially with respect to changing climatic scenario, because today we know how we are undergoing the climate change, and in this climate change, how to have an agriculture which is resilient to climate change. So I think that this is also a wonderful topic. And of course, the highest in the hierarchy, that is the mammals, they will also not be left untouched with the talk of Dr. Kamala Kanan from the Zoological Survey of India from the Western Ghat Regional Center, Kerala. We have also with us the head of the Department of Zoology and the Dean, Professor Subodh Jain, who has taken a lead in this direction. And of course, the two pin persons from Zoological Survey of India. Anjum is already laughing. She knows that her name is going to come. Yes, you're right. We have Dr. Anjum Rizvi. I know she's a very dynamic personality. And equally dynamic we have here in our university also, Professor Shweta Yadav. And they have wonderfully coordinated together to organize this meet. And as it was just said, that this is a joint venture of the Zoological Survey of India and Dr. Harisingh Gour, Sagar Central University. It is indeed a joint venture, and it is an outcome of the MOU which we have signed. So now talking about ZSI, I think that no zoologist can, during his or her lifetime, it cannot be possible that we have not come across ZSI. And similarly, being a zoologist myself and thinking about biodiversity, working on biodiversity, throughout our career, we have been linked up with the Zoological Survey of India, describing species one after the other. And then, of course, whenever we find something new, we remember ZSI and we knock at their doors. And it is they who give us recognition in the form of holotypes and paratypes, and we go on for our publications. So, just talking about MOUs, I must say that since I have always been linked up with ZSI, this is the third MOU which I have done with Zoological Survey of India. Incidentally, when I was Vice Chancellor at Chhatrapati Shahuji Maharaj Kanpur University, that was our first MOU with ZSI. When I was Vice Chancellor at Tilka Manjhi Bhagalpur University, that was a second MOU with Zoological Survey of India. And I left it to their hands to proceed along with ZSI. And now being the Vice Chancellor of Dr. Harisingh Gour Sagar University, this is the third MOU which we have done with ZSI once again. And I'm sure that this MOU is going to go a long way in developing research bond between the two institutions. Well, as a result of this only, we are having this first meet, and I'm very happy that due to the efforts of Professor Shweta Yadav and Dr. Anjum Rizvi, we have got such a wide audience, and all of them are linked up with this program today. So having at the first go, having more than 300 people linked up to the program, I think that it is a wonderful initiative which has been taken up by the two organizations. Of course, we plan to do much more, and this is just the beginning. And I'm sure that the coming year, because now it is December, and it's time to say bye-bye to 2022 and to welcome 2023. And 2023 will begin with an invitation from our side to call the ZSI team to Dr. Harisingh Gour University. And of course, we'll follow suit. We will also visit Zoological Survey of India. And that way, because once going and visiting the institutions, then only we come to know about the actual facilities which are present in that institute. And I'm sure that it will develop into a still better bond between the two institutions. Now, I'm very happy that Dr. Banerjee, she talked a lot about biodiversity, and she also tried to link up nature with biodiversity, because I think that both of them are incomplete. If we talk about nature, we cannot ignore biodiversity. If we talk about biodiversity, we cannot ignore nature. They go together, hand in hand. And when we are talking about biodiversity, we go back over the years and try to recall when did life originate. So it was about 3.5 billion years ago when life originated, and that was the beginning of biodiversity. And gradually, the story went on. We had more of the species belonging to the zoological group and also to the plant species. And today, scientists say that, of course, there are different figures, but according to some figures, they say that about 2 million currently living species are known on the earth today. Out of 10 billion, just imagine, it is estimated that about 10 million are there. And if Anjum remembers the time, I'm referring to Anjum because she's like my younger sister, and she has been, we are guru sisters. I mean, we both of us have had the same guru. So I think she remembers very well. And when we were working on biodiversity and trying to find out new species, often we said, "Arey, ab kya reh gaya hai? What remains? Everything is done. There is nothing left for us to done." But gradually we started working on species one after the other, and I'm very happy I could identify 51 new species from, I mean, only parasitic species. And this was something, then I realized that there's so much of biodiversity which still remains, and it is only a fraction of work. And I know many taxonomists are working on that, and they're trying to find out species one after the other. So overall, we would say that 80% of the species, they have not yet been given a name. So that much needs to be done. So we are at that point of time when we talk about this collaboration, that we have a huge sea, an ocean where we have to make discoveries on the new species. So we can go ahead, and this is also a message to our youngsters. I see a number of young scholars who are attached with or linked up with this program, the research scholars as also the PG students. So we have to work on that. So on one hand, On one hand, what we are worried about is, the worry is not that we have so many species. Of course, we have workers and we will keep on discovering them. But the point of worry is that about 20 species of animals and plants are becoming extinct every day. This is very important. And if we move at that pace, it is estimated that in about 30 years, that is 30 years from now, around 2050-52, we will have about 100 species which will become extinct every day. So this is the point of concern, that today when we are talking on this very important issue of the webinar, that is role of biodiversity conservation in ecosystem functioning. So ecosystem, let me come back to what is ecosystem. Ecosystem means that we must have, this is a law of nature. We must have all the species who are working together. It may be plants, it may be animals, and it is this food cycle, this food web, which works together to maintain the biodiversity or the so-called nature which we find today. So we have to think about how we have to preserve this biodiversity. And if we just see the nature, I feel that our solutions lie in the nature itself. Because it is we who are working on the nature, it is we who are destroying the nature. And due to our activities, we find that nature is being depleted day by day. So our youngsters, again, I'm addressing this to our young scholars of MSc and PhD, that how is it that the species are becoming extinct? And if we know the causes, then of course we can find the remedy as well. So I think we should identify the causes for species extinction. And for that, I would request our youngsters to remember the word hippo. You have heard of the word hippo, I'm sure, that the huge animal. So hippo here, I'm not referring to that animal, but I'm just taking the initials, HIPPO. So just we can elaborate these. H will go for habitat alteration. That it is we who are altering the habitat. Say for example, deforestation. There was a natural forest, but we are cutting out the forest to have multi-storied buildings, industries, and that is how we are losing this habitat. So what about the forest animals? Of course, they are going to decline because now they are not getting their natural habitat. Then the next letter in the word hippo is I, that is invasive species. Invasive species, that is those species which were not earlier present in a particular locale, they now come and invade that area. So now what happens, that becomes an interspecific competition between the species. As a result, there is a decline in the population. So here again, we suffer from biodiversity loss. Then there are two P's, HIPPO. So these two P, I think it's the same letter and they also go together. One is pollution and the other is population growth. So they are linked with each other. Let me take a population growth. As we are having population growth, simultaneously we are increasing the pollution. So together with this, when our population is increasing, when we are increasing our pollution, obviously pollution, it may be the air pollution, it may be the aquatic pollution, we find a loss in biodiversity. Professor Malhotra is sitting in front of me, and I think we have done a lot of work where we have seen this pollution, how it is destroying the aquatic life and what is happening to the parasites as well. And finally, O. O stands for over exploitation. So now we have the animals, we have the plants, and we are over exploiting them. That is, we are simply trying to, it may be fishing, it may be wildlife hunting, and when we are over exploiting, this is biodiversity loss. So this simple hippo tells us that how we are depleting our biodiversity, and as such, I think we have to think on all these issues. That is how we can preserve the habitat. It may be grassland, it may be water, it may be the air. We should not disturb them. We should stop invasion of species from one place to the other. Similarly, we must have control. The government is doing a lot for population control, pollution control. We have different acts. What about these different acts? We have all these different acts. Also, we have the Biodiversity Act, we have the Pollution Act, we have the Air Pollution Act, we have the Wildlife Conservation Act. Are we preserving? Are we following these acts? So if we are only trying to follow these acts, then I think we would succeed to a great extent in conserving our species. So now when we're talking about biodiversity, diversity, does it refer only to the species which I am talking about? I would say that it does not refer only to the species. It refers to genetic diversity, it refers to species diversity, it refers to ecosystem biodiversity. These are three major components of biodiversity. So this, I think we are, I think, well aware of our biological wealth by now. I must say that it is a wealth, it is a treasure. Why do we go to zoo to see the animals? Because we find a wealth of animals who are present there. Why do we go to national parks? Why do we go to different types of parks which are present? It may be the plant conservation, it may be cactus plants, it may be the rose gardens, it may be the tulip gardens, and we are very fond of going and visiting all these places. So it is this natural beauty or this natural biodiversity which reflects us. So we have to let us try to think in this way, that it is a time for reflections. It is a time to analyze ourselves, that what we are doing to the biodiversity, and we have to find out the opportunities which are prevalent then, and also the solutions, so that we can build back a better future. So I think that it is a very good topic which has been selected by our coordinators right now. And we have to preserve our biodiversity in several ways. I told you what are the causes. We have to, and now the question comes, another very basic question. I'm just talking about the very basics. That is, why conserve biodiversity? ठीक है, biodiversity नुकसान हो रहा है, होने दो. We can also become very casual. But no, we cannot become, because biodiversity is very important for us. It is not only from the sense of beauty, but we utilize it for food, for fuel, for fiber. We utilize it for shelter. We utilize it for air and water purification. We utilize it for waste decomposition. We utilize it for climate stabilization and modernization. We utilize it for nutrient cycling. We utilize it for soil fertility. We utilize biodiversity for pollination. We utilize biodiversity for pest control. We utilize biodiversity for genetic resources. So just see, we cannot in any sense, we cannot ignore biodiversity. So it is time that we think very gravely about it. The topic is so interesting that if Professor Shweta allows, I can go on speaking for hours, but I think I only have 2 minutes more. So I must wind up, but I must compliment the organizer. And I feel very happy. I feel delighted from the core of my heart that we have had a bond with ZSI. And I remember the visit of our Dr. Anjum team when they came to Kanpur also under this MOU, and how much biodiversity they could find in the campus there. Let me tell you, we have a very rich biodiversity here in Sagar also. So we invite you on behalf of Dr. Harisingh or Sagar University to come to Sagar and to see the biodiversity which is present here. And of course, we have ZSI on whom we can bank upon. We can deposit our species and make the biodiversity even richer. So I convey my compliments for the success of this webinar, and may it pave a new way for further studies on biodiversity and conservation of species. I once again thank, of course, Dr. Dhriti Banerjee, the director of ZSI, who has been kind enough to be with us on this day, and all the eminent speakers who are so enthusiastic and energetic. And I'm sure that they will be delivering wonderful talks for our young speakers. And I would request our organizers to work very deeply on the feedback forms, because we can move ahead only after we have a feedback. I'm very happy that a feedback form has been generated. I would request all the participants to fill up these forms so that we get your feedback and we can chalk out a further line of action. So let us end with a thought of nurturing our nature for a better biodiversity in future. Thank you very much. Jai Hind, Jai Bharat.
जय हिंद मैडम, कैसे हैं?
जी जी बहुत अच्छी हूँ, आप कैसे हैं? How are you?
I am also fine. Thank you very much for inviting.
So happy to see you, and I'm sure that all our participants will be benefited with your talk.
Yeah, okay, thank you.
Thank you so much, and heartiest welcome.
Okay, thank you.
Thank you, Madam, for wonderful lecture and highlighting biodiversity in terminology of hippo. That is really wonderful. And you have given emphasis over the recording of the species. We would be happy to inform you that department is already engaged in discovering new species and recording new data. And after this joint venture, we could explore more in different portfolio. We have already planned to have a visit of JSI in next year, in the beginning of the next year. So we can make you assure that we will put our best effort to recognize the biodiversity and will encourage our young scientists in this area to build up the biodiversity conservation. And this is the time now for the vote of thanks. While thanks is a very short word, but I would request Dr. Anzum Rizvi to place vote of thanks.
Thank you, Dr. Shweta. So now, before we begin our webinar, we would like to conclude our inaugural session with vote of thanks. So good morning, everyone. Honorable Vice Chancellor, Dr. Harisingh Gour, Vishwavidyalaya, Professor Nilma Gupta, Honorable Director, Zoological Survey of India, Dr. Dhriti Banerjee, respected Dean and Head, Department of Zoology, Professor Harisingh Gour University, Professor Subodh Jain, respected Head of Office, ZSI, Dr. Gurpada Mandal, respected Officer in Charge, Technical Section, Dr. Souvik Sen, respected Joint Coordinator, Professor Vishwavidyalaya, distinguished speakers and dear participants. Gratitude is the blossom which springs from the soul that makes happy both the giver and the taker. It's my privilege to propose a formal vote of thanks on behalf of Dr. Harisingh Gour Vishwavidyalaya and ZSI, an entire fraternity of both collaborating institutes. Success cannot be achieved without the proper guidance, and in this regard, sincere gratitude to our two dynamic personalities, Professor Nilma Gupta, Vice Chancellor, and Dr. Dhriti Banerjee, Director ZSI, for encouraging us to organize this seminar. Thank you, Madam, for your guidance and support throughout, and also for delivering enlightening presidential and inaugural address for the younger generations. We are thankful to Professor Subodh Jain, Dean and Head, Dr. Harisingh Gour Vishwavidyalaya, Dr. Gurpada Mandal, Head of Office, ZSI, Dr. Souvik Sen, Technical Section for the support in organizing this webinar. We are also thankful to our distinguished speakers, Professor S.K. Malhotra from Allahabad University, Dr. Rajendra Kumar from IRI New Delhi, Dr. Subramaniam K.A. ZSI Chennai, Dr. Umar Sharif, BSI Coimbatore, Dr. Umesh Tiwari, BSI Kolkata, Dr. Kamal Khan, ZSI Kerala, who have taken out time from the busy schedule and agreed to address our younger generation to create awareness about conservation of biodiversity. I also take this opportunity to thank Professor Shweta Yadav for a very wonderful and fruitful coordination. We also thank Mr. Vikramjeet for technical setup of the webinar. Last but not the least, I thank on behalf of both collaborating university and institute to the enthusiast participants for registering for this webinar. We are also thankful to the entire team from both the university and the institute who have actively participated in preparation of the webinar. Thank you one and all. Have an enlightening and fruitful day ahead. Thank you. Over to Dr. Shweta.
Thank you, Madam. Before closing to inaugural session, I would request all participants to please open camera. So we will have a photo session with you. Dear participant, you are requested to please open camera. Dear organizers, please have a screenshot of the screen. There are still some of the participants who have not opened their camera. Please click on it.
I'm very sorry to inform that our director has left for another meeting just now to BSI. She had another program, so she has told me to please say on behalf of her. Just she had one important function at BSI also, so it was clashing, but she agreed to first give the inaugural address and then she goes. So she has to rush to 20 kilometers, so she has just left.
Okay, no issue. We will see her next time.
Yes.
So, thank you. Before calling to our next speaker, I would like to request Dr. Ansu, please confirm his availability. Dr. Subramanian.
Yeah. Dr. Subramanian is already there. So, Professor Sweta.
Subramaniam.
Yes, ma'am.
Okay, okay. So we have, shall we start, Dr. Anju?
Yes, ma'am. Dr. Subramanian is already online.
Okay. Uh, Dr. Subramaniam, we welcome you to this webinar. We have a very esteemed speaker to this webinar, Dr. Subramaniam. I am just presenting his biodata. Dr. Subramaniam is a scientist and officer in charge of Southern Regional Center, Zoological Survey of India, Chennai. He specializes in the taxonomy, ecology, and biogeography of aquatic insects, especially Odonata, Ephnoptera, and aquatic Hemiptera. He is also specialized in biodiversity databases and GIS applications in biodiversity research. He has published 8 books and over 130 research papers in international and national journals. He has described many new taxa of Odonata and Ephemeroptera, Hemiaptera, and Socoidea. He has carried out extensive field surveys in different biogeographic zones of India. He is a member of IUCN, Odonata and Invertebrate Species, Specialized Group, and Intergovernmental Panel on Biodiversity and Ecosystem Services. He is a recognized research guide in the University of Madras, and his students work on taxonomy and ecology of different invertebrate taxa. He will be speaking on role of aquatic insects in wetland biodiversity and conservation. May I request Dr. Subramaniam to speak, please?
Good morning. Am I audible to all of you?
Yes, you are.
Yes.
You are.
Thank you. Very good morning. So I will start with my presentation. I will share my screen. Is my presentation visible?
Yes, it's visible.
Yes. Thank you very much. So to the dignitaries, all the respected dignitaries and organizers of this seminar. So today I'm going to talk about aquatic insects and their role in wetland biodiversity and conservation, how aquatic insects are very important. And let me begin with this slide. So if you look at the amount of fresh water that is available on earth, it is very small quantity. Like most of, as you know, like most of the water what we have on earth is salt water, and only a small, about 3% of water is what we are having is fresh water. Out of this 3% of water, only a tiny fraction, about 0.3%, is there in the surface of the earth, that is lakes and rivers and other water bodies. That's what we call it as wetlands. Rest of the water is underground, or they are stored as glaciers or permanent snow cover, which is not accessible not only to human beings but also to the other organisms. So the amount of water that is available on Earth is, which is used by biodiversity or also by human beings, is a very small fraction. And this is very important because if you look at the proportion of biodiversity that is found in this small amount of water related to its extent, it is very, very high when compared to marine or terrestrial ecosystem. If you look at their area of extent and their availability, the amount of water that is available is very less. So when we talk about freshwater ecosystems, it is not just standing wetlands. So the rivers are also very, very important, like wetlands, like lakes and marshes are what we generally, when we say wetland, it comes to that. But the rivers are also very, very important. And in fact, rivers are vital to the landscape. So if you look at the Indian subcontinent, so Indian subcontinent is drained by various rivers, like major rivers like Ganga, Brahmaputra, Indus, or the Narmada, Tabi, or the Godavari, Krishna. So all these are all very, very important rivers that drain the entire Indian landscape, and they sustain the life and the activities of the. So, looking at the biogeographic classification, like the terrestrial, you may be aware about the terrestrial biogeographic classification of the Indian subcontinent. Similarly, the wetlands or the uh, biodiversity or of the rivers and wetlands is classified into 20 regions, so which has a distinct fauna and flora composition, largely based on the riverine ecosystem. So the freshwater ecoregions, what we call it, there is a global freshwater ecoregion classification. So India has 20 distinct freshwater ecoregions, and these 20 regions have distinct composition of flora and fauna. So this is, we have to keep in mind when we are doing research, especially when we are comparing different river basins and biogeographic zones. So each area is very, very distinct. So just to have a glimpse of some of these riverine ecosystems, so you have the riverine ecosystems of the subcontinent is as varied as the land. So you have high altitude rivers in Himalaya, or you have high altitude rivers in Western Ghats in peninsular India. Then you have very fast flowing hill streams in different parts of the country, or you have waterfalls, and you have swamps. There are some swamps called Myristica swamps in Kerala. These are forest swamps like mangroves, but these are freshwater swamps. Then you have lakes. There are several natural lakes and man-made lakes in the.
Dr. Subramaniam, your slides are not moving, and second thing, it is not in the slideshow mode. Can you
Okay.
please make it in the slideshow mode?
For me, it is in slideshow mode.
Okay, but we cannot see. We are seeing the screen like
Can you move? Can you see the slides moving? No.
slides. No, first slide is only there.
Okay, okay, okay. 1 second. I think there is some
Yeah.
problem. Let me do this. Okay. Sometimes this Google Meet this happens.
Yes, sir. Yes, sir. Because of that, because even there is students are also telling that slides are not moving.
Right. I will share.
Or you can reload once again.
Yeah, I'm reloading.
Yeah.
I think this. I think this should be fine. I think. Slightly increase. Is it okay now?
Slideshow mode.
No, it is not. I think it will not come because I'll try.
If it is not in the slideshow mode, then you can continue and increase the size. Then also we can see.
Is it in slideshow mode now?
No, it's not in slideshow mode.
Okay. Okay, then I will. Now is it visible? The slides?
Yes, slide is visible, but it is not in slideshow mode. You can increase the screen size and then you can continue, I think so. Then you will have to move the slides from this point,
Oh,
sides.
yes.
From here, we have to move. Instead of going through the keyboard.
Okay. So this is what I talked about, the different availability of freshwater ecosystems. And so this is the
Yeah.
drainage map of India. Now we can see like different drainages. And this is what I talked about, the freshwater ecoregions of India. So you have 20 different freshwater ecoregions with distinct flora and fauna. So these are some of these pictures of the different aquatic or wetland ecosystems. So this is the high altitude rivers of Himalaya. So now is it moving?
Yes, yes, yes.
So these are the high altitude streams in the Western Ghats. So you have hill streams in the torrential hill streams, or you have waterfalls, or you have these forest swamps called Myristica swamps. Then you have these lakes. This is a Loktak lake, but there are several similar natural lakes and also man-made lakes. So these are some of these man-made lakes and reservoirs. Then you have ponds, largely man-made ponds for agriculture and other activities. Then you have other ecosystems like this, wetland ecosystems. These are transient wetland ecosystems like paddy fields. Why I'm showing all these different kind of wetland ecosystem is that the diversity of these wetland ecosystems vary a lot, and there are species specialized to live in these kind of habitats. Different species are there, and why wetlands are very very important. So because of their ecosystem services they provide. So like if you broadly we can say that this ecosystem services they provide are provisioning services, supporting services, maintaining services, and cultural services. These are the broad wetland services they have. Like provisioning services, like you have fish, or you have the renewable resources, or you have the genetic resources, or supporting services like food web supporting, nutrient cycling, water cycling, or you have cultural. I don't need to mention a lot about cultural services, because most of our wetlands are worshipped, and either river stretches are worshipped, or you have sacred lakes and sacred ponds throughout the year. So culturally they are very very important. So like this is a picture of Ganga Arati. So how the rivers are very very important, I did not tell to you. Or agriculture services, how the wetlands are providing important agriculture services, I will tell. And livelihood. So several wetlands support the lives of millions of people. Inland wetlands, so either providing food or they're providing income and um, income generation. So these are very very important. So this is a background to our talk. So because wetlands are so important in the life and economic activity of the country, so it is very very important to conserve them. So but now coming to the our main subject, that is what are aquatic insects. So you know that insects are the most diverse group of organisms on earth, as the madam already told. So like the number of species that exist on earth varies a lot because of different kind of estimates. So the estimate varies from like 2 million species to 1.3 billion species according to different estimates. But latest or the most consensus estimate is about there are 8 million species there on earth, excluding of course the microorganisms and bacteria. Of this, about only 1.7 million species are named. So remaining so many species are yet to be named. So the diversity is enormous, especially insects. So like terrestrial ecosystems, insects are also found in freshwater ecosystem. So here one important point we have to remember is that there are not many insects specialized in the sea. So the insect diversity in the sea is very very less. Only one insect, one family or one genus of the insect that we can say is purely marine, that is purely pelagic, that is one species of one genus of water strider. Most of other species that is found in coastal area are either in estuary, or they are found in the sandbanks or the terrestrial ecosystem. So most of the insects are found on land, and there is enormous diversity of aquatic insects. So these are the insects that specialize in freshwater, and over 1 lakh species are found throughout the world. So over 1 lakh species in 13 insect orders, these are found. So why like we study these insects? So like they are, first of all, they are very good biocontrol agents. So they are very good control of, there are several species that are predators, and they feed on other aquatic very dangerous aquatic organisms, and they are key component in food web because they support a lot of other higher vertebrates and invertebrate predators as a food source. And they're very easy to study. Like you don't need to have sophisticated instruments, or so they are found everywhere. So it is very easy to sample them and study. It's very good model system for ecological studies or evolutionary studies, and they are very good indicators of ecosystem health. And because the recent studies have shown that they are also very good indicators of changes in climate, so many people have started using them as indicators of changes in climate. So they have multiple roles in ecosystem, and they are very useful. And if you look at their evolutionary history, I'm not reading out all the thing, but their evolutionary history starts from Carboniferous to Jurassic, so different orders. So some of the oldest insects, like the mayflies and dragonflies, were originated during Carboniferous. So from that time, these insects have started colonizing freshwater, and some of them are primarily like they are the first insects to colonize the water and land. So they are like their evolutionary history is very long, and they have this one. And if you look at any wetlands, so wetlands are known for birds, wetland birds, and there are a lot of studies on wetland birds. And all these birds, they depend on aquatic insects as their source of food, directly or indirectly. So most of these birds, and at one part of their life stage, they depend on the aquatic insects for their source of food. So the wetland with lot of very good population of aquatic insects support good populations of wetland birds also. And they are the intermediate linkages in the food chain, especially in the they are the primary consumers of phytoplanktons, and they also form the prey base for the fishes. So they are the intermediate uh, in the food chain, and they contribute significantly to the wetland productivity. So, this, I am not going to elaborate, but I'm just telling you in brief. So, like when a river flows from their headwater to the estuary, so there is a lot of changes happening in the nutrient cycling and also in the entire process, how the entire nutrient dynamics of the riverine system. So, there is a turnover of species from the different stages in the river, from the headwater, that is from the small stream to when large river grows. There is a continuous change in the species community, both in the invertebrate and vertebrate species community. So aquatic insects also change their species composition when they move from the headwater to the downstream. So there is a continuous change in the community. So the diversity also changes from when you start sampling from the headwater to the downstream. So this is very important to remember, because the and there is also change in seasonal changes is also there. So they are very sensitive to these changes in the hydrology and the water quality and seasonality. So they are like each group or each community of the aquatic insects. If you look at the community, you can clearly say in what is the place where you can see this kind of insects. I'll come to that little later. So there is why I'm saying this is that they are very important when you are considering the conservation of the riverine ecosystems and also your wetland ecosystem, because you can track how your conservation intervention is performing, like by looking at the composition of the species community. So other most important thing is that why the aquatic insects are most important is that if you look at the like most ubiquitous and most dangerous aquatic insect is mosquito. So like mosquito, you may not immediately think it as a aquatic insect, but primarily it is an aquatic insect because the larva, they lay egg in water and the larval stages are in water. And if you look at the world's most deadliest animals, it is the mosquito is the most deadliest animals. You may think it is snake or a tiger or a elephant, because that is get more media attention. But if you look at number of people dying every year, more than 7 lakhs people die every year because of the mosquito bite, malaria, dengue, and other vector-borne diseases. So aquatic insects are important not only from the economical beneficial point of view, but also in the terms of looking at human health. Also, they are very, very important. So, but what are the aquatic insect habitats and where they can be found? So, they are found in all kind of wetlands. So, they are found in streams. Uh, they are found in waterfalls. But remember, each of these wetlands have different types of aquatic insects. These are some of the aquatic insect habitats. So, these are stream cascades. So here the species are adapted to live in very fast flowing rivers. So I'll come to that, how they are adapted. So some of them live in tree holes called phytotelmata. So these are very, very interesting habitats. So these are very temporary habitat. During rainy season, the tree holes get filled up with water. Then suddenly whole community of aquatic insects colonize. So they have a very short life cycle. So before the water dries up, they complete their life cycle. So they have very interesting life history strategies because the eggs can remain dormant till the next rain comes. So that is how they remain in the that is how they survive in the dry condition. So there are several strategies these species adopt. So this is a very interesting system to study, the phytotelmata. And there are species, especially in breeding in phytotelmata, like some of these dragonflies, which they live in the canopy and live in phytotelmata. They complete their life cycle there. And there are some species that specialize in other living inside other species, like this mayfly larva, which lives inside the gill chambers of mollusca. So the aquatic insects, they live in diverse habitats. So like even a small tiny water collection can have some species of aquatic insects. So if you look at a wetland, also different places, different part of a wetland is used by different species of aquatic insects. So here, like I've taken just an example of aquatic hemiptera. Here, there are species which live in the benthic form, like in the among boulders and rocks and other things. Then there are species that live in water column, in the swimmers. Then there are species that live in the water surface. Okay. Then there are species that live in among aquatic vegetation. So even within a group like aquatic hemiptera, different species occupy different part of a wetland ecosystem. So how we sample aquatic insects? Mostly by netting. So what we usually do is that we have a, systematic sampling protocol. So like if this is a stream, so we have quadrats in like specified quadrats, like 10 meter by 10 meter quadrats. We collect samples using different types of nets. So if it is a large river, our sampling is much more larger. So we both collect from the river and also from the riparian zone, because several species adults can be found in the riparian zone. Like if it is a pond, then we do sampling across the different wetland part in different places in the wetland at different depths. We do. So this is for rivers. There is a method called kicknet sampling where we use a flat screen with a specified mesh size, and we collect all the insects that is getting washed away in the water current what we do is that we disturb the uh, substrate before the net, and all the insects that is getting trapped in the net is collected. Or if it is a small wadable, like smaller stream, we use it a hand net, like what we use, like something similar to the aquarium net, small net we use. Or like this is also another kick net being used in a shallow stream. Like this is how the once we collect the aquatic insects, they can see them in the net, they will get clinging. So with from this you can collect them using a fine brush, you can collect them, and like you can transfer them to a tray. You also get some other aquatic animals like fishes and crabs when you are doing sampling. So at night we deploy light trap next close to the wetland. So that, and the emerging insects, so that immediately after sunset, several of these adults of aquatic insects come out. So we collect them using light trap. So I'll show you. There are some moths also in this picture, but this is how a light trap collection looks. So this is a light trap collected mayfly. So how do we preserve? We preserve using 70 to 90% methanol in small vials, and we put all the data labels, and we identify the species by using microscope and manuals, identification manuals. We identify. So now let us have a brief overview of the aquatic insect diversity, what we are going to talk about. So if you look at the freshwater insect fauna of India, so like nearly 10,000 species are described from India. Nearly 10,000 species are described, out of which about more than nearly 50%, that is nearly 5,000 species are insects, aquatic insects. So rest of them are mostly crustaceans, annelids, and arachnids, and only small like fishes are also major important. But fishes and birds, but other groups are all small, tiny fractions. So they are going to talk about only aquatic insects, which is about nearly 5,000 species are known from India alone. So globally, more than 1 lakh species are there, but India alone, 5,000 species are known. So if you look at the different groups of aquatic insects, so this the data is continuously updated because every day some new species are uh, described. Some new aquatic insect species are described. So nearly now already the figure is very close to 5,000 species. So this is how different groups. So the highest diversity is found in Diptera. So if you see the terrestrial ecosystem, it is the Coleoptera that dominates in that terrestrial ecosystem. But in aquatic ecosystem, it is the Diptera that is dominant throughout the world. Then there are other groups that is also very diverse, like the Trichoptera, more than 1,200 species are there. Then Hemiptera. So all these species are having very high diversity. So I will introduce now some of these common aquatic insect orders. So this is Ephemeroptera, adult mayflies. So adults are terrestrial. They are called Ephemeroptera because they are ephemeral. Ephemeral in the sense that their adult lifespan is only for 1 or 2 days. Their sole purpose to come out as adult is to mate and lay eggs. Because adults are non-feeding and they don't feed, and they don't have like proper mouth parts. The mouth part is very vestigial, so they don't feed. So they just come out, they molt again, and they have two molts. Like after coming from the nymph, then it's called subimago. Then they have another mold called imago. Then they mate and lay eggs and die. So that is why I call they are Ephemeroptera. So this is a brief life cycle of Ephemeroptera. So the female lays egg in the water, and the nymph develop. So nymph have a very long life cycle depending on the ecosystem or the water temperature. In some cold regions, they may take more than 1 year. Then the subimago will emerge as a nymph. Then they will transform into adult. Then they mate egg and they lay eggs. So this is a very short life cycle of the mayflies. So this each species of mayfly have a very particular larva habitat. So they are very sensitive to water quality. So Ephemeroptera are very good indicators of the water quality. So if you have Ephemeroptera in your in your sampling, so that means the water quality is very good because they require highly oxygenated and uncontaminated water. So they are very good indicators of the water quality. There are several families and species. More than 19 families are found, and more than 230 species are found. So these are some of the morphological variations of Ephemeroptera. Here you can see a diverse morphology. So each species have a particular habitat. So some species are burrowing form. So they burrow inside the sand in the riverbed and they live there. And some of them are clingers. So they cling to the substrate. Some of them are found on the boulders and rocks. Some of them are found under the stone. But all their morphological adaptation is to survive in the water current. So they have very flat body and they have very so that their resistance to water current is minimum. And they have abdominal gills, so the gills are on the back of their abdomen, so that is used for respiration. So they require very highly oxygenated water with low temperature so that they can survive. So they are very, very sensitive to water quality. So these are some of the pictures of the mayfly. So like just to give an idea of how they look and what are their different body parts. To identify to species level, you require a help of microscope. But to identify to family or generic level, you can easily identify by using a simple microscope. But to have a very detailed identification, you require a help of good microscope. So this, like you can see that these are some of the uh, species of mayflies from the different parts of India. Yes. So next comes the Placoptera or stoneflies. So stoneflies are again very less diverse in India. About 140 species are found, and most of the species are found in the Himalaya. So they are highly cold adapted species, but few species, about 21 species, are found in peninsular India. Here also they are all found in the hill streams. You cannot see Placoptera on a pond or a lake. So most of these species are found on the hill streams, very fast flowing and cold water hill streams, and they are very, very good indicators of the water quality. So they are very, very sensitive to water quality, and they are very good indicators of water quality. And the larva is aquatic and the adults are terrestrial. So the larva are predators and they live in water for like more than a year, and they adults come and molt. So they have unlike the mayflies, they have a single life cycle. So the adults come out, molt, and the mate, and adults live for a longer time. So some of the adults are also feeding stages, but the larvae have a long larval duration. So these are some of the nymphs of the uh, Placoptera. So we have several species, and the interesting thing is that unlike the mayflies, where the gills are abdominal, they have gills in the thorax. So you can easily identify a Placoptera nymph by looking at the thorax, where the nymphs are having gills at the thorax. So these are some of the pictures of different types of Placoptera nymphs you can see in our system, Indian species. Next comes the Odonata. It is one of the best studied insect groups like the butterflies. And globally, we have a lot of data. So more than 6,000 species are there globally. Of this, nearly 500 species are uh, reported from India. So dragonflies and damselflies together called Odonata. So the adults are completely terrestrial. So they are found in the very close to wetlands. They are in the riparian zone. Some of them, of course, can be seen far away from the wetlands, deep inside the land. And most of the species live very close to the wetland. And larva is aquatic. So the larval stages vary with the species. So some of them are having very long, very long larval duration. So the adults are terrestrial and larvae are aquatic. There are two suborders in this, that is the Zygoptera and the Anisoptera. And Zygoptera are the damselflies where the wings are folded over their back when they rest, like this one on the right hand side. And you have the dragonflies where the wings are spread out perpendicular to the body. So these are called Anisoptera. Aniso means dissimilar. So the hind wings of dragonflies are much broader and bigger than the forewings. So you have a dissimilar hindwing, that is why it is called Anisoptera. But in case of damselflies, they called Zygoptera. Both the fore and hind wings are similar. So that is why it is called Zygoptera. So this is how the larvae of dragonflies and damselflies look. So both the larvae are adults are predators. They are very voracious feeders, and uh, they they have very the main difference is that the Zygoptera, that is damselfly larva, have something called a caudal filament at the end of the abdomen, and the Anisoptera have some, they what they have is called a caudal gills. So they can respire through their gills, in the caudal gills, and it's also a gas exchange part, and also uh, a adaptation for exchange of the salt. So, and in dragonflies, they use the caudal gills as a mechanism for jet propulsion. So they use this mechanism as a jet propulsion thing to move in the water. But damselflies don't have that, and they are very good predators in the aquatic ecosystem because they can have a very good eyesight, and they catch small aquatic invertebrates, insects, even smaller fish. The large dragonfly larvas can catch even small fishes and feed on, and sometimes they even catch the emerging dragonflies of their own species. So they are highly cannibalistic. So they are highly cannibalistic. They feed on each other if you get an opportunity. And this is how the damselfly larva look, and their most potent weapon is their mouth part. So they have a labium that can be extended, like suddenly, like uh, it is like a what we call it is like the front part of a JCB. So they use that one to catch hold of their prey and also to uh, feed on them. So they slowly follow their prey. When the opportunity comes, they are ambush predators in the sense that they sit on one place and look for the prey to come close to it, and suddenly they catch their prey. So next comes the Blatodia. So we have some aquatic cockroaches, but we don't have much information on them. So this is a lot of potential area to research on aquatic cockroaches. So they are mostly found in wetlands. So they are definitely having important ecosystem roles, but we are not much aware of it. Then there are some other aquatic minor groups, aquatic orthopterans, like the several species of crickets and tetragids, that is this pygmy grasshoppers. They are found in semi-aquatic environment in wetlands, but here also we don't have much knowledge on them. Then comes other less specious and less, but very interesting groups called Megaloptera, alderflies. So we have about 26 species in India. Of this, only 2 species are found in the peninsular India. This group is also highly adapted to very cold environment. So they are found only in streams of very low temperature, or they are found in high altitude, or they are found in streams and rivers of Himalaya. So we have only 2 species in Western Ghats, that too found in very high altitude regions. But in Himalaya, we have several species of them, about 24 species. So this is the larvae of a Megaloptera. So our larvae are predaceous. So larvae live in water. So like the dragonfly larva, they also feed on other aquatic insects and other aquatic invertebrates. So they can be easily identified by the abdominal gills and the spine on their abdomen. So they are very easy to identify in the field. Then comes the Neuroptera. So these are the species of Neuroptera that are adapted to live in the freshwater sponges. So the larvae live in freshwater sponges, and adults are terrestrial. So we have recorded about 19 species in India, but we do not know the full ecology of these species. So these are also very interesting groups. So then comes the one of the major groups of aquatic insects, what we call it as aquatic bugs. So these are hemipterans, aquatic hemipterans. So the species like of aquatic hemiptera are very unique in the sense they cannot breathe in dissolved oxygen. So what they have to do, they have to come to water surface frequently to take atmospheric oxygen. So they have so evolutionarily these are all secondarily aquatic insects, because their evolutionary ancestors were primarily terrestrial, and later on they came to water for uh, later on they came to water, and they have morphological adaptations to take water from the surface of atmospheric oxygen. So they have body hairs which trap the gas bubble, or they have siphons at the end of the abdomen, and which they use it like a snorkeling device to take atmospheric oxygen. So we have like about 13 families of aquatic bugs are there, and all of them are predators. All of them are predators. So they like Geridae, Velidae, Hydrometridae. So these are very common aquatic bugs, and you can see them in all the kind of most of the kind in wetlands like the ponds, lakes, and reservoirs. You can easily see them. Even some of them even occupy the your garden pond and other. So these are some of the common aquatic insect aquatic bug families. Like Belostomatid is a large aquatic bug, we call giant water bug, and it is economically very important in the northeast because it is consumed as a food item. So like you can see them like people consuming these Belostomatids because they are very large. Like it is about 6 inches they grow, and they are very fleshy. So people consume these aquatic insects. So these are some of the common species that you can see in our wetlands. So these are ubiquitous, or you can see them in throughout the most of the wetlands, even in paddy fields. So, and they are, as I told, most of them are predators. So they play an important role in controlling the populations of several aquatic uh, insect species like mosquitoes. They feed on extensively feed on aquatic larvae of mosquitoes, and these are other aquatic common species. So some of them are having interesting biology, like this Diplonicus annulatus. So what happens is that they have a very interesting parental care mechanism where the female lays eggs on the back of male. So after mating, the female lays eggs on the back of the male, and it is the responsibility of the male to carry the young ones till they will be carrying their young ones till they hatch and till they grow to some certain size. So you can see frequently see this in the field that the Belostomatids males are having cluster of eggs over their back. So this is very interesting parental care behavior they have, males especially. So though like these are all microscope photos, so they look very clear and very distinct. But when you go to field, you see them that they are very cryptic. It is not very. They are very well camouflaged in their aquatic habitat, and it is not very easy to locate them. By track by using different types of methods, by you can easily spot the legs, and they use water surface attention to live on the water surface. So some of them like these Lacotryphus, they live under the water, and they use their long siphon at the end of the abdomen or renatra and the end of the abdomen to survive in the water. So this is another species, Cylindrostetes. Some of them are found in large numbers. So some of them live in water under the stones and rubbles in the water. Next comes another species group. Species diverse group is more than that of the uh, bugs in the water in India. These are aquatic coleoptera, so they are very diverse group. More than 700 species are found in India, and many of them are predators. So some of them can here you can see them feeding on a frog, a skitter frog. So some of them are highly predaceous. They feed on diverse aquatic organisms, and some of them are uh, feed on vegetation also, and some of decaying organic matter. So some of these common species like this whirligig beetle are very interesting because you met interesting morphology. Like they have two pair of eyes. So each, like most of the insects have only one pair of eye, but these whirligig beetles have two pair of eyes, one on the top and one pair on the top and one pair on the bottom. So simultaneously they can see under the water and above the water. So this is a very interesting neurological uh, mechanism also, how they are simultaneously processing the information. So they are very alert, so that they can see predators coming, or also they feel like prey, aquatic prey, and they can also see how the predators are coming. So they are very interesting in that sense. And if you look at their morphology, they are very streamlined, and they are like, it's like more like a boat. So there is with least resistance to water. So there is no body is very smooth, very oval shape with very smooth surface. Even the legs are modified for swimming, so they cannot walk on the water. So their legs are like an oar, and they are very highly specialized to live in water. So these are another group of aquatic beetles called Diticidae. They are predaceous, and very good potential in mosquito control and especially natural wetlands. They are all species of Diticidae, and they have other interesting, like these groups having another interesting mechanism. So they have what we call it as the suction pads on their forelegs, or adhesive pads, like what we have these vacuum like vacuum hangers which we use in our walls to hang things. So similarly, they have these vacuum pads which they use it to hang to the substrate in wetlands, especially in streams, very fast flowing streams. So they have very interesting morphological mechanisms. So some of these larvae, like this is a so their larvae are modified into a disc, like they are called water pennies. They may not very first streams you can see them. Then comes this aquatic Lepidoptera. So you have like very few aquatic Lepidoptera, mostly pyralid moths. So they live in the water, when some of them live in the very fast flowing streams, and they use silk to survive in the water. They make a case, like here you can see that white patches on the rocks are actually larval cases. They live inside the larval case. They make tunnels of this silk, and they move around there, and they feed on aquatic algae, especially many small aquatic algae they feed. And they are also, some of the species are also agriculturally very important. Then comes the caddis flies or the Trichoptera. So this is also highly specious, like the like beetles and Diptera. The caddis flies are one of the most specious groups in wetlands, and in like they are found in all types of wetlands, but high diversity is found in streams and rivers. More than 1,200 species are recorded, but still many more species are being described from India. So the larva is aquatic and adults are terrestrial, and the larva has very interesting mechanisms. The very interesting aspect of this is that by looking at the larval case, we can identify even a genus. So each genus have a very distinct larval case. So here there is a larva to catch the small debris and other small aquatic organisms, and it held together. So that we call these aquatic things called genus. And they use even stones. So this species use stones to lead. Stone is used as a weight so that they don't get carried away by the water current. So like other, so in the insect it is the Trichoptera larvae that has the to create, they can evolve to have silk. So they have silk glands. So the silk gland pebbles and stones, and they also make an anchor with that, and they move around the water and they feed on the everyday uses.
I think there is interruption from Dr. Subramaniam's side. We lost him. Dr. Anjum, we can see that many of the speakers are interested to have his presentation. Dr. Anjum, could you please speak to Dr. Subramaniam and get the updates?
Hello, Dr. Subramanian.
Okay, there was some.
Do you have any question?
Yes, I will read.
You have.
Can you tell me what's that? Hello.
Hello, participants. Participants, if you have any question, you may please
Can you
ask
hear
Dr.
me?
Subramaniam. Yes. Hello. Participants, can you hear us?
Can you hear me? Can you hear me, Dr.
Yeah,
Anjum?
I can
Yes,
hear
sir.
you. Yeah, I can hear
Yes,
you.
sir. You're audible, sir.
Yes,
Sir, your presentation was gone in between, so I thought it is over. It was
From
not
which
there.
place can you tell me?
I think so it was larval caddis flies. That slide is there. Slide number, it is 44. So actually I was on phone, then suddenly I saw that means slide number 78. One person is saying 78.
Okay.
So after 78, I think so you have to continue. Because your presentation has already. Yeah, 78.
Okay.
Please start from 78 slide number.
Okay. So the new stream. The aquatic, they treat hill streams and other wetlands, and they have very good by looking at the larval cases. So they each of these family or the genus have distinct larval case morphology, and this can be used easily. You can identify up to genus level what this case, what this larvae is. Like here, you can see uh, the the larval case of Astinopsychidae larva. So they make like basket-like cases, and with their silk. So this like the Lepidoptera, the Trichoptera larva also produces silk, and with the silk they construct this basket-like net, basket-like net, and they collect all these organic debris and other small organisms that is coming in the water current. So they keep the this net about the case, what we call it as larval case, against the water current, so that it is opened up. And there are some other like the Hydropsychidae larvae, which uses the stones, small stones, into they use the silk to scrape all these small stones, and they live in water, and they use this as a ballast, in the sense they to add weight to the larvae, so that they don't get carried away by the water current. So, but they also get anchored by the silk. So these are some of these adaptations they have, and some of these Lepidostomatidae they use as small pieces of the woody debris or the leaf bits to make box like larval cases. So which they live in the water, and you have some of these like Liminophilidae, which uses the small pebbles and small stones they use to the this one. And there are other interesting larvae also, Mesocentropus, which uses two pieces of leaves and leave the larva live inside this water in the so the leaves which is falling on the streams, these larvae collect and they use two leaves to stitch with and use silk to stitch them, and they live inside this one. Or there are some free living Trichoptera, other species like Helicocycle, which uses a helical, they like a snail. They use small stones and small pebbles to make helical case. Or there are this Goura, which uses the like one small pebbles in the center and large pebbles on the sides. So there are Goiroides, which this you can see in the tree holes also. This is also Phytotelmata living caddisfly, and they uses the small box-like uh, with the leaves. They make these small box-like cases. So each group has a distinct this one, and this Trichoptera play an important role in wetland ecosystem because many of them are like they are very important in nutrient cycling, in the sense they process the organic material that is coming into the stream, like the uh, uh, pieces of the leaves, pieces of the woody debris that is coming into the streams are broken down into smaller bits by the groups like this, many of these Trichoptera, and upon which all these microorganisms start acting another thing. So this Trichoptera, Ephemeroptera, these groups feed on different kind of organic material that is coming from the riparian landscape. Then you have very minor aquatic groups like this aquatic Hymenoptera. These are the parasitoids of aquatic insects. So we know very little about them. So only about 10 species are known from India, but there may be more species, but we are not having much information on them. Then comes the largest group, the Diptera. So more than 6,200 species are known, and India, we have more than 500 species of more than nearly 600 species are there in aquatic and semi-aquatic, and they are very, very important economically in the sense that many species transmit diseases, both for in human beings and also in the our domesticated animals. So economically, they are very important. So a lot of studies are there on aquatic Diptera. So some of them are found in large swarms, like chironomids. Like sometimes you can see them in huge swarms, and some by people mistakenly consider them as mosquitoes, but you can see them in huge swarms of chironomids, and they are very important in the sense they also support good food chain. And like species like a black fly, Simulium, they transmit diseases like onchocerciasis or river blindness. But fortunately, though we have several species of black fly in India, they don't transmit disease. But in Africa, they are a major vector of onchocerciasis or river blindness. But though India, it is a very nuisance pest in the sense that they bite, especially in Himalaya, they bite like very not when we do field work or we are outside, especially during monsoon, they bite very badly. Then you have mosquitoes. There are more than 330 species of mosquitoes in India. Of this, about 15 species are very important vectors of various diseases, both malaria, chikungunya, and dengue. Like these are very, very important. So studying on these aquatic diptera are very important. And tabanids, there are several species of tabanids that they transmit diseases, like especially in the cattle and sheep, like this Sura disease or the babesiosis. So all these diseases are transmitted by the aquatic dipterans, and they have very, very interesting adaptations to live in water. So like this example, this Blepharaceridae or net-winged midges. So these are larvae of Blepharaceridae. Here you can see there are some species in the Western Ghats and also in the Himalaya. So they live in very fast flowing streams, and they have suckers. All these round things you can see at the ventral side are suckers, vacuum suckers. They are found attached to the very fast flowing streams in the rocks and substrates. So they have very, very interesting adaptations to live in water. they are net wing limitations. I already told you, live in like they have silk. They are filter feeders, so they feed on very small organic material that is in the water, and they play an important role in the nutrient dynamics of the streams. And they have silk, so they get attached to the substrate using their long silk thread. Then you have chironomas. So chironomas are bloodworms. So they are mostly indicate pollution, drain flies or the other polluters of the polluted water. Then comes Tipulidae. So this family has 1,200 species in India. The Tipulidae. So Tipulidae are also very diverse. So they are very, very important because they feed on decaying organic matter in the streams, and they process a lot of organic matter. They are called daddy longlegs. But now coming to the so this is a brief overview of the aquatic insect diversity from different groups. So now coming to the next part of my talk, how they are important very in the terms of conservation of wetlands. So just to as a bridge slide, so we like either it be aquatic insects or other aquatic organisms or even birds. So the important thing is that we collect a lot of primary data from field observations and museum records. Then that is identification descriptions, and that is being used as a knowledge product to understand the ecology and other things. So these all feed into the our policy and action of biodiversity conservation, whether it is wetland or anything. And this this is very important because our primary focus should always be on the understanding the system from a field perspective. So understanding how these organisms interact and how these ecosystem functions, how these are contributing to ecosystem functions is very, very important. And for that we require a lot of studies, field studies, and we are trying just beginning to understand in Indian system how these things are mutually linked and how these things are contributing to the overall biodiversity and the overall ecosystem function of the wetlands. So, like the human footprint on wetlands are huge. Like I need not elaborate on this because of the most of the wetlands are highly stressed, and they are contributing. Human activities contributing significantly to the decline of the wetland. So, like we are in the consumption. Like earlier in the today's discussion, there were mentioning of the how what are the possible drivers of the biodiversity or ecosystem loss. But recent studies have shown across globe that it is the consumption pattern of our species like human beings that is driving the that is driving the ecosystem decline or even biodiversity decline across the world. So the amount we consume as natural resources at current rate we are consuming about 1.6% of the biocapacity of what is being produced. So the amount we are consuming is that annually we are consuming more than what is being produced by the earth. So that is, so we are in a declining phase. So that is what the global studies have shown. So this is impacted the wetland ecosystems also. Like if you the global studies have shown that the biodiversity of riverine ecosystems across the world is declining drastically, especially in the highly populated and highly industrialized areas, like especially in the Western Europe or the Indian subcontinent or the eastern part of China or eastern part of America. There is a significant decline in the biodiversity of the riverine ecosystems, and this has led to the uh, scarcity of the water resources, especially in the Indian subcontinent. And in fact, the our Indian subcontinent, many rivers are in such a bad state that it is worse than the sub-Saharan Africa. So many of our river systems are highly stressed and highly deactivate, highly polluted or highly impacted due to human activity. So, like this is largely in Indian subcontinent is largely due to the different kind of stressors we are having. One is the big hydroelectric projects which dries up the river periodic flooding. So the hydroelectric project, you may be aware that when there is a peak generation of electricity, all the water will be released into the riverine system. So there is a in a daytime when there is no peak demand, so water will be less or it will be totally stopped. So there's a dry period. Then at night when there is a lot of peak demand, so water will be released. So periodic cycling and drying of the riverine system totally wipes out the population of diversity, any organism. So this such kind of rivers you cannot even see a small fish. So because when it happens, then there won't be any. So these are some of these impacts of the hydroelectric projects. Then you have mining, open cast mining in several of these in the headwaters, so which also impacts all these heavy metal contamination and other thing. Then, or you have this riparian deforestation. As already mentioned that several of these aquatic maturity and the growth. So the riparian ecosystems are very like deforestation is another important aspect. Or you have this huge infrastructure projects without taking into consideration of the physical properties or geographical properties of the landscapes goes into the stream. Or you have pollution, like especially in hill streams where there have tourism activity, there's a lot of pollution due to tourism. Or in urban areas you have all the sewage drains into this one. This is another picture of the same. Or you have intensive plantations in the in the headwaters, because all these agricultural runoffs ends in the riverine systems like pesticides and other agrochemicals. Elaborating. So then next comes is this invasive species. There are several species of invasive plants, aquatic plants that is impacting, especially this icornia. Then you have now problems of mycenia entering into streams and covering up the entire streams. So these are some of these this ones. And how we use breaks or aquatic process. So they have to be very sensitive to the systems, and capable are relatively independent of sample size, and they are very easy to sample and cost effective. And these are some of these methods by which you can these are pre-qualities of the indicator species which we use. And like there are different methods by we use aquatic insects as an indicator. Either you can use them as bioassessment, like based on functional structural aspects of biological community. So this is entirely field-based studies. Or you have biotoxic studies, toxicity assess bioassays. This is laboratory-based methods. Or behavioral bioassays. So you can see the sublethal effects of toxins, how these are exposed, what will happen to them. Or bioaccumulation studies. So the toxicity bioassays or behavioral bioassays or bioaccumulation studies, you require good laboratory facilities, so which cannot be done everywhere. So mostly if you want to do wide scale, like large scale studies involving students and other general public, we support the bioassessment, where globally this is very well accepted and well recognized, and there are several studies and several countries are regularly doing this bioassessment of wetlands using aquatic insects. So you here what you are seeing that you are with change in habitat, you see the change in species composition. Like example, appearance of a tolerant species, pollution tolerant species, or disappearance of pollution sensitive species or number of species, change in abundance of species. So by looking at all these things, you can assess the water quality or the wetland health. But there are, of course, limitations also. It is not that it is completely limited. So you have uh, you have these are not substitute for physical or chemical methods, but these are supplement to the physical chemical methods, and you can do cannot differentiate between what is happening due to natural or due to anthropogenic effect. So for that you need to have very controlled conditions. But advantage is that it will tell quickly what is the overall ecosystem health, and it is very easy and economical to sample, and it will also indicate long-term effects of the disturbance in the ecosystem. So these are some of these advantages which we use, and globally there are several studies which support this aquatic ecosystem. So then there is something called uh, biomonitoring working party score system. So this is very frequently updated, and here you don't need to have species level identification, and family level identification is enough, and scores of individual taxa is well studied. So they have done a lot of studies to identify what is the tolerance limit for different families, environmental tolerant limits for families, and a standard table is prepared, and they have scoring system. So like you have a scoring system or a number of scoring taxa. They have developed a scoring system, and based on that one, you can easily assess what is the bio health of the ecosystem. So say for example, I just given one example, like high score means they are very sensitive to pollution. So they are this species. If these families are found, that means they are the indicators of the very pristine aquatic ecosystem. Okay, so these are the some of these methods, and you have mean like low scoring means they are very highly pollution tolerant species are, like for example Chironomidae or some oligochaetes. Oligochaetes are annelid worms. So these are all having low they are very tolerant to pollution. So they can see them in like sewage kind of water. So if you are getting highly polluted water, some of these species will appear. So based on this scoring system, you can identify. So I will stop here with this, and I thank you for opportunity, and I'm very sorry that I couldn't finish it in time. So I can take some of these questions.
Thank you, Dr. Subramanian. You have given a wonderful lecture. Many
Thank
of
you.
participants are asking for your PPT presentation.
Okay, no problem, no issue, madam. I will share it with the Dr. Anjum. I
Okay.
think
All
Okay.
right.
Okay.
Uh,
Thank you, Dr.
I'm very happy
Subramanian. It
that
was so
Dr.
wonderful.
Manpreet is there. So he's a leading expert on Trichoptera in India. So I think he's the only expert now. He's also from ZSI.
Actually, sir, your lecture is so enlightening and so full of knowledge that students are eager to read it again and again. That's why they have requested in the chat box that we want it for our
Yes, yes.
increasing
I will share.
knowledge of
I'll
the
share with you.
students.
You can distribute. No problem.
So, okay, thank you so much for giving your time and really enlightening our students and generating the interest in our students in this research. So, thank you so much for your
Thank you. Thank you.
effort. Thank you.
Thank you.
uh,
Thank you. Thank you.
we can take one or two questions.
Yes.
Dear participant, if you have question, one or two question we can take.
We have lot of appreciation in the chat box for you, Dr. Subramanian.
Thank you.
Yes, please. Any question?
Sir, good afternoon, sir. May I ask a question?
Yes, yes, please.
Sir, I am Manigandan from Chennai, Loyola College. And my question is, I don't know whether it might be a question or not. I just felt and I'm going to ask. As we biologists or scientists, we know the role and functioning of insect or any other flora and fauna. We know how can we involve the normal people to conserve the insect and the flora and fauna, any other?
See, only thing is that only by awareness and education you can do that. Okay, so like we may be aware that before 1972, when this Wildlife Protection Act was there, people are killing lot of there were a lot of hunting everywhere throughout India. There were a lot of hunting there, and like people were hunting, and a lot of wild animals were like really population had destroyed. So after 1972, like globally there were a lot of awareness on wildlife conservation after 1970. I'm saying, I can say, and like slowly by people were aware that okay, we should not hunt, we should not kill animals, and we should. It happens gradually, it doesn't cannot happen overnight, and we have to do continuously. Like people will continuously talk about, people will be awareness through media, paper, and local languages, songs. So like it's like a cigarette smoking, no? Like people were like earlier, if you remember, like I don't know what generation you belong to, but like one some of us will remember that during our college days, like all were smoking, all our many of our classmates were smoking. So smoking was not considered as a bad thing. So in fact, it was a style statement, and slowly like awareness creation, it happens over generations. So like that, our environmental uh, commitment or environmental conservation is unlike police or military. It is entirely a government thing, no? Like the home security or national security or a home security is a police or it's a government responsibility, though we have small role. But environmental conservation is a participatory thing. Both the public and the government have to work together. It is not that government is giving some law, then it is law. It is not like that. You also have to do your part. Like small things you can do. You should not use pesticide at your home. Okay, small, like you cannot, simple thing as
Sure,
that
sir.
you should not use the pesticide in your garden. So that is a simple thing. You can put some like for example, we are now using lot of exotic plants which is not used by our insects. Like you can use some butterfly food plant in your home. Okay, at least one species can grow in your home. Some one butterfly you can grow in your home. Like you can don't need to put like all these exotic lawns, no? Like you can see that lawns are there that is not used by even a grasshopper. Okay, like people put weedicides, like if something like they don't weedicides there. So these are small things everybody can do. That is how you can put a lot of food plants that will encourage a lot of insects to come.
Sure, sir. Thank you so much, sir. Sir,
And actually, like you are like Layolo campus place, like you have huge campus, you can have a butterfly garden.
yes, sir.
Yes.
One more question.
Good
Yes.
afternoon, sir. Thank you for enlightening us, sir. I have a similar sort of question. So while these you have mentioned in your slide about the wetlands, these wetlands are highly biodiversity regions, high biodiversity regions, and continuously we are facing the problems by the people about the encroachment of the wetlands, particularly. While it is not worth to mentioning here, but I am from the Jammu and Kashmir region, and while saying that particular region around the Dal Lake, uh, day in and day out, the government is trying to demolish that structures, but the people are continuously encroaching it. So how can we communicate, or how can we talk the people to lessen this encroachment? Secondly, sir, you have mentioned that we cannot spray pesticides in our gardens. But at the large scale, when we see these insects damage the crops. So how to deal with that?
Okay. So you are asked two questions. One is the wetland encroachment. No? The other one is
Yes,
about.
sir. Yes, sir.
So see wetland encroachment is not just a problem of your place, but it is throughout the world. This problem is
True.
there. Okay.
Yes, sir.
So only by, as I told, only by awareness, and you have to give other opportunities also to the people. No? Like why people are encroaching wetland is that they are not having any other livelihood options. Okay, they are not having other source of income. Most of them will be either poor fisherman or poor farmer who don't have other options to live. So only by dialogue and giving them alternate, like they can be part of tourism, they can be given other
Yes, sir.
employment opportunities. So actually, fishing, all those things are uh, are part of the wetland ecosystem. So like they also have to utilize these wetland resources. Then only it will be conserved in long term. Like we cannot have a very Puritan approach. No, like you cannot fence it and keep it. Like not just for beauty. The biodiversity resources has to be used. The best way to conservation is by eating. Like the other when the whatever people are eating, those things are conserved. Like people will conserve like vegetables and other things are like it will grow. So these can be resource. These are renewable resources. They are not non-renewable resources. So need to be used. And the second part you are saying pesticides. You know, like pesticide use is a large and large scale pesticide use that everybody knows it has to be reduced to a substantial level. And people are also like there are new generation pesticide that is constantly coming, but uh, constantly it is happening. And if you look at there are pesticides which is becoming lesser and lesser harmful, like new generation pesticides are coming, but they are also harmful to insects. And people are also, it is not that people are not aware about it. Government agencies are aware about it, and even the scientists are aware about it, but it has to be reduced to minimum. What is not happening is that people wanted quick results. So like when we are saying that you have to spray only this much amount, but people are spraying 3 or 4 times more than what is required so that they get quick results. So like they are saying that like I have seen
But
farmers who are saying that okay, see, I am spraying pesticide, even bird is dying, so the pesticide is very good. So
but
like so that is the thing. Like like it is like taking medicine. No, like the doctor is saying you should take this much dosage on this date, but people take like continuously they take antibiotics. So pesticide also we need lot of
but
awareness.
sir, there is a problem that these insects have developed resistance against
Yes, that
particular
is
sort of,
that
yes
is
sir.
the that is the
That is why sir people are going for higher doses. They
yes.
are leaving behind that recommended dose.
Then also I'm not
Yes sir.
elaborating, but what happens is that there is also spurious pesticides are there. Okay, it's not the correct one. Quality pesticide, low quality pesticides are there. So they have to spray more in the market. No, like the market in our system. So you are saying that this much concentration is there, but it is not actually there. So the label says something, and inside bottle something else is there. So they have to use more pesticides. All those kind of things. I think I am not
The-
going to because elaborate because
Yeah,
other
now
speakers are also.
Thank you sir.
Yes, yes.
Thank you sir. Thank you sir.
Thank you, Dr. Subramaniam. Dear participants, if you have any more question, please put in the chat box that will be communicated to Dr. Subramaniam, and the answer will be forwarded to you. We are not in position to take more
Yes, yes.
questions. Thank you, Dr.
Thank you. Thank
Subramaniam.
you. Thank you.
thank you, Dr.
Thank you.
Subramanian.
Now is the time for our next speaker, Dr. Umesh Tiwari. He is here. Dr. Umesh Tiwari is from Botanical Survey of India. He is known in angiosperm taxonomy, medicinal plant conservation, ecology, and ethnobotany of the country. He worked on systematics and ecology of Barbadariaceae in Uttarakhand state. He had post-graduation in angiosperm and phytochemistry botany. 52 research papers, articles, 2 books are in credit to his account. He described 5 new species to science, 12 new records for India, and several new reports for the state. He worked across India from Leh Ladakh, Himachal Pradesh, Uttarakhand, Rajasthan, Gujarat, Arunachal Pradesh, and many more states he worked. He had a very beautiful biodata. Now I would request him to deliver his lecture and enlighten us on the plant diversity of Arunachal Pradesh and ecosystem services. Now it is over to Dr. Tiwari. Dr. Tiwari, please unmute yourself.
Thank you, madam. Good afternoon to all. Thank you, madam, for a brief introduction of mine. And actually, I have little time to discuss here because I have to attend one meeting with the director. So I will conclude my talk within a short period of time. And let me present my slide, ma'am.
Yes, please. You have 45 minutes. You can sum up in 40 minutes. So we will catch over time. Okay. Yes, your slides are visible.
Ma'am, is it moving?
It is not moving. It is displaying the first slide, plant diversity of Arunachal Pradesh. Please go on the next slide so I could assure you.
क्यों नहीं जाता यार ये? नहीं, प्रेजेंट तो किया था मैंने. entire screen. Yes. Now share. Select कीजिए. Yes. Share. Now. ये जा नहीं रहा है ना. इसको फुल स्क्रीन हाइड करके फुल स्क्रीन करना है ना. बस. Ma'am, my slides are moving.
Yes, yes. Now it is perfect. Please go ahead.
Good afternoon, all. Now today I have to talk about the plant diversity of Arunachal Pradesh and ecosystem services. As we all know, the Arunachal Pradesh is very rich in its floristic diversity, even in faunal diversity of India, and it is also one of the place which is found in the biodiversity hotspot of the country. So next slide is that what is plant diversity? Actually, first we should understand that what is plant diversity and why it is important. And we all know that the plants are the primary producer, and they are very important. They play a very important role in this ecological pyramid. So what we want that the plant diversity is a part of a diversity, and it is a critical health to the ecosystem, and this is plays a very important role in regulating the natural process and growth of the cycle. That means that if one part of the natural environment become too dominant, then other has to balance it, balance back to the system. And if you see that the diversity of plant life exists for many reasons, why it is existing in particular region, that if you talk about the Arunachal Pradesh or Western Ghats, where the plant diversity is more. But in central India, if you go, that in Gujarat, Rajasthan, why it is not available in that much diverse condition? Because based on the condition of the particular area, and the environmental factors are also affecting on the distribution of plant. But in Western Ghats and this in the eastern Himalayan region, if you consider the eastern parts of India, that there rainfall is more, and it's favorable condition that there the plants are growing in lavishly, and the diversity of the plants are rich in this particular area. And because of this adaptive nature, they are more in that particular region. And these are the things which are because of that the plant diversity is more in this particular region. Now if you look at this map, then this in this map you can see that the India is having some of the areas, Himalayan region are having a green patches. Then in northeastern region you can see the green patches are there, little bit in central India, eastern Ghats, and then along the coastline of western India you will get the western Ghats. There the maximum green portions are there. That means we have this much forested area, and good forest covers are there in this particular region. And why this is that India having this particular region is that the India is divided into 10 biogeographical zone based on this particular because of this biogeography of that this thing, that the first one is trans Himalayan region, which is found above 4,000 meters. Just it's like a cold desert area. This is found above in Jammu Kashmir, some portion in Himachal Pradesh, then Uttarakhand. It also it is available. Some part of Sikkim it is available. Then the other portion is the greater Himalayan region, that is 2A, 2B, 2C, it's like that. It is found in the Himalayan region. And then the third one is the Gangetic plain, which is from Uttar Pradesh, Punjab, the floodplains of Ganga. We can see that this particular area is divided into Gangetic plain, covers the up to Bihar, West Bengal, some part of the West Bengal. Then we have a Deccan Plateau in central India, which falls up to the this between Western Ghats and Eastern Ghats. And then we have the two region, that arid region and semi-arid region. And then next one is the coastal region of the India, which is having a very much diverse thing. And this is coastal region. Most of the mangrove plants are available from right from the Gujarat up to the Sundarban. So this is the important area where the mangroves are found, and these are the green patches which are shown in the map. You can see that these are the important phytogeographical diversity of India. Most of these species are lies in that particular region. Then we can see that the next one is the four bio hotspot of India is the Himalayan is considered as the hotspot of India, and then except Arunachal Pradesh, other part of the northeastern state like from Assam, Mizoram, Manipur, Nagaland, Tripura are the Indo-Burmic region of the hotspot. And then Western Ghat is also third one is Western Ghat, which is considered as the hotspot of the India. And then next one is the Sundaland, which is Nicobar Island, which is comes under the hotspot region of the India. Then if you look at this table in the right side, one table is given. This is the floristic diversity of India compared to the world. There are gymnosperm, angiosperm, bryophytes, pteridophyte, virus, algae, fungi, lichen. These are the groups of the plants which are available in India. And if you see that the gymnosperm throughout the world, it is 1021 taxa are known from gymnosperm, and in India it is represented by 82. That means 8% of the world gymnosperm flora are present in India, and there in among these 82 species, 8 species are endemic to India, and these 7 species among these 82 are uh, considered as a rare and threatened species. Their population are declined. The next one is the angiosperm. If you look at the angiosperm, there are 268,600 angiosperm taxa are recorded, almost around now 3 lakhs. We can say that 3 lakh angiosperm taxa are reported from angiosperm and in the world, but India represent only 21,984. That means it is also coming around 8% of the world floras are present in this country. And in this, among these 21,984 taxa, we have 8%, this 4,102 taxa, which are strictly endemic to India. So that means, and among this 1,700 species are assessed as a threatened species for India. Then if you talk about the bryophytic floras, then throughout world 16,236 species are reported from throughout the world, and in India it is represented by 2,800 taxa. And when you compare that, the 17.25% of the bryophytic floras are present in our country, and there are 629 taxa which are endemic to the India, and 80 species of bryophytes are also assessed as a threatened and endangered species. Then pteridophytic flora, if you talk about, there are 12,000 plants are present in all around the world, and in India 1,314 species are present. Taxa, we can say 1,314 taxa are present in India, and this is also covering 10% of 10.95, and that is 11% of world flora. Pteridophytic floras are present in our country, and then 47 endemic species among this 47 species are endemic to India, and 414 taxa of the pteridophytic taxa are reported as a threatened and endangered species. Then the talk about the algae, this is the number actually. 40,000 algae are present in all over the world, and among this 9,008 species are reported from the India, and this is also in India 22.52% of the world algal flora are present in India. Then 1924 species which are endemic to India, and then talk about the fungus, 98,998 species of the fungus species are reported from all over the world. Among these, 15,602 species are found in India. And that is 15.76% of the fungal flora are found in India. And then 4,100, among these 4,100 species are endemic to India. Then and 580 species are threatened category of this fungus species. And then lichen, if you talk about, 17,000 species are recorded from all over the world. And then in India, in 2,989 species are from India. That is 17.58% of the lichen flora are also found in India. And then 520 species are endemic to India. But for the lichen perspective, if you see that most of these species are not assessed, and then assessment need to be done. So, so overall, we have in floristic diversity, if you consider, then we have 55,048 taxa are present in India, and this is the chart which we can see in the down. If you look at the floristic angiosperm diversity of India, how these angiosperms are distributed throughout the country in different states, then Tamil Nadu represents the maximum diversity of angiosperm found in India, and it stand first. And then later on, after that, Arunachal Pradesh is having a 23.62% of the Indian all the angiosperm flora represent in Arunachal Pradesh, 23.62%. Then let after that Sikkim and Kerala, Karnataka is like that. Go on. So maximum diversity, if you consider the maximum number of species, because of this Western Ghat, Tamil Nadu having a maximum this coastal region, and then other thing. So that's why Tamil Nadu is also very rich in floristic diversity. And then other thing is that in Arunachal Pradesh, because of Eastern Himalaya, it is very rich in its flora diversity. And then in Sikkim also, it's like that. Now, if you compare the floristic angiosperm diversity with the only with the northeastern state, that Arunachal Pradesh, Sikkim, Meghalaya, Assam, Manipur, Nagaland, Mizoram, and Tripura in these state, Arunachal Pradesh stand first, and then 5,192 taxa are present in Arunachal Pradesh, and then Sikkim is also just followed by 5,068 species. And other states are not that much in diversity, but these two states and including Meghalaya, maximum number of the species which are found in northeastern state, you can see that in these three states it is a maximum number of species represented. Now we'll go to the Arunachal Pradesh. Now everybody knows that Arunachal Pradesh is lying in the northeastern part of India, and it is the largest state among all the 7 states found in the northeastern region. Its area about 83,743 square kilometer area, and administrated by 26 districts. So if you see that full area is coming under Eastern Himalaya only. And then other thing is that the physiographically, if you see, there are, it's divided into three parts. That one was the lower part of the Arunachal Pradesh bordering to the Assam, and this there the Shivalik and foothills are present. And then middle portion is the lesser Himalaya, which is around 800 meter to, you can see that 1800 meter or 2000 meter it goes. Then above that it is a greater Himalaya. This about 2000 to up to 7,000 meter it is available. So 7,800 meter. That is the highest peak in Arunachal Pradesh is Gorichang, which is bordering to the China, Tawang, East Kamang, and West Kamang district. There the altitude goes up to 7,800 meter. So because of this altitudinal range diversity, means from 100 meter it goes up to the 7,800 meter, that obviously the flora and fauna of the state will be high. And then major drainage system, if you talk about, there are these are the rivers, the Kameng, which flows from the West Kamang to East Kamang, and then Subansiri is from the central this from Karadadi, Kurumkume, Papare is this Dapo Reserve. Is from that particular this lower Subansiri it is flowing, and then Siang River is in the Xiaomi, Upper Siang, West Siang, Siang and East Siang. There the Xiaomi River, Siang River is flowing, and then Lohit district and this Anjar district Lohit river is there, and between Dibang and lower Dibang and upper Dibang, the Dibang river is flowing. These are the major river which are found in this particular state, and then there are several small rivers are also present. Then, if you talk about the people and culture of the state, there are 26 major tribes are present in the state of Arunachal Pradesh, and they are divided into 102 sub-tribes. And each tribe, these 26 tribes, have different culture, different custom, and they are not actually mingled with each others. They are just have a different identity, and so they are following their customs, rituals separately from other tribes. So this is because this is also rich in we can say that cultural diversity also is very rich in this Arunachal Pradesh. And then a protected area, if you see that look at the then in Arunachal Pradesh there are two national park, 13 wildlife sanctuary and one biosphere reserve, which is very important. One in Pake Tiger Reserve is also there. Then we can say that one Namdafa Tiger Reserve is also there in Arunachal Pradesh. So these are the protected area which are present in Arunachal Pradesh, and these are the distribution of ethnic groups in Arunachal Pradesh district wise. If you see that Monpas are Monpas and Aka, then these fellows follows the Buddhist this, and then Nishis, and they are earlier they used to follow the Donipalo, and then now they are changed to Christianity. And then every state is in every tribe is like that only. And then Mishmi in Mishmi Hills, in Mishmi tribe is there. Then in Idus are there in this Dibang, Lower Dibang valleys. Then Tagins are there, Galos are there. So like that, 26 different ethnic groups are present in Arunachal Pradesh, and this is the distribution map of their in state. Now we will talk about the forest types. So Arunachal Pradesh is having a maximum forest cover in the country. If you compare to the other state, Arunachal Pradesh represents a maximum forest cover, and most of the areas are not under forest jurisdiction, but forest cover is well maintained, and more people, those who are living in that part nearby the forest, they are maintaining their ecosystem, and they are actually only, except if you see that zoom cultivation, if you see that they are just using some of the areas nearby their villages, or some of the little far from the villages. Otherwise the forest is intact, and very healthy forest is available in Arunachal Pradesh. And due to, because these forests are available because of this altitudinal variation, climatic condition, and because of these two factors, the forest is very rich. And these forests, we can say that these forests can, we can divide into three category. First one is the tropical forest, which is found in lower part of Arunachal Pradesh, which is bordering to the Assam. It goes up to 800 meter, we can say. And this is also then further divided into four category: northern tropical semi evergreen forest, we can say that then northern tropical evergreen forest, then northern tropical moist deciduous forest, and then secondary bamboo breaks. Some of the areas were fully dominated by bamboos. And then little bit above 800 meter to 2000 meter if you go, then in most of the part, then the subtropical forest will come. And these are the in this typical forest, this is divided into two part: East Himalayan subtropical broadleaf forest and subtropical pine forest. This subtropical pine forest is mainly dominated by pine species, and in broadleaf subtropical forest you will get the most of the Fagaceae members, that is Quercus species, Castronopsis, and then these species are mainly dominated in this particular. Then Lauraceae members are also there in this particular forest. You will get a maximum number of Lauraceae members. And then next one is temperate forest that we this is also a very important forest patch where it's start from the 2000 meter and goes up to 7000 meter, but in subalpine zone and alpine zone, alpine meadows. And then lower portion you will get the conifer forest, that is a mix of abies, suga, then Pisceas are there, then Taxus are there. These are the species which are dominated in temperate conifer forest. And then in subalpine woody scrub, if you go, then you will get the most of the robust species. Rosa species are there, berberis are there. These are species are which are very then small rhododendrons are there, and these are the species are very much dominant in subalpine woody shrubs. And then some of the Bombay species are also found in the Arundel area. Species are also there in subalpine woody for shrubs. Then alpine meadow, if you go alpine meadow, you will get most of the gentianaceae members are there, which are very common in this particular area, and like gentianaceae and then astraceae members are there, which are very dominant. Then caryophyllaceae members. So many of the families you will go that this particular area, subalpine meadows, these families were dominated in this particular area. Then temperate broadleaf forest, if you consider this, this is mainly dominated by Fabaceae members and Lauraceae members. These are the some of the picture of the tropical forest. You can see that first forest, which is up to 800 meter. Most of the things just little hillocks are there. Bamboos and musas are dominant in this particular area. Then Euphorbiaceae members are very much dominant. Macarangas are there. Then Bishophia are there. These are the members which are these are the plant mainly dominated by these forest areas, tropical forest. Then subtropical, if you above this, you'll go this. These are the area where you'll get maximum number of Fabaceae members, and this broadleaf species are there. Then this Quercus are there. Then Lithorex are there. So many species are there which are found in this particular area. Then Litzias are there. These are the species which are dominated in particular this forest. Then above 2000 meter you go, then the broadleaf forests are there, and these are the where the rhododendrons and then mix up with Quercus and Lauraceae members like Litzia and this Michaelis species, which are very common in this particular forest. Then this is the some of the pictures of alpine forest, where the alpine forest and alpine scrub, where the viburnum species are there. Then this you can see in the right side, the top, you can see the Picea and Suga forests are there, which is just below the subalpine and alpine zone. So these are the common communities. We can see that these are the families were dominated in particular this type of forest. Now I'll talk about the brief history of the plant exploration in Arunachal Pradesh. The Arunachal Pradesh was established as a statehood. It has received in 1975, 72 to 75, and then before that, in British time, then first exploration was made by R. Wilcox and Captain Bedford. These two persons were visited first time to the Arunachal Pradesh from Shillong, and then first, they explore the Mishmi Hills. Mishmi Hills that is found in Dibang Valley, the upper Dibang Valley, lower Dibang Valley. This particular area they had visited, and they had collected several plants, but could not identify those plants, and just kept. Then after that, Griffith, who in 1836 he came, and he has worked on his their collection, and he had described 900 species of flowering plant and 22 he has recorded 900 flowering plants and 22 species of fauna and allied, and published a book that is known as the Flora of Mishmi Hills. Then later on, after this, the Thomas Booth came, and he has explored another area which is in Lower Subansari Valley and east this Kurumkume district. There they had collected some of the this to the Siang district. They had Mainly, he had worked on the rhododendron of that region, and he had described some of the rhododendron boothi and other species from that particular region. Then later on, after 1800th century, then 1900, the Burkil has I.H. Burkil, the first person who went to explore the Abor expedition. He had conducted the Abor expedition from West Siang to this particular area and to the higher altitude, and he has written a book, Botany of Abor expedition. Then Kingdom and Ward later on in 29 to 1929 to 1931, and he did the botanical expedition, and then in the Mishmi Hill again, and then Kingdom and Ward is very renowned scientist. He visited that particular area of Lohit Valley and Tirap and Changlang, a particular area, and then he has reported some of the species from there. Then Anil Bore, who was actually known for the grassman grasses. First, he come up with the grasses of India, and then he scaled the vegetation of Akka Hill, which is from western districts of Arunachal Pradesh, especially in West Kameng, and then up to Tawang a little bit, but not below, means above Shela Lake. But from western part, he has recorded some of the species from that particular region. Then from 19 to 41, from Indian scientist from KP Viswas, he visited that particular Akka Hill, and he recorded 1549 species of flowering plant from that particular region, and then 9 species of gymnosperm and 58 species of fern. In this flora of Akka Hill, he has published this thing. Then after that, PC Kanjilal, GK Deka, RS Rao, K. Srinivasan, in 1950 to 55, they had explored the central portion of the Arunachal Pradesh, Kameng district, Subansiri districts, and this particular area up to a little bit of part of the Siang district. And they had collected several, they had made several collections, and these collections are deposited in Assam. This is in Shillong Herbarium, our Shillong Herbarium, and some of the part in Arunachal Pradesh also it is kept. Then R.S. Rao, G. Panigiri, J. Joseph. These are the several scientists who had made a collection between 1962 to 80, and their collections are also kept in Arunachal Pradesh. Then H. Chaudhary in 80s to 90, we can say these are the scientists like H. Chaudhary, S.K. Das, Pramanik, Vaishya, Jagdish Lal, G.D. Pal, and Dr. A. Mao, this our present director, also were there in that particular region, and they had also made collection, and their collection are kept in Arunachal Pradesh Regional Center. And then K.C. Sahani, H.B. Nathani, and Bahadur Bennett from FRI Forest Research Institute, Dehradun, they had also made some of the collection from this particular area, and their collection are also kept in FRI Herbarium, some of them in Forest Research Institute, SFRI, State Forest Research Institute. They're in Itanagar, it's maintained by Forest Department of Arunachal Pradesh. So some of the collection are kept there. Then after that, in 2002 till now, the SS Das, Benjamin Gogoi, Manav Bhomik, Kanwal, and Rawat Tiwari, K. Krishna Chalu, these were the scientists who were their collection were also made from Kurumkume district, and then East Kameng district, then West Siang, East Siang, and then throughout Arunachal Pradesh, this grasses of Arunachal Pradesh did back M.K. Kandwal's, and then V.K. Rawat, all teratophytic flora were done by V.K. Rawat and A. Binyamin for Arunachal Pradesh. Then I work on East Kameng district, flora of East Kameng district, and then Krishna Chalu did work on orchidaceae of Arunachal Pradesh. She was still doing. So these are the workers, those who have worked in this particular area, and all the collection were made, and they are kept in our herbarium in Arunachal Pradesh Regional Center. We are in Arunachal Pradesh. We have 36,000 actually, 35,986 accessions were made, and this is in all the specimens were kept in Arunachal Pradesh. And now we'll talk about based on this collection, what we had concluded now that the Arunachal Pradesh is represented by these are the 4 groups of plant which were studies were made, not in algae and not in this fungi. Very few studies and very less studies were made. So there is a scope of study in algae and fungi. But in other groups also, you might be knowing that every year almost 10-20 species are coming from Arunachal Pradesh, from angiosperm, and then from bryophytes also. So there are a lot of scope for studying this particular region. Now we can see that in this India, angiosperm is represented by 21,984. Then in Arunachal Pradesh, as I told you, 5,190 species are present in Arunachal Pradesh only. And then gymnosperm, if you compare, then 82 species of gymnosperm are found in India, and then 33 species are represented in Arunachal Pradesh. So maximum this we can say that then we can say that next one is pteridophyte. That pteridophyte is 1314 species are there, and in Arunachal Pradesh 650 species. Among this 1314 species of pteridophytic in India, 650 species are found in Arunachal Pradesh. So maximum, almost 50% of the pteridophytes are found in only one state, that is Arunachal Pradesh. Then bryophytes, if you take, then 2,800 species are recorded from India, and then among that only 217 species are recorded from Arunachal Pradesh. I think there is a lot of scope for bryophytic flora also. This number is very less. I think there need to be more studies so that lower groups of plant like algae, fungi, bryo, and pteridols are less study in this particular forest, and then people can take that study and get more and more new species from that particular region. Then lichen also, you can say that 2,989 species from India, but only 827 species are lichen are present in this particular state. It's a too less number, because if you see, the lichens need a less polluted area, and it is an indicator of that pollution. And this Arunachal Pradesh is very dense forest, and very good habitat is available for growing the lichen. So lichen also need to be studied well there, and a lot of scope is there to study the lichen of Arunachal Pradesh. Then if you look at now I am an expert of angiosperm, so I will talk about the angiosperm. Then the dominant family in Arunachal Pradesh in taxa wise, if you see, look at that, Orchidaceae having a maximum number of taxa, that is 659 taxa are present of Orchidaceae, that among 1300 species found in India. If you see that out of that, 50% of Orchidaceae are represented from Arunachal Pradesh itself only. And then Ericaceae, if you talk about, the 289 taxa are found from represented for Arunachal Pradesh. So 312 taxa from Ericaceae in India are maximum number of species are found in Arunachal Pradesh. So these are the we can say that Orchidaceae and Ericaceae, maybe this is the center of origin of the this family. Most of the species present in this particular area, maximum species are distributed from this particular area, from Arunachal Pradesh to the nearby state or the country. Then if you talk about the Poaceae member, then 255 Poaceae taxa are present in Arunachal Pradesh. Later, after that, Asteraceae, Leguminaceae, then it's like that, so on. So for Orchidaceae, there is a lot of scope in for the this not Orchidaceae, this Rosaceae and Articaceae. Very less study has been conducted. So there is a scope in Articaceae and Rosaceae members that can be conducted in Arunachal Pradesh. Now I'll talk about the this earlier slide I had just discussed about the what are the floristic diversity of a country, and then in present in Arunachal Pradesh. Now I'll talk about their uses and their availability in a particular state. That you people might be knowing that the gooseberry which we are eating in the market, it is available, but in Indian flora, if you talk about we have a ribis species in Himalayan region. There are 5 species of ribis found in this particular region, and but this none of the species were used in our market, and this because of this nutritive analysis need to be done for this these species, and if it is good, because local people are eating these fruits of this particular plant, this 3 4 species of ribis they are using, and this but in Indian market there is no use of this thing. So if further study of this nutritive analysis of this particular fruit is done, then we can also get marketed this, and this will help to the livelihood of that particular region or the particular community, that it will help to protect the and the conserve the forest also. If they will get some money from this. And then in if you know that these are the 43 taxa are present. Oh, sorry, something is wrongly written. Ribis is only 6 taxa are present, not 43. Then rubus, if we'll talk about the rubus, also is very important genus which is present in Arunachal Pradesh. Maximum number of species are found in Arunachal Pradesh, that is 43 taxa. It is present in Arunachal Pradesh itself. And if you talk about the whole Western Himalaya and Eastern Himalaya, then only 53 species are there. So rubus is also very important genera which are very much diverse in this particular region, and the fruits are edible. Whenever I used to go in field, whatever species there are 43 taxa, maybe 14 or 15 taxa I had also tasted, and these are the very good fruit. When you are tracking and you are not getting anything, you can just have this fruit, and you can walk this. And this the same thing we have to think of this, how we can market these fruits, so get marketed, and this nutritive analysis and other things, some industries can come, and then any product can be used, like strawberry. This is our strawberry we are eating, and instead of that, if you can use this also, it will be good. So these are the some of the species like Ellipticus, Floribundus, the Parviflorus. These are the some of the species are present in Arunachal Pradesh. Then we will now talk about the rhododendron diversity to the Arunachal Pradesh. This rhododendron is also very important genera to India, and if you see that in Arunachal Pradesh, overall India, it is represented by 132 taxa. Throughout India, it is 132 taxa present, but in Arunachal Pradesh itself, it's 119 taxa of rhododendron are present in Arunachal Pradesh. So, for the rhododendron diversity, if we talk about, the Arunachal Pradesh is very important. And these plants are very useful for the local people, and even in this in Western Himalaya, if you there go, then they are considering this rhododendron. They are selling Buras juice. In Western Himalaya, the rhododendron arboreum is used as for extracting this preparation of this juice. The flowers were used from and they prepare the juice and they sell in the market. But in Arunachal Pradesh, in Tawang, only Tawang, they are using these flowers, but other part of Arunachal Pradesh it is not used. And maybe in Arunachal Pradesh, this can be promoted, and some of the small industry can come, or local people can be uh, the methodology can be transferred to them, and then they can use this, how they can extract the they can use the flowers to extract and prepare the juice, and they can sell in market also. This is very good, thinning blood, and these things are studies are going on this species, and these are very important group of plant which we can utilize, and this can help in sustaining the local people of this particular area where it is available. The next thing is that the orchid also very much. If you look at the Arunachal Pradesh, there are 659 taxa of orchids are orchidaceae members are present in this particular region, and these orchidaceae are also very important. The flowers are very showy in many of the meetings and all. We can see that orchid flowers were used, and they are for decorative purpose. Then many of the orchid species are used in this perfume industries, and then some of the orchids are used in this Ashtavarga. You might be knowing that Ashtavarga plant group. There are three orchid species are used in that preparation. It is a health tonic. It is many ways it is used in curing many things. So the orchidaceae also very much diverse in Arunachal Pradesh. If you see that there are 1252 species taxa are present in India, and these taxa among these 50% of orchids are present only in Arunachal Pradesh. So this state have a high potential of orchid cultivation, and it can be marketed because most of the Silogyne and these dendrobiums, these species are extracted from the wild, and they are sold in the nearby countries to the Myanmar, Thailand, and these particular they are illegally traded because it is all whole family is reported under CITES, but how the people are this illegal marketing is going on. So if you can promote this, the people can grow this plant in nearby their houses and they can sell, and it can be encouraged to do this for the conservation of many of the species in the particular region. So these are the some of the orchid species which I had placed here. You can see that right from the Cymbidium, look at the beautiful flowers and showy flowers are present, and then these can be used then for the commercial purpose also, and for conservation also. We can highlight these species, the spot, highlight the where it is available and how we can protect them while extracting from the wild. So these are the some of the pictures. You can see that these are the orchid which are found particular in the state. These are the some of the Arundinaria, then Anthogonium, then Dinia. These are the species. Then in the third row, you can see that one Phaphilipedium fairy. It is an endangered species, very critically endangered. Actually, not endangered, critically endangered species, which is found in Arunachal Pradesh. Now, all the population, most of the population are wild out. Only one of two location it is available. So these locations were identified, but due to the lack of knowledge, the local people were not knowing the importance of this species or where localities of this species. They are just clearing the forest and they are using for their cultivation, and they don't know the actual the important what are the important plants available nearby their particular area. So what we are doing that this local people we used to aware this, these are the species which are not found anywhere in the world. So this is found only in your few localities of Arunachal Pradesh, and this is the one of the locality in Papumpari district or the East Cameng district, and this these need to be conserved, and you have to come up. And then ecotourism also, this is a very showy flower. You can show them, all the people will come and watch, the student will come, other people will come. So the some of the people get convinced and some of the people get agitated, why you are interfering our area, and this is our forest, we will do whatever we want. So we have to handle in such thing that okay, we can protect the there also rights, and then we can protect the plant. So in that way we can try and conserve this plant in some of the, try to formulate some of the other thing area we have to identify, and then we can place them, we can try and grow them. If it is growing, then we can transplant from that place to other places, and we can conserve this plant in wild also. And nursery and in situ conservation is going on, but it is not that much useful. Then next group of plant, if you see in Arunachal Pradesh, then the bamboo. Bamboo is the lifeline of the hill people, actually, we can say. And there are 42 types of bamboos are present in this particular region, in this Arunachal Pradesh itself. And maximum this 70% of bamboos are present in Arunachal Pradesh as compared to India. So maximum number of species present in Arunachal Pradesh, and people are using in various ways. And now biofuel, then timber, and then their local livelihood and everything, they are here dependent upon the bamboo species only. So this is also one of the important group of plants which are present in that particular area in subtropical belt and this tropical belt also. Some of the bamboos are growing up to that region. So This is very important plant group and which are useful for them. Now we can see the next thing is that wild useful edibles of the Arunachal Pradesh. This pie chart you can see that there are 742 species which we had conducted one project. We are doing that, but it is not completed. But what we found that 731 species we had collected, and these they are used in some of other kind of uses are there for the local people. And then we had first one is that 8% of the species are used, flowers were used for edible purpose, and then 38% of the plants were used, leaves were used for the edible thing, and then 4% of the tender shoot of the plant were used for edible as a edible plant, and then whole plant, some of the whole plants also 5% of the edible plants were used for edible purpose, and then some of the 20% are used as a stem. And then next one is that utilization purpose. If you see, then 60% of plants are used as among the 706 species. We can say that 60% of the plants were used as a vegetable. Then 34 this like that is there. Then if you talk about the tubers and these fruits, the many of the plants where local people are using the local wild varieties and wild plant species from the, and then some of the tubers they are using most of the Dioscorias, they are using Amorphophilus, Colocaceae, and these are the mainly they are using as tubers. And then some of the flowers like gingibraceous flowers, then alpinias, then this gingiber, and these flowers they are using for their uses. And then some of the fruits they are using mainly this Solanaceae members, Solanaceae, Solanum americanum, Solanum nigrum, Solanum, this species they are using for their food purpose. Now I'll talk about the another group of plant which belongs to the Calamus genus, which is from family Ericaceae. So this is also locally known. Commercially it is known as bent. So you might have heard about the bent. So bent is also very much important in the particular this particular area, and people are collecting. And most of the thing they are not actually preparing these things. They are just cutting and selling to the Assam and local nearby market. They are collecting and they are just preparing this sofa set and other items. So which are used in for decorative purpose or looking good. So people are using this paint products. So most of the species, there are 7 species found in Arunachal Pradesh. 3 species are used in commercial purpose, and they are just selling from the wild. And now many of the areas earlier, when you talk about the paint distribution and population, many of the areas where earlier it was known from the many of the areas, but local people, if you discuss, they are telling that earlier it was, we used to collect this many things and now we are getting this many things. So that means the population is almost 60 to 70% of the male population is almost wiped from the many of the districts, and very few areas which is not accessible or not approachable from by the local people are still there. But maybe they can cultivate nearby area, they can plant nearby their villages, and they can extract this from a wild gene. We have to conserve. So we are just telling them that don't collect from the wild, whatever fruits are coming, and then you can collect and you can grow in nearby your houses. And if it is coming good, then you can sell it from the sell to the market, but don't collect from the wild, because if once it is wiped out from your area, it is not going to come next time. Then you have to depend upon the other area, or you have to go to the some other part of the place, and then there will be clashes between the communities. So instead of doing that, you conserve your whatever you have resources in your surroundings, and then try to cultivate in a nearby area or start to plant the open areas, and then you can utilize in for many more years. So like that, some people they used to think that okay, this is the good option, we can try. But some of the areas like in West Siang, the Kusi village or Basar village, they are trying. So it's like that, it's going on. Then I earlier I talked to the bamboo species are also very important. Then in Arunachal Pradesh there are 43 taxes of bamboos are present in Arunachal Pradesh, and these are the Arundinaria, Bambusa, Tendoplanus, Melacoa. Then these are the different genera which are present in Arunachal Pradesh. How they are using, you see, they don't have infrastructure or the government support. So what they are doing, little government support and their own engineering, they are making the bamboo bridge to the big river, almost 100 to 100 100-meter or 150-meter width of river. They are making this type of bridges, and this is very dangerous also, but it's very good also, we can see. Then bamboo shoots are also very famous in Arunachal Pradesh. They are used as a pickle in Tenga. They is a little bit sour in taste, and so they are using this bamboo pickle or bamboo curry, and then many of their items, food items, they are mixing with this, even in fish or chicken or any mutton they are using, they are just adding this thing and eating. So bamboo is very important. And now bamboos are useful for the biodiesel, biofuel. So the more and more studies are coming. Pulp industries, paper industries are very important. These bamboos are there. So this can be utilized for growth of these people also. And some of the money, if whatever industries are coming, they can some they can give to the local people for the development of these local people, then this will be the good option. These are the some of the pictures. The local people, they are using, they are not using, actually they are, most of the vegetables they are collecting from the wild. So wild vegetables are very famous. Leafy vegetables like piper species are there. Then astraceae members are there. These impatiens are there. Then brassicaceae members are there. So these are the actually families. They are most of the species they are collecting. The impatiens are there. Only one species of impatiens, that is impatiens valley. They are collecting. So these species are there. So these species they are collecting from wild. Chenopodiaceae are there. Amaranthus are there. So in these families, from many of the species they are collecting from wild and eating. And some of the fungus, this you can say that Pleurotus is the group maximum they are using from the wild, and using that them. Then bamboo shoots are also they are using as a vegetables. Then Arunachal Pradesh. Now I'll talk about the medicinal diversity of Arunachal Pradesh. Arunachal Pradesh is very much rich in medicinal plant diversity. In Ayurveda, there are 1,679 plants are reported from Ayurveda, and then among these 502 species are present in Arunachal Pradesh. So most of these species are present in Arunachal Pradesh. So Arunachal Pradesh is rich in medicinal plant diversity, and many species are now threatened and endangered because of illegal trade and collections. Because many people don't know that the value of that plant, was just a person coming from other state or some other region, they are coming, they're showing the picture to them that this plant, have you seen that particular plant in your particular area? They say yes, we have seen in this our forest, this is plenty. But they don't know the actual use of that plant. So what they are telling, okay, you just collect this thing, we will give this much money. I will give you one example, like Paris polyphylla, which is illegally traded from the northeast region. And Hindi it is known as satwa, in satwa, and in Ayurveda also it's satwa. The Paris polyphylla was earlier throughout Arunachal Pradesh is were present. Then from 2003 the collection was started, and then up to 2013 when I conducted one study, I found that most of the Arunachal Pradesh, this Paris polyphylla was almost gone. Most of the areas which were known from the earlier location, from our herbarium or the local people, they said that it was a very plenty of the plants were available nearby area, but now it is not available. Even in if you go to 2, 3, 5 kilometer of the periphery of the or villages or inside the forest, you will not get this species. It's almost collected and sold out. So like that the people are not don't know actually the use of that plant. So because of that they are just in ignorance they are selling this plant. There's one more plant is that is panax also illegally collected from the particular area. So the government of Arunachal Pradesh, BSI, and other forest departments, they can come up and they can distribute this pamphlet and these things that if somebody is coming and just telling that these plants are available in particular area and collect and give to us, then they have to develop some kind of system that the information need to be passed to the forest department or the authority that they can take action against them. But people are poor and they don't have any other option. So what they are doing, they illegally they are selling. So something we have to develop in that way. We have to think in that way that we have to conserve this our bioresources that, so that it can be utilized in further way. Or sustainable way of the harvestation is also important, that we have to teach them. And if they get understood and then some benefit will be if they are getting, then definitely they will try to adopt this system, and this can be encouraged in this particular area. So there are a lot of medicine plants available in Arunachal Pradesh. So these are the some of the pictures which I have presented in my slide. Now I will conclude my record. I will sorry, I will conclude my talk. This basically most of the part I had heard, because this is a very big topic and very in limited time period. If I complete the Arunachal Pradesh diversity, it is not possible. It is very diverse in nature, in culturally it is also diverse, and then plant fighter also it is very diverse state. So a lot of things are there. Now will everything what we are doing is that why we need to conserve this plant of flora and fauna in our region, because we have to we are actually ecosystem services. If we talk about this, we know we want this plant for our future generation, also for the betterment of this earth. So what if we talk about the ecosystem services, what are we are getting? Hello.
Yes, yes.
Ecosystem services, if we talk about, then what are the things we are getting? Then carbon sequestration, plants are helping in carbon sequestration. Whatever the atmosphere, we are releasing the carbon dioxide, everything is absorbed by the plants, and it is the then water purification they are doing. And then climate regulation is also very important, that is regulated by plants. Then nutrient cycle are also there. So everything is interconnected, connected. So this is a kind of web. If one thing is gone, then other thing will not sustain. It's like that. So we know some of the we have some of the knowledge that these many species are there. So what are their role and how they are functioning in that ecosystem, that need to be studied. And after that only we will get to know that whether this is important or not. Because if we will ask the local people this, they will say that okay, these are the plants, these are the just like plants we don't want, because they don't have the use, they don't know the use of that plant. So that's why they say okay, it is weed, it's just like a waste for them. Just only they will use as a firewood or something, that they will protect them or they will utilize them. Other thing they will just cut it or throw it. Even like the other things, the development activities are going around. In India, we are developing nation, we have to develop. So we have to sacrifice some of the forested area and other things. But before doing that, we have to find out that the some other way, that if something is important, like some of the patches were very important, we have to conserve them. How we can conserve them, and we can avoid this thing, or we can bypass those things. If these things are included in policy making and other thing, then we can conserve our plants and bioresources whatever is present in our country. So this with this talk this now present government is mainly focused on this our India stands very low in these things. So EPI score. So what present government want that India should come up and the information need to be generated. So we are lacking in this information generation, that environment performance index. So BSI, ZSI, and other organizations are also working on that. So uh, by collectively we can achieve the target, and most of the areas large gaps are there. We have to fill this time information we have to collect, and then we have to present in that particular portal. Then maybe India will go in high some other ranking, not in the base of that table. So based on that, the SDG, the UN, United Nations has given 2013-17 SDG, Sustainable Development Goals, and then where everybody should get food, nobody should get means sleep hungry, and everybody means hunger need to be zero hunger, good health, quality of education, everything whatever is there in 17 goals. The thing is that everything is related to the environment. If environment is not good, whatever we will develop or whatever we are doing for the people, if environment is not good, then this all this thing will go in waste. So better thing is that we have to think that how we can protect our environment, or whatever the floras and biodiversity, whatever the floras and faunas are available, what are the importance of there, and what are the role they are playing ecosystem services. If you talk about that 1 hectare of land, if you assess, you are not able to assess, like in Sundarban. If you take Sundarban example in West Bengal, I'm staying here. Sundarban is very important this forested area in West Bengal. 1 hectare of Sundarban, they had assessed that 1 hectare of Sundarban area is giving almost 1,000 crore of revenue. Actually, is required. Means it is giving like that. That means they are giving this much amount of services. 1 hectare of land is giving this much services. So the Sundarban is very big area. So like that, whatever forested or protected area or the we have, so we don't know the, actually we have not conducted any kind of study or the assess what are the services we are getting from them and their value is what. So we are getting in free, everything we are getting in free. So that's why we don't care about these things. So these things we can also we can develop some model that the students also get understand that what is the importance of the this biodiversity and other thing, and then the role of this each component, and then we can this something we have to develop for the local people, those who are living this particular area. They should also understand what is the use and importance of this, so they can also involved in the protection of these species, and we can utilize these resources in sustainable way, so they can then we can achieve our target in this particular goal. Now I will with this I will would like to conclude my talk. Thank you very much for giving me opportunity to talk about this uh, uh, phytodiversity of Arunachal Pradesh.
Thank you, Dr. Tiwari. Thank you so much for taking us to Arunachal Pradesh. You have shown beautiful diagrams, beautiful pictures, photographs. That was all wonderful. Uh,
Thank you.
the the presentation is open for discussion. One or two questions we can take. Yes, from participant you can ask questions. And if there is no more question, I would like to ask Dr. Tiwari,
Yes, ma'am.
what is the reason behind the rich biodiversity of Arunachal Pradesh? There are many states in the northeastern region, but why the most diverse region is Arunachal Pradesh?
Yes, ma'am. Actually, Arunachal Pradesh lies between Eastern Himalaya. Actually, if you see, most of the diversity is present in Himalayan region. There are 10,000 plants are present in Himalayan region. So, climatic condition also favor the this diversity. And second thing, The altitudinal variation also play a very important role in the diversity of the plant. Plain, if you consider, some of the areas may be up to 200 meters, 300 meters. So there is no scope of that much diversity. Some of the plants which are specific to that particular region, they can adopt, other they will not grow. But as if you see, look at the Arunachal Pradesh, from 100 meter is go to 7,000 meter. Even second thing is that the rainfall and other things are very important. Most of the tropical belt, if you consider Arunachal Pradesh, that having a very good diversity. In temperate region also in subalpine, then alpine region. So this is because of that Arunachal Pradesh is very rich in diversity. But if you see that floristic number of component in Arunachal Pradesh, 5,600 I told you, 5,000 I told you. But in Western Himachal, if you talk about Uttarakhand, they were also having a similar number of floristic taxa. So Himalayan is the very important role. Altitude plays the very important role in the diversity.
So much. If we will receive any question, that will be forwarded to the speaker.
Thank you, Dr. Tiwari, Umesh Tiwari. It was really very interesting talk, and really we had a good glimpse of the how many types of plants are there, how vegetation is growing. It was really, as you have taken us to a journey, like Dr. Shweta told, that we have taken a journey to the Arunachal Pradesh. We felt that we are just going and traveling through Arunachal Pradesh. It was very interesting lecture. Thank you for your valuable time, and we are really very, very thankful to you.
Thank you, madam.
Thank you.
Excuse me, ma'am. I want to ask a question. Hello?
Yes, yes, yes.
Am I audible?
Yes, audible.
Yeah. Sir, you were talking about Arunachal Pradesh, few species which are getting smuggled by other countries. When they come to, they ask or buy within a very few or low price at a very lowest cost, I can, what we say. But they are priceless, right? They are Ayurvedic priceless herbs. So I want to ask, what are the ways you are I don't I want to ask that what are the ways or you provide teachings to or educate people not to get sold those priceless botanics to those smugglers, or I mean, you
Yes,
understanding, sir?
I got your question.
Yes.
Actually, the thing is that, madam, there are few species which are illegally traded in Arunachal Pradesh, not all 500 species. So, like example, we'll say the Paris polyphylla. The uh, rhizome part was collected and it was sold. If you collect the rhizome of the plant, that means the whole population is wiped out. And the rhizome, what we suggested that the people can, because it is illegally collected, the forest department has to come and they have to make that some arrangement that the nursery need to be prepared nearby the forested area that they can grow the plant and they can distribute the seed to the local people. They can cultivate in that particular area. It can be grown. So after 4 or 5 years of age, the plant can be utilized and the root can be extracted from that plant. So this was the suggestion what was given to the thing. The thing is that the illegal trade is there. You cannot stop it because most of the people don't want to do the cultivation and other things. So then you have to monitor in such a way that the whatever the population are there, you identify those population, you keep it as it is and just supply the seeds to the local people so they can grow it and then it can be channelized in legal way. So this was the suggestion to them.
Thank you, Dr. Tiwari.
Yes, ma'am.
Uh, Dr. Anjum, shall I proceed to
Yes.
next speaker?
Yeah. Dr. Umar Sharif is already there.
Yeah. So we welcome Dr. Sharif. I would like to brief
Thank you.
about, I am just reading the biodata of Dr. Sharif. Dr. Sharif is a native of Mysore, so we are having Mysore person here. his master's degree from PG Department of Botany, University of Mysore in 1986. He worked in cytogenetics, plant breeding. Later, he pursued aromatic and medicinal plants of Karnataka. He was awarded PhD degree from the same university in 1994. He served as a lecturer in the same department and later he worked for the another college. Then he joined the Rubber Board, Government of India, Bangalore, as a senior field officer, and he worked there for 5 years. By his constant efforts and zeal to join the research field, and it is very impressive, he was selected as a senior scientist in Botanical Survey of India, Kolkata, where he is working nowadays. He was posted to a very challenging place, Andaman and Nicobar Island, as a circle officer at Port Blair, where he carried out his work on floristic diversity and ethnobotany of aboriginal tribes. He has visited almost all the island of that challenging area and collected and documented many rare, endemic, and endangered species with his great emphasis on the orchids of Andaman Nicobar Island during the study period. In addition, he turned out a very interesting study on ethnobotany of the aboriginal tribes of Andaman Nicobar Island. During his study, many of the interesting potential plants he discovered from this region, and it was submitted to Government of India, New Delhi, through the Directorate of Tribal Welfare, Port Blair. Really, it was a challenging task he did. Then he was again given the next challenging task as a deputy director of AGC Bose Indian Botanic Garden, Kolkata. And he carried out the survey, introduction, conservation of arity plants with emphasis on the orchid flora of Northeast India. He attended several conferences, seminars of international, national repute, presented many research papers. He has been invited as a resource person from different universities, colleges, institutes to deliver talk on floristic diversity and ethnobotany. He has published 55 research papers on tissue culture of aromatic and medicinal plant, ethnobotanical study of Andaman Nicobar Island and orchids. He is also the team leader for the forthcoming volume 28 of Flora of India, which involves 24 angiosperm families and 504 taxa. Presently, Dr. Sharif is working on the projects Flora of Kerala and Flora of Tamil Nadu also, as per the approved action plan program of BSIE. From 26 May 2022, he promoted as a scientist B by Ministry of Environment, Forest and Climate Change and acting as a head of Botanical Survey of India, Southern Regional Center, Coimbatore. He has a very beautiful biodata and worked on a challenging task. He visited and collected Andaman, Nicobar Island, and very important thing is that he took a challenging task to document every island of Andaman Nicobar Island. So, Dr. Sharif, I welcome you on behalf of the organizing committee, and we are eager to listen to you.
One correction I want to make. Actually, he was promoted as scientist E.
Okay.
Yeah, he was promoted as scientist E. You, by mistaken, you said scientist B. It
I
is a
don't.
joint director position.
Uh,
Yeah, it's scientist E. So
uh,
he was promoted as joint director, scientist E, and head of the Coimbatore division of the Botanical Survey of India.
uh,
That's all.
Respectfully, Dr. Anjum, I would like to submit what the biodata I have in my hand
Yeah,
is a
yeah, it was
scientist.
there. It was there.
Yeah. Okay.
That
But
only I corrected. That only I corrected.
thank you, please. Thank you, please. Thank you for correcting me. He is holding a
Not
very good
correcting you,
position.
correcting whatever you got, but that B and E was there. So I thought I'll correct just it as internal matter, doesn't matter.
Thank you. Thank you.
Okay, now
Thank you.
over to Dr. Mohammed Sharif.
Thank you very much, Professor Shweta Yadav, for brief biodata. I am just attaching my the PowerPoint presentation, sharing my PowerPoint presentation, 2 minutes. Yes. Full screen. Is my PowerPoint presentation is visible?
It's visible, but it is not in PowerPoint mode. Kindly make it in
Yes,
PowerPoint mode.
slideshow. We are just seeing slideshow.
Yeah.
Slideshow. No, this one.
Dr. Anjum, I think Dr. Sharif is facing some problem.
They're trying, I think so. Maybe some connectivity issue.
Okay, maybe. So,
He's just trying to do.
meanwhile, we can discuss few things.
Yeah.
I got some messages over the phone, and participants are asking few things. I am just reading because he is not there, so we can. Yes, Dr. Sharif
No,
is okay. Then we will
he's there.
discuss it. Yes.
I think
If
so. He's there.
he is ready, then we will discuss this later. We were actually
Later. He's just uploading his presentation. Yes. Yes, sure.
Just no presentation again. Is presentation is visible? Hello? Hello?
It's like you have shared the screen instead of sharing your window. I think so. There is a problem. You have shared the screen.
Share screen.
Okay. Non problem.
Entire screen.
PPT should be open first there, and then you have to share.
PPT will open first.
Already open.
Oh, open.
Don't minimize the PPT. Keep the PPT open, and from below you just start sharing. And then select the PPT. You will find your PPT ka small icon. There you select it. First you had shared it properly, but only thing was slideshow. So shared.
Yes.
Yes, it is coming.
Oh, now it is coming. Is it?
Yeah. Now it has come.
Yeah, and it is moving also.
If there is any problem with the slideshow, then you can start from the side. The
Yes.
sidebar only you can move the slides.
My slides are moving also, no?
Yeah, it's moving.
Yes. Okay, I
Yeah,
will start. Sorry for the inconvenience.
No, please go ahead.
Sorry for the inconvenience. Yes,
No
okay.
problem. No problem, sir. Please go ahead.
Yes. Good afternoon, everybody. At the outset, I thank ZSI authorities in general and the authorities of Dr. Harisingh Gour Vishwavidyalaya in particular for giving me an opportunity to share my experiences of 2.5 decades of my work in this BSI organization on conservation and documentation of biodiversity. Actually, biodiversity is now today a very important component of life. It is the essence of life and it is the different entities on this Mother Earth. I will just start my presentation with two important quotes. One is the quote which is of William Shakespeare. You'll be taking away my life if you take away the means upon which I live. Shakespeare has told long back, if you take away the means, you'll be taking away the life from me upon which the means what I am just living. So that is the quote what Shakespeare has given. And the second quote is from the father of our nation, Mahatma Gandhi ji. Nature provides enough to meet the needs of the person, but not the greed of the persons, like that. So nature provides enough to provide the need of the person, but not to meet the greed of the person. So this is the two important quotes what I wanted to tell. Our forests and oceans are the cradles of biodiversity, no doubt about that one. And moreover, India is a mega diverse country and it is gifted with a huge quantity of biodiversity. Sustainable utilization of biodiversity is very, very important. If you are not sustainably utilizing the biodiversity, the reactions will be very, very serious. So that's why today biodiversity has become a very important component of Mother Earth, and it has become a sovereign property of any nation under consideration. So with this background, today I will be presenting my talk on conservation of biodiversity, a Botanical Survey of India perspective. Just a brief introduction about our department, because since I am in this department for last 2.5 decades, BSI is an organization coming under MOEF and CC, Ministry of Environment, Forest and Climate Change, and it is very much committed for documentation and conservation of floristic diversity of our country. The sister concern is Zoological Survey of India. Both are the childs of MOEF and CC. Botanical Survey of India is having its headquarters at Kolkata with 11 regional circles spread all over India. These 11 regional circles, one is Coimbatore, Pune, Hyderabad, Allahabad, Jodhpur, Dehradun, Gangtok, Shillong, Itanagar, Port Blair, and Solan. So these are 11 regional centers which are completely associated with the headquarters which is seated at Kolkata. Besides having its headquarters at Kolkata, Botanical Survey of India is having 4 more divisions in Kolkata. That one is AJC Bose Indian Botanic Garden. The second one is Central National Herbarium. The third one is Central Botanical Laboratory, and the fourth one is Indian Museum. So 4 divisions also present at Kolkata, along with the headquarters which is situated at Salt Lake. This Botanical Survey of India, a brief history, was established in the year 1890, and the founder director was George King, who was the then superintendent of AJC Indian Botanic Gardens, which was earlier known as Company Bagan or Royal Botanic Garden. So later, after independence, the Botanic Survey of India was reorganized, and for the definition of Botanic Survey of India, the person responsible for was none other than the Iron Lady of Indian Botany, Dr. E.K. Janaki Ammal, who was appointed as an officer on special duty with a special request by the then Prime Minister Jawaharlal Nehru ji. So this is the brief background of Botanic Survey of India. Coming to the objectives of Botanic Survey of India, five objectives, very important, five objectives are there for Botanic Survey of India. Collection, identification, and conservation of floristic diversity of our country. The second one is publication of regional floras and red data books. The third one is setting up of regional herbaria, botanic gardens, and museums. The fourth one is to conserve and multiply rare and endangered plants. And the fifth one is documentation of traditional knowledge, tribal knowledge, or ethnobotanical knowledge. So Botanic Survey of India is having its network of herbarium holdings also. All the regional circles are having their herbarium accessions or herbarium holdings, among which the prime one is Central National Herbarium, which is the biggest herbarium of our country. And it is having more than 20 lakh herbarium accessions of our country, as well as some of the foreign herbarium accessions are also there. So like this, their acronyms and other regional circles which are having their own herbarias and their holdings are completely shown here. And then beside that, Botanic Survey of India is having a network of botanical gardens also. Among all the regional circles, 11 regional circles, 9 regional circles are having their botanical gardens, where ex situ conservation work is under progress. Among all these 9 botanical gardens of other regional circles, the first and prime botanic garden I wanted to just highlight little about the work of the garden is AJC Bose Indian Botanic Garden, which is the mother of all botanic gardens, and it is the biggest botanic garden in Southeast Asia. Besides that, last decade, Government of India, MOF and CC has asked BSI to have another botanic garden, that is Botanic Garden of Indian Republic at Noida, and that is also under the jurisdiction of BSI. That is also coming up with a very great work, and Government of India want to make it as a very important center for ex situ conservation, because many of the foreign delegates, whoever are visiting New Delhi, they just visit this botanic garden of Indian Republic for seeing the ex situ conservation and the rare and endemic plants which are being conserved from the different parts of our country. They are brought here and they are conserved. So this is the network of botanic gardens what Botanic Survey of India is having. I told in the second objective, publication of regional floras, red data books, and some of the dissemination of knowledge, whatever which is completely gained in the field, that is being disseminated to scientific fraternity in different ways by publishing in different regional floras, red data books, in which we account many of the RET plants which are completely present in different areas of our country. Besides that, every year Botanic Survey of India releases plant discoveries, and especially in the World Environment Day, 5th June, which falls on 5th June, MOE of Minister of Environment and Forest, he releases plant discoveries. Whatever the plants which are discovered every year by the scientists of the Botanic Survey of India are documented in this one, and this will be released for the benefit of the scientific fraternity. And besides that, Botanic Survey of India is having its own official journal, also scientific journal, with the name of Neelambo, and it will completely publish many of the research articles. So this is the background of the botanical activities of Botanic Survey of India. Now coming to the today's topic, which is related to the today's webinar, conservation of biodiversity, role of biodiversity conservation in ecosystem functioning. Just I want to give the chemistry of conservation of biodiversity, how it is being conserved, and what is the importance of this biodiversity, and what is the importance of cultural diversity. All this I wanted to just put before the August gathering, which has just joined this today's webinar. Yes, friends, let us come to this biodiversity. Biodiversity is a very important term and very important component. It is a sovereign property of any nation. Constitution, I will say. Biodiversity is an umbrella term all the living entities which are categorized themselves, they fall under this umbrella term biodiversity. So different, based on the different categories, different type of terminologies also has been used. We are having floristic diversity for plants. We are having faunal diversity for animals. We are having fungal diversity, microbial diversity, whatnot. Even agro biodiversity also is also existing. So different types of terminologies has come up for understanding this whole concept of biodiversity. Today, biodiversity has become a very important subject, and it is coming up as a science rather than a subject, I can very well say. So biodiversity is very important, and conservation of biodiversity has become a necessity rather than a fashion. Earlier, biodiversity was considered as a necessity, as a fashion. Some of the botanic gardens were raised. Some of the Mughal gardens were raised as fashion to bring about beauty or to increase the aesthetic importance of the area. They were completely brought out as the biodiversity was considered as a fashion, but now it has become a necessity. It has become a very important constituent. It has become a sovereign property of any nation under constitution. All the countries all over the world, they are completely concentrating on conservation on this important entity, the biodiversity. Of course, India is one of the megadiverse country. India is one among the 18 megadiverse countries. So some of the megadiverse countries you may be knowing: Brazil, China, Mexico, Indonesia, Australia, Madagascar, and then we have Congo, Philippines, United States, Ecuador, whatnot. These are the megadiverse countries. Among them, India is also one of the megadiverse. It is gifted with megadiversity. We are having, if you see the biodiversity profile of India, we are having nearly 50,000 12 plant species and about 90,000 animal species are there. It is a huge biodiversity which is existing in this our country. So how to document that one, how to know that one? So there should be a chemistry involved to understand this huge biodiversity which is existing in our country. So besides that, whatever the biodiversity which is existing in any country or any nation, we have to first know what are the entities which are involved. If we are not understanding what are the entities which are involved, then whatever the strategic programs, whatever the action plans, what we are advertising, the result will not be very effective. So that's why understanding and documentation of biodiversity is very, very important. Once if you document, then the effective program can be very well brought about and the results will be very positive. So that's why the documentation of biodiversity is very, very important. Again, once the documentation of biodiversity is coming up, then we should be very careful. We should be very careful that documentation should be properly done, and then only the further conservation programs will come up. So that's why biodiversity is a very important component today. How much importance this biodiversity possesses, that much of threats also it is having. Both anthropogenetic effects are also eroding this important component from this mother earth, and beside that, even the natural calamities are also completely eroding this important component from this mother earth. Anthropogenic problems in the form of urbanization, deforestation, and refrigeration completely eroding this biodiversity from the mother earth. Whereas the natural calamities in the form of tsunamis, floods, forest fire is completely engulfing this important component biodiversity from this mother earth. So we have to be very carefully plan our projects for documentation of this biodiversity. So that's why, based on that, a conclusion has been given that there are 3 positive mantras of biodiversity, and these positive mantras are exploration, documentation, and conservation. Besides these positive mantras, and in the contrary, negative mantras are also working, and these 3 negative mantras is: one is urbanization, the second is deforestation, and third one is refrigeration. So this biodiversity, even though biodiversity is existing from long ages, from the inception of the mother earth, or from the inception of the life, as the vice chancellor of Dr. Harisingh Vidyasagar Vishwavidyalaya, Professor Nilima Gupta told, from the inception of the life itself, biodiversity is existing, but definition of biodiversity has been given very recently in the 1980 by 2 important scientists, McManus and Norse, as the sum total of genes, species, and ecosystems in a given region. the sum total of genes, species, and ecosystems in a given region is the simplest definition given to this important component biodiversity. Besides giving the definition, classification of diversity was also done in the form of genetic diversity, species diversity, and ecosystem diversity. Three categories were made for this biodiversity as genetic diversity, species diversity, and ecosystem diversity. Besides that, some people, instead of genetic diversity, species diversity, and ecosystem diversity, some people refer it to as alpha diversity, beta diversity, and gamma diversity. Besides having these three types of classification of biodiversity, one more category in 1997, Haywood has added one more category of this biodiversity, and that is nothing but cultural diversity, or otherwise known as ethnobotany or indigenous knowledge, which is completely related with the tribal knowledge, or which is completely related with the indigenous knowledge of indigenous people. Everywhere, wherever the biodiversity, all over the world, wherever the biodiversity is existing, it is completely associated with many of the anthropogenic effects. So that's why cultural diversity was completely added as a one more category, and I will be throwing some of the light on the cultural diversity of Andaman, Aboriginal tribals of Andaman and Nicobar Islands in the second part of my lecture. Excuse me, slide is not moving. Just a minute. Slide is not moving. Slide in the next slide. Next. Yes. Now, so now I told now conservation of biodiversity has become a very important component, and it has become a necessity rather than a fashion. And now many of the countries and many of the areas are now considering to conserve this biodiversity. And one best example of conservation of biodiversity is done by none other than Professor C.R. Babu, the ex-Vice Chancellor of Delhi University, Pro-Vice Chancellor of Delhi University, the green doctor who restored Delhi's lungs. This is a very, very important example of restoration of ecosystem by setting up of a biodiversity park in Yamuna Express Highway. And whatever the area which was inundated area, which was completely barren area, he converted this area into a very effective biodiversity park. And many of the wild animals also he has started completely visiting regularly this biodiversity park, which I said. Many of the wild birds also started visiting and this area, and it has become a very, very good example of restoration of degraded ecosystem. And based on this, the Union Ministry of Environment, Forest and Climate Change has directed all the chief ministers of the states to create biodiversity parks in each of the capital. This has become an example not only for our country, but even some of the countries like Sri Lanka and Bangladesh are also have started setting up of such of the biodiversity park for restoration of degraded ecosystems. So this is the best example of restoration of ecosystems. So coming to the conservation types, biodiversity is generally conserved by two important methods. One is in situ conservation and second one is ex situ conservation. In situ conservation is carrying out by conserving the plants and animals or living entities in its original habitat. And the second one is ex situ conservation, in which the living entity is lifted from its original habitat and brought to other area, either it may be lab or it may be garden, and providing near natural conditions and conserving it is as ex situ conservation. Okay, in situ conservation, again, it is brought out by three important methodologies. One is by setting up of biosphere reserves and protected areas, and the second one is by national parks and by sanctuaries. I will not be touching upon on this aspect. Of course, just briefly, I will just tell biosphere reserves are very important places where we are having atmosphere, land, and water. Such biosphere reserves are best examples of in situ conservation, and forest departments are the best custodians of biosphere reserves. Okay, biosphere reserves as a concept also is very important for conservation of biodiversity. All over the world, if we take into consideration, we are having more than 738 biospheres spread in 134 countries. Whereas in our country, if you take into consideration, we are having 18 biospheres, full-fledged biospheres existing, and some of these biosphere reserves are, one is Nilgiri Biosphere Reserve, the second one is the Gulf of Mannar Biosphere Reserve, then we are having Simipal Biosphere of Orissa, Manas Biosphere Reserve, Nokrek Biosphere Reserve, then we are having Kanchenjunga Biosphere Reserve, Pana Biosphere Reserve, then Great Nicobar Biosphere Reserve. So different types of biosphere reserves are there, and they are in one way or the other protecting many of the living entities in the form of animals, as well as many of the rare and endangered plant species in their area. So now this is the remaining things. National parks, we are having 103 national parks, and we are further we are having 543 sanctuaries. They are also considering these living entities of our countries in different ways. Coming to ex-situ conservation, I will just concentrate a little bit on this aspect of ex-situ conservation, because BSI is carrying out much of the ex-situ conservation work with regard to conservation of this biodiversity. Again, this is brought about by three important methods. One is by tissue culture, the second one is botanical gardens, and third one is gene banks. Of course, gene banks and tissue culture are interconnected. Here, in gene banks, the plant germplasms are conserved at liquid nitrogen temperature, and we are having a very big gene bank at ICRISAT at Hyderabad, which conserves many of the agrobiodiversity of the crop plants. Besides that, NBPGR at New Delhi also is having a very good gene bank conserving many of the rare and medicinal plants. So this is about the gene banks. Coming to tissue culture, tissue culture is also one of the very important effective method for conservation of biodiversity. I will not go deep into, because paucity of time is there. These are all very big aspects with regard to the conservation of plant entities. Tissue culture itself is a very big topic of nearly 1 to 2 hours. Of course, just if we come to tissue culture, we should just remember tissue culture is a very important technique of propagating many of the plants in short time and space. And it is defined as the culturing of plants, cells, tissues, organs, and embryos and protoplasts on synthetic media under aseptic conditions. And once, if you are talking about tissue culture, we should not remember the name of the pioneer worker in tissue culture, Gottlieb Haberland, who is regarded as the father of tissue culture. He proposed this concept of tissue culture on an innate capacity of plant tissues or plant cells, that is called totipotency. Total potentiality of any cell and tissue to give rise to a complete plant is called totipotency. Based on that, Gottlieb Haberland proposed this technique of tissue culture. So by tissue culture, in short time and space, we can very well bring about many of the plants and we can rehabilitate them in their original habitat. So like this, here I am just showing an example of Silogan nitida, a rare orchid which has been propagated by tissue culture, and many of the plants are being sold to some of the visitors who are coming to National Archidium and Experimental Garden on air card. So the such type of tissue culture methodologies has been standardized, by which many of the RET plants can be multiplied and they can be rehabilitated, and biodiversity conservation can be brought about. And one more example is Artemisia pellens, an aromatic plant which is the source of Davana oil, which is the costliest aromatic oil. That also has been standardized, and many of the plants can be multiplied in short term and space. Here, long-term cultures can be maintained, and independent of time and season, we can take some little tissue which is there in the culture room, and independent of time and season, by just subculturing it onto a fresh media, we can very well we can propagate many of the plants. Here in tissue culture, the main requisite is presence of media, different types of media, the like MS media, Murashik and Scoop media, LS media, Lynchner and Scoop media, then Hildebrand media, Mitra media. These are all medias are available. High media are also available. Ready-made high medias are available, and we can very well practice tissue culture and conserve and multiply many of the RET plants. So this is one of the aspects of conservation of biodiversity through ex-situ means. And then like this, many of the crops, bamboos, other medicinal plants, banana, papaya, many of the plants, and even cut flowers also has been multiplied by this tissue culture. And many of the private organizations in the form of Harisingh, Malayalam, we are having Speak India company, then Indo-American, Mahiko, they are all completely multiplying many of the plants through this one and generating much of the economy. Now coming to the second method, by botanic garden. Botanic gardens are very important plants where effective ex-situ conservation can be brought about. Since I have got a service of nearly 7.5 years at the AGC Bose Indian Botanic Garden, I want to highlight some of the examples of effective tissue culture and many of the programs related to biodiversity conservation, how it is brought about in botanical gardens. Just highlight some of the information here. Of course, botanical gardens are very important place where effective Except conservation of biodiversity can be brought about. They are the places where scientific data can be effectively generated. They are the places where effective conservation methods and multiplication methods of RD plants can be brought about. They are the places where many of the environmental awareness programs can be conducted, and young minds can be sensitized with regard to conservation of biodiversity. An effective way of conservation of biodiversity can be brought about in botanical gardens. So here is the example of one very important botanic garden that is known as the Acharya Jagdish Chandra Bose Indian Botanic Garden, which is established in the year 1787 by none other than Colonel Robert Kidd, a colonel in the British India Company, East India Company. So besides having this botanical garden, Botanic Survey of India, besides having this very big, Asia's biggest garden at Howrah, Calcutta, 9 regional circles are having the regional circles except Deccan Regional Circle at Hyderabad, and recently started Solan Circle, 3 years back started Solan Circle at Himachal. All the remaining 9 regional circles are having their botanical gardens, where the plants are collected from different forest areas of the region, and they are effectively conserved, and the effective biodiversity conservation methodologies were studied in their botanical gardens. Besides the botanical gardens of these 9 botanical gardens of different regional circles of our Botanic Survey of India, along with AJC Bose Indian Botanic Garden, other departments are also having some botanical gardens, and one important known botanic garden is NBRI Botanic Garden at Lucknow. They are also carrying out extensive conservation of very RET plants as well as medicinal plants. Then another botanic garden is of Tropical Botanical Garden at Trivandrum, TBGRI it is shortly known, and another is FRLST, now known as Transdisciplinary University Bangalore. They are also conserving, and Regional Resource Center at Orissa, also Bhubaneswar, is also carrying out effective extensive conservation work through these botanical gardens. And now let us know in detail little about the work of Botanic Survey of India with regard to conservation of biodiversity, which is going on in Acharya Jagdish Chandra Bose Indian Botanic Garden, situated at Howrah, the twin city of Calcutta. This botanical garden was started in the year 1787, and it is started by by Colonel Robert Kidd, the then Colonel of Fort Williams. He was the honorary superintendent of this botanical garden and served the botanical garden for nearly 6 years. And then the next person who took the charge of this botanical garden as superintendent, and who is the first salaried superintendent of this botanical garden, is William Roxburgh. Of course, this garden was started as a starting point. Colonel Robert Kidd was having an imagination that let us have a botanical garden, and he brought about all the economically important plants in the form of tea, coffee, cinchona, nutmeg, whatnot, whatever sago, whatever the economically important plants, what we are seeing in our country. Now, all this has been tried first as a base in this botanic garden, and later they have been shifted to different areas in our country. He started, of course, his motto of Colonel Robert Kidd, the vision of Colonel Robert Kidd was also very important here to be noted. So many things are, but due to paucity of time, I cannot go deep, because only one just I will touch upon. Colonel Robert Kidd proposed to have the botanical garden to the East India Company, and they marked at this person, what is this, Colonel Robert Kidd, you are doing? We have come to take away whatever the important amenities which are present in this Indian country, and you are thinking of establishing yourself here. No, time is not short. People will make us to run from here like that. No, then he will just bring the vision which is there in his mind. He writes to East India Company, no, I am having, instead of taking one bag of pepper or any other gram masala, if you are taking 100 bags, is it not advantageous for East India Company like that? And his vision was completely different. Britishers has come to our country to take away whatever the important minerals, whatever the important plant resources, whatever animal resources are present. And instead of taking in little quantity, if you are taking a huge quantity, it will be very beneficial for the East India Company like that. He just proposes, and immediately the East India Company gives the nod for starting this very important botanical garden in the Howrah of this West Bengal. Of course, another motto was also there. Another reason quoted by Colonel Robert Kidd is the only conveyance from their from their country to India was through the sea route, and many of their ships were getting deteriorated. And another reason he quoted is, if you have got a botanic garden, we can have our own timber yielding plants. And at that time, the mahogany was the best, considered to be the best ship building wood. So that also the avenue which was raised by that at the time of Colonel Robert Kidd is still existing in the AJC Bose Indian Botanical Garden. So this is the motto how this AJC Bose Indian Botanical Garden was started by the vision of Colonel Robert Kidd. But later, after Colonel Robert Kidd, when William Roxburgh took the charge and he served this botanic garden for the 21 years, and he completely changed the scenery of this botanic garden and made it as a very important, effective center for ex situ conservation of plants. And he described many of the plants of our country, and he was referred to as the father of Indian botany, William Roxburgh. His contribution to the Indian botany, whatever botany now it has come to our country, to our different departments, to our different universities, it is the nothing but the efforts of, it is the nothing but the efforts of William Roxburgh. So here is the list of different people who have taken charge as the heads of this. They have acted as the superintendents of this great agency. Indian Botanic Garden, it started as Company Bagan. Later, it was converted as Royal Botanic Garden. Later, after independence, it was referred to as Indian Botanic Garden. And from 1997, our then Ministry of Environment and Forest, Jairam Rameshji, changed the name as agency Bose Indian Botanic Garden to commemorate to respect the works of famous plant physiologist A.J.C. Bose, Indian Botanic, A.J.C. Acharya Jagdish Chandra Bose, the plant physiologist of West Bengal. To commemorate his work, it was named as A.J.C. Bose Indian Botanic Garden. So these are the different persons who have taken charge as the superintendent of this botanical garden. And this is the map of this botanical garden. It is just lying on the west coast of the river Hooghly, which is just running, and the garden is spread to an area of 257 acres, and it is on the west coast of west bank of this river Hooghly. And the garden is divided into 25 divisions, and it is having 25 artificial lakes. Whatever the lakes you are seeing here, they are all Prine Lake, Kings Lake, Diwan Lake, Leram Lake, Scotchini Lake, Sadir Lake. They are all the artificial lakes which were completely raised by George King. And for the work what he has made for this landscape of this garden, dividing into 25 divisions and 25 artificial lakes, he was given the knighthood as Sir George King by the East India Company. So this is the map of this agency Bose Indian Botanic. One important structure I wanted to show: 19 great banyan tree, a great prized possession of botanical garden is there. And then we are having different lakes which are interconnected by one another, and they are finally connected to the river Hooghly. Once the high tide comes in the river Hooghly, water completely enters and feeds all the lakes in the garden, and low tide water goes through the canal, which is completely present in the periphery of the botanic garden. So with this landscape, the moisture content will be very more, and conservation of effective conservation, ex situ conservation of plants has been brought about in the agency Bose. The concept of King George King was completely appreciated by East India Company, and he was given the knighthood as George King. So he is the founder of Botanical Survey of India, and this is one of the lake where boating also is underway and giving revenue to the Botanical Survey of India. So he is the founder scientist of this Botanical Survey of India, and founded the Botanical Survey of India in the year 1890. And now let us have the important components in this botanical garden, agency Bose Indian Botanical Garden. The one prized possession inside the botanical garden is possessed of great banyan tree, Ficus bengalensis, and it is nearly 255 years old. Only one tree covering an area of more than 5.5 acres, and the canopy is, the circumference of the canopy itself is more than 500 meters, and it itself looks like a miniature forest and harbors many of the other plants, ferns, orchids, and it is a abode for many of the birds also. So even the logo of Botanical Survey of India is sufficing this great banyan tree. It is a very, very important entity and crowd puller of our botanical garden. New Year Day and on the Christmas Day, more than 30 to 40,000 people, they visit this botanical garden, and they have a look of this great banyan tree. Even it is a very important tree of a very affectionate tree of many of the foreign delegates, Chinese delegates, and Indonesia, Malaysia, Thailand. Many of the foreign delegates they visit, ambassadors they visit, and spend much of the time inside this great banyan tree. Of course, in 1925, the central stem has been removed. Only memorial is present because there was the chance of fungal attack, and the spread of fungal attack to other parts was there. That's why in 1925 the central stem has been removed, and still it is growing, known as walking tree, and nearly 4,500 prop roots are there. With that, it is increasing. A very great senior citizen of this botanical garden, Botanical Garden. And different sections are there in this botanic garden. We are having the Charkudyan, the medicinal plant section, where more than 500 medicinal plants of northeastern India as well as West Bengal has been conserved. And we are having a large farmhouse where nearly 35 species of palms. Of course, in the garden, nearly 110 species of palms has been conserved, collected from different parts of the country. We are having pinetum, more than 10 to 12 species of pines, that is gymnosperms, has been conserved. And we are having arboretum with nearly 12,000 to 14,000 trees has been conserved, belonging to 1,400 species. We are having cactus and succulent section also, where more than 100 species of succulents has been conserved. Bamboo system is also very well developed, where 35 species of bamboos collected from different parts of the country has been conserved. And conservatories in which many of the RET plants collected from different arid zones and different other desert areas has been conserved in these conservatories. And these are some of the heritage buildings. Of course, one I will not go deep into this one because time is also very less. Next part also is there. Now here is Kidd Monument to commemorate the contribution of Robert Kidd for establishment of this garden. East India Company has kept the Kidd Monument exactly in the center of this botanic garden, and people were coming from this River Hooghly, coming giving the respect to the Kidd Monument and seeing through the going through the garden, and then they were returning through the River Hooghly. This was the trend when the Britishers were having the rule over this country in this garden. So the different types of buildings are also still. This is Roxborough building, where Roxborough carried out much of the taxonomic studies by and control of this great botanic garden of East India Company was done by Robert Brough. And this is a Roxborough building, and this is a curator building where many of the programs were being conducted by the Britishers, and it was a dance floor also for the dance floor also for the many of the excuse me. This is a dance floor of many of the artistic programs which are going on in the botanic garden. So these are the different type of heritage buildings and monuments which are existing in this botanic garden. And another prized possession in the botanic garden is the presence of a giant water lily known as Victoria amazonica and Victoria cruziana, two species of giant water lily. This is the biggest aquatic plant in the world. In the 1884, Britishers has introduced this plant for the first time in this Indian botanic garden. In a native of Amazon basin, they have introduced this, the biggest aquatic plant in the world. One is the giant water lily, Victoria Amazon lily, Victoria amazonica, and the Santa Cruz lily, Victoria cruziana. Two species are there, and the background of the lower margin of the leaf is so strong that it can hold the weight of a child of 5 to 6 years old. So this is the giant position where the rare plant which has been conserved ex situ by ex situ means in the AGC Indian Botanic Garden. And another important prize question is present of double coconut, the biggest seed in plant world, botanically known as Lodesia maldavica. By the maiden effort of the scientists of Botanical Survey of India, the fruit setting was also done, and this is native of Seychelles Islands. Of course, only one tree again in 1882, Britishers had introduced in large farmhouse of this AGC Indian Botanic Garden. If you remove the outer rind of this double coconut, the inside this one, the seed will be like this. Of course, the kamandulu is completely made, and much of the water is, of course, it is known as the elixir of life. Many of the kingdoms, many of the rulers in the Seychelles Island, they were keeping this water in their this one, and it was increasing the longevity of the rulers. And to make carry out a big rule in the country, they were using it as elixir of life. The water of this double coconut, very medicinal water, and very rare plant native of Seychelles Island. Only one tree of our country it is there in AGC Indian Botanic Garden. Besides that, many of the RET plants, many of the rare plants are also has been conserved in this botanical garden. This is one branch palm, Hyphne tabaica, which is the native of Egypt and West Indies. The Britishers has introduced again in this botanical garden. We are having cannonball tree, Chauropita ghenensis of the family Lecythridaceae. We are having candle fruit tree, Fermentera cerifera, it is also there, very rare plants. And then another is dancing doll tree or orchid tree or tree of heaven, Amentia nobilis. Britishers has introduced one avenue of Amazon and Oblis is there. Once flowering season comes, complete that avenue will be full of flower, and they will be dancing once the breeze is there. Beautiful tree, one of the tree of heaven it is called. And we are having wish tree also, Adensona digitata, which is rich in minerals. Some people call it as the kalpataru, and even people they say if we make a ring and touch our forehead, our first wish will be fulfilled. And many of the foreign delegates they visit, and even many of the West Bengal people they worship this tree as the wish tree. So many secrets are also there. If you see the base of the tree, it will look like the foot of the elephant, and the markings will be like the eyes of the elephant. So many of the West Bengal people religiously they consider as very near to Lord Ganesh, and they worship it. And many of the renowned visitors they come, and here is the then Forest Minister Dr. Harshvardhan visiting AGC Post Indian Botanical Garden, and having a happy time inside the Great Banyan Tree. And we are presenting here the memento of Great Banyan Tree to the then Honorable Minister of Environment and Forestry, Dr. Harshvardhan. And here is our CK Minister, our IS, our Secretary, along with Joint Secretary Manju Pandey visiting. Dr. Kailash Chandra, the then ZSA Director, also is here. Our present Director, Dr. Mao, also is here. And here is the Seychelles Islands Ambassador visiting and discussing the MOU, thinking of MOU with Dr. Paramjit Singh, the then Director of Botanical Survey of India, for with regard to this double coconut and contribution of some of the saplings to the AGC Post Indian Botanical Garden. The MOU is being signed through this Ambassador of Seychelles. And besides that, we provide much of the environmental generating services, and many of the public services also we render. Many of the forest IFS officers, rangers, foresters, they visit and we render. Besides that, we even give know-how for this National Biodiversity Authority, and here I am attending the 14th National Meet of State Biodiversity at NBA Chennai, and Dr. Kailash Chandra also one of the participant, and we are just giving the this one the metadata whatever which is existing with regard to the plant, and we are giving the words of our director with regard to sharing of our metadata for raising the people's biodiversity register by the state biodiversity boards. So this is the type of, and we conduct many of the programs. Ozone Day, we conduct World Environment Day, and then we conduct flower show, and we even present our research results in Indian Biodiversity Congress, and provide the public services. And different this is one of the flower show which was been organized by Botanical Survey of India, and it was organized by the then governor of West Bengal, Narayan, Dr. K. Narayan, Sri Narayanan inaugurated this one, and many of the aspects of conservation were completely discussed. Besides that, we conduct many of this World Environment Day, one more day, in which the school children were invited, and much of the competitions were done on biodiversity, and the young minds were sensitized, and planting program also is being conducted. Sit and draw competitions were held, and on the topic biodiversity topic, and they were rewarded for best drawings what they will carry out. A rally also is conducted, and these are the programs which are conducted with regard to biodiversity. And of course, even whatever the effective conservation is carried out in biodiversity, so many programs problems will also we have to face. Associations, morning workers association, they will be conducting, they will be encouraging some other areas. They come and cook in some of the peripheral areas, and put the biodiversity conserved in that garden. The threats will be conducted. Threats are there, and they will be conducting many of the rallies and demands, useless demands they put before the director as well as the authorities of AGC Indian Botanical Garden. And you see bad use of the important trees which are there. They carry out, they sit on very important trees, and sometimes they just and due to the pressure, continuous pressure, sometimes the trees will just this one, and once the wind and storm is there, many of the trees get uprooted, and loss of important components will be happening. So these are the threats what we face with regard to this conservation of biodiversity. Of course, here I'm showing some of the figures of our field work, what we carried out with regard to conservation, documentation of biodiversity, and some of the works which has been carried out northeastern also with regard to orchids. Of course, now Umesh Tiwari has given little information about the orchids. We also had one topic. We had also taken an action plan project on conservation of orchids in northeastern India, because I was transferred from National Arcadium, Tamil Nadu to AJC Bus Indian Botanic Garden. Same work I continued, and then I took a project on orchids of northeastern India, in which we surveyed at Assam, Nokrek Biosphere Reserve, Arunachal, and then Tripura, and then other areas of northeastern we conserved. We just surveyed, and we conserved many of the orchid species of this northeastern in Indian Botanic Garden. And Cristata, Dendrobium jenkinsii, Dendrobium palpebrae, then this is Pleon perax, blue Vanda cerulea, then Vanda stangenia. So these are completely Dracula with blood stations. Here moth orchid, Epigenia amplum, then green spider orchid, Hebineria ovilifolia, then dove orchid, Catonia pedunculoris, the bumblebee orchid, then Rincostalis retusa, the foxtail orchid, snake, Papillum spiceranum, Papillum insigne, Papillum villosum, Papillum hirsutism, Papillum ferianum. These five are in North distributed in North India, and only one lady slipper occurred in South India is Papillum drury. This is present in Agastyamalai hills of Kerala state. And other animation forms, duck orchid, Thymia bicornis. This is, of course, an exotic orchid called dancing doll orchid, Oncidium goldianum. And here is one more interesting orchid called eagle orchid, as though the eagle lifting the chick from its beak and leg. This is called Xenophia Vaidya. Very beautiful flower and exotic orchid, and showing the animation form as the eagle, the eye of the eagle, feathers, and then the tail lifting the chick from its eagle orchid, Xenophia Vaidya, eagle flower. And then how these orchids are conserved, different ways of conservation of these orchids. They are having different houses, Cymbidium house, Dendrobium house. These bulbophyllums are completely conserved in these bamboo planks, and different methods. Bamboo planks are used like this, and even the dry tree ferns were used, and some of the hangers for the attraction of some of the visitors in National Arcadium. These are the methods which are followed for conserving this biodiversity, this important component of biodiversity, orchids. And we even publish some of the rare species or novelties are being published in some of the journals. This is one bulbul from Panjite, which was first reported new species from Sikkim, Gangtok, and it was to commemorate our then director, Dr. Pramjit Singh. It was named as bulbul from Panjite. And many of the publications, survey and conservation of RET, rare and endemic orchids of Western Ghats in the Orchid Society, and we have presented all these works in the seminars organized by Orchid Society of India. And of our student Dr. Suleiman has published the inventory of this Kozhikode district of Kerala state. And besides that, we take other groups also, aromatic plants also, as the project from 2017 to 2020, we have taken ex situ conservation of aromatic plants, and effective conservation of aromatic plants has been brought about in following ways. You can have a look of this one. And different areas or different blocks were raised, and plants were brought, established, and it was inaugurated again by Dr. Pramjit Singh, the then director, aromatic garden. Instead, the effective ex situ conservation is brought about, and the plants are documented, and plants are completely worked out, and they were first rehabilitated, and then afterwards they were introduced in different plots. And like this, these are some of the activities under progress. And coming to the next aspect, the second one is cultural diversity. I told cultural diversity in 1997, Haywood has added one more category of diversity, that is called cultural diversity, which is ethnobotany, or the traditional knowledge, which has been shown here. And William Hershberger is regarded as the father of ethnobotany. He only coined the term ethnobotany. This is also a very, very important field of plant studies or life science, which is having very, very potential because of because of this bioprospection and newer drug development programs can be very well. Much of the work is going on, and even economic botany, the international journal also publishes, and of course. Ethnobotany, the work on ethnobotany, this knowledge on ethnobotany, Dr. Pushpangadan, the NBRI director, he is well known for a work on the only one plant, Trichoepis xylanicus, which completely changed the life of Kanya. So minimum balance of biodiversity balance should be there. Otherwise, the complete food chain or the complete ecosystem will collapse. Of course, this completely regulates the chemistry of atmosphere, as a water supply, and have complete hold on agriculture system of any nation or any country or any area under consideration. So there also it plays very important role, conservation of biodiversity. So the genetic diversity of plants and animals is preserved by this conserving this biodiversity. It also provides immediate benefits in the form of recreation and tourism. Ecotourism nowadays, many of the botanical gardens also providing ecotourism services and earning money. Even agency based Indian Botanic Garden, more than 30 to 40 lakhs every year agency based Indian Botanic Garden through this boating and then visit to the different different important areas inside the Botanic Garden. And even this battery vehicle, all this is completely contributing to the economy of this of any organization or any nation economy. And finally, it serves as an insurance policy for the future generation. Otherwise, days will not be far to our future generation. We have to just tell in stories to our future generation. There was an animal with big trunk and pillar like legs, but animal will not be there to show. So that's why conservation of biodiversity acts as an insurance coverage for the future generation. Coming to the conclusion, biodiversity is an umbrella term which encompasses all the living entities categorized by itself. It is an essence of life on Mother Earth, and its documentation conservation is the need of the hour. India, with its innumerable tribes and ethnic groups, offers ample scope for ethnobotanical knowledge, and their cultural diversity, documentation of cultural diversity will be very, very potential and leads to many of the newer drug development programs. Andaman Nicobar Islands, the hotspot of biodiversity with fragile ecosystem, are very sensitive from conservation point of view. The forest reserves contain germplasm of many of the potential plants, especially mangroves. Medicinal plants are very, very potential. Timber yielding plants. Of course, the reason for setting up the penal settlement inside the botanical inside the Andaman Nicobar Islands, where the Britishers is the source of many of the timbers. The paddock, Pterocarpus, Dalberger, and Dipterocarpus, which are the very, very prized timbers, are present in this Andaman Nicobar Islands. And still the Chatham sawmill, which was established at the time of British, is still there in Chatham area of this Andaman Island. So Andaman is rich in germplasm. Since paucity of time, I could not touch upon all those aspects. And then coming to biological screening and phytochemical analysis of many of the plants of Andaman Nicobar Islands or other biodiversity important components are very, very helpful for new drugs and bioprotection. Aboriginal tribes are under extinction, and these important tribes are to be protected for the future benefit of the society and for the future balance of the environment in the Andaman Nicobar Islands. These sons of Mother Nature are to be protected in their natural habitats before they disappear completely with a rich treasure of indigenous knowledge. Of course, and Government of India also is giving very much concern with regard to the protection of these Aboriginal tribes. Even if any Jarwa or any tribe is falling ill, he was be shifted from the tribal area, and all 5-star treatment is given in GP Pant Hospital, and they are completely treated, and they will be completely left to their area. So these are the type of arrangements what Tribal Welfare Director has following in this Andaman Nicobar Islands. Thank you very much. I thank all the participants for the patient hearing. Very sorry I could not complete because the topic are very still many of the things I have just cut short. And thank you very much for the patient hearing. Thank you very much. I again thank the authorities, and I thank even Professor Shweta Yadav and Dr. Anjum Rizvi for the excellent coordination for conducting this one-day webinar. Thank you. Thanks to one and all.
Thank you, sir. Thank you very much for enlightening us about the activities of the BSI and how they are helping in the conservation of plant diversity. Thank you so much, sir. You have really enlightened us a lot. Thank you, sir.
Thank you, sir. Thank you.
Thank you.
Anything?
Now, we'll take up. We have participants. If you have any questions, please give it in a chat box with your email IDs. We'll conduct due to shortage of time. We'll then get back to you. Okay, sir. So we'll contact you in case we have any queries regarding your presentation. So now we'll move on to the next presentation.
Thank you very much.
Thank you, sir. Thank you, sir.
Namaste.
Okay. Dr. Shweta. Hello, Dr. Shweta. Dr. Shweta is not. Dr. Shweta, are you there? Connectivity is happening, everybody. So there is some connectivity issue with Dr. Shweta. Dr. Shweta, are you there?
Hello, ma'am. Am I audible?
Dr. Shweta is there.
Hello.
Hello, who's that?
Yes, ma'am. Hello. Am I audible? Ma'am is connecting right now. She is working on a connectivity issue.
Okay. We
just
wait.
2 minutes.
Yeah, we wait. So please bear with us. 1 minute. So as all you know, that next lecture is about is being given by our respected Professor S.K. Malhotra, sir. He's from Allahabad University, and it's like about his biodata. A brief biodata will be read by Professor Shweta Yadav, so that you will be aware of some of the highlights of Sir's research, what he has done up till now. And I think so most of you must be aware because he's a very renowned person and very well known in his field. And he has done lot of like work on fish nematodes. And he will give you a very interesting lecture. I'm sure you all will enjoy. So now over to Professor Shweta Yadav.
I am very sorry.
Yeah, please. Yeah, no problem. Please carry.
There was some problem with the connection. I would like to brief the biodata of Professor Malhotra. He is having 22 pages biodata over the Google. Last night I searched and I am presenting what is this of his biodata. He is a very senior professor for about 40 years. He has the experience. He guided 37 researchers and he was the fellow of the Zoological Society of India and he was again the fellow of the Zoological Society and one of the another important society of the bioscience. He was a member and he was the fellow society of the environmental sciences. He had 15 major research projects funded by DBT, DST, UDT, and ICAR, many more. He submitted 150 sequences to NCBI, very important what he has done. And he contributed 9 books, 5 review papers, 59 chapters, 165 papers, 2 done brief communication. And he also filed patents in 2007 for an improved application for collection of honeybee toxins. He was a member of many expert selection committees, including AMU, Kashmir, Rohil Temporary University, and many more. He participated in many conferences, including USA, Malaysia, Hong Kong, Thailand, Vietnam, Argentina, and I am not quoting all the names here, including Poland. He was also nominated as the expert panel in Agriculture Scientist Recruitment Board. He he signed MOU with University of Allahabad and the National Institute of Oceanography, Goa, and UNDP, Ministry of Environment. He visited 18 countries, and wonderful, I found over the Google that during the 4 years from the 19 to 2022, he published 10 review papers in the International Research Journal. He was designated for the 15 gold medals for pursuing his research interest. Presently, his research interest is the parasitology and the helminthology. Here I am presenting a very brief overview of his biodata. He has contributed many things to his account. So now it is over to Professor Malhotra. We are very excited to listen to your talk. Thank you. It is over to you.
Thank you, Dr. Shweta. I'll share my slides. Double click. करिए इसको share. चेक करिए पीछे है प्रेजेंटेशन है तो क्या stop. जाओ जल्दी आओ नहीं पूरा स्क्रीन ना Hello. Do you get my voice?
Yes,
Hello.
sir.
Yes, sir.
Yes, sir. You are audible,
Okay.
sir.
Thank you. So this is how I begin with. I wanted to talk about nematode diversity, and there are so many things together. It was very difficult for me to compile for a short title for my talk. You look for bioinvasion, and these are the minute creatures which invade any virtual ecosystem. But point was, how do we utilize diversity in the terms of what do we get after we get knowledge about biodiversity? So what immediately struck to me was evolution, that is the introduction of species or extinction of species from the chain. Anything would lead to the break in chain, and that is how evolution is either disturbed or deviated from its normal path or natural pathway. So that is how I choose nematode diversity, invasive reverse co-evolution. Evolution as such, this is what a nematode of mammal, and that is, has my slide changed?
No, sir.
No, sir.
But I have changed my slide. देखिए इसको फिर से रीशेयर करें. Still not changed? What? Do you get second slide? उसको इधर
No, sir. No, sir. It is still.
I'll redo it. बंद कीजिए, stop sharing करके दोबारा करिए. नहीं नहीं, वो तो फिर दौड़ता चला जाएगा, है ना. इसे मत छेड़िए, आप इसे बंद कर. हाँ, वो इसे बंद करिए. स्टॉप शेयरिंग आएगा ना. कीजिए, वहां कीजिए, पहले डबल क्लिक वहां कीजिए, डबल क्लिक वहां. प्रेजेंटेशन अपनी जगह है, उससे डबल क्लिक कीजिए तो शेयर आएगा. देन यू विल शेयर. डबल क्लिक कीजिए. ये अब शेयर कीजिए. Bear with me. The presentation is not coming.
Professor Malhotra, you please share window, not entire screen. So it will work. There may be one option on right hand side, share window. And open the window first. Open the window first and share. If there is any trouble, please call me or ask your student to call me so we can guide him.
Yeah, we are sharing window only. Just wait.
Okay.
The presentation is not showing on the screen.
Earlier, when you log in, it was showing, it displayed the first slide we have shown.
But only first, it stuck
on
up
the
there.
first, yeah. It was not moving.
I'll try to join to, afresh. Huh?
Please, please.
This window is open, share an application window, but there is no presentation here, only window. Okay, I'll rejoin. Please give me some.
Dr. Anjum, are you with us? Dr. Malhotra is working on his presentation. Dear participant, please be with us. Meanwhile, you can put your data in the feedback. On putting the entry, you can download e-certificate. You may also put some of the questions or the requirement, what you want from us. That will be helpful in organizing the next event. If you need another kind of activity. Yes, Dr. Malhotra. Now we can see your presentation. Please unmute yourself, Dr. Malhotra. Dr. Malhotra, would you please unmute yourself? Your slides are perfect, they are moving. Yes, yes, it is working now, Dr. Malhotra. Yes, please go ahead.
So this is one nematode which is found in mammals. And what I wanted to show is their advancement in development of mouth armature. That is ankylostoma. You must have heard about this. And this is the second one. I'm getting you an introduction of how does it look like in primitive organisms. This is from a fish nematode, and Procamellanus is the genus. You find a buccal capsule but without any armature. Earlier you saw a heavy armature there. This is the third one, which has Paracamellanus, the genus, and it has cuticular striations within buccal cavity. Now, our study was basically through the river Ganges at Allahabad and down to Kolkata, followed by Gour, central west coast of India. So this was the stretch we covered for the last 30 years, and the results we are presenting here. Normal course of evolution will tell us that it passes from fishes, amphibia, reptiles, birds, and mammals. The evolution means the character emerging one after the other. These are the vertebrate groups, and you are very well aware that there are increasing complications emerging after group after group. What I am interested is that the parasites are within the body of the vertebrates, and when they are in fishes and the fish evolve, the parasites also evolve gradually along with them to enter into amphibia. Then amphibia would evolve into reptiles. That's the normal process of evolution. So the parasites also, which are present, the cystodes, the nematodes, they would evolve to settle down into the reptiles with advanced characters, followed by up to the aves and mammals. So the mammalian is the highest one. The trouble arises when suddenly we started finding parasites, nematodes with highest form of structure which was present in the mammalian parasites, and these worms which we found occurred in fishes. So precisely repeating it, the fishes had the worms which developed organs of whatever oral armature to papillated structures found in worms are there. They were similar to the ones that were developing in mammals. So this is what is contrary to the process of coevolution, that they skipped all the in-between groups, that is no reptilia, no amphibia, no aves-like characters, and straight away fishes to evolution gave us the characters of that occurred in mammals. This is very, very disturbing, and normal form of diversity will gradually give us the way that to study these characters, to study the pathways of evolution of these characters, we will have to do something extra to understand that this is not co-evolution. Group after group, helminths are not evolving parallel to the host groups, that is the bird, mammal, and those kind of things. Professor Neelaman this morning referred to genetic erosion as well. So once again, this is finding some practical appearances here, that permanent evolutionary changes are brought about by the bioinvasive species, and suddenly some new forms will appear, suddenly some existing forms will become extinct. So this is the genetic erosion which has really disturbed our process of evolution. And that is why we are getting those. Let us study these things a little more. So these are bioindicators, the helminth communities we commonly encounter. These were the fishes, the coral reef ones, Jonius the Sumeri and Lujanus malabaricus, which were the focus of our study. And suddenly we found these organisms that were not earlier in occurrence. I'll explain a little later. Rotundo coloret is the one which is...
Sir, your slide is not moving, sir. Excuse me, sir.
कहाँ पर रुकी? Oral armature. There seems to be some trouble. This was the condition earlier as well. But you can hear me all right?
Yes, sir.
What do we do? There's a problem with presentation. What do? We go back. आ रहा है उसमें. Try करिए दूसरे. I'll try another system.
Hello, sir.
Yes. Yes,
Sir,
please.
zooming है ना आपके नीचे हिस्से में presentation के.
यार मैंने तो full screen किया हुआ है. I'm seeing nothing. I'm trying another system.
No, sir. उसी में नीचे हिस्से में presentation होगा. आप वहाँ पे click करेंगे तो presentation आएगा.
हाँ, presentation आया. उसको मैंने full screen किया. फिर उसके बाद sharing किया. And then I started. ये तो अच्छे से होता है. पता नहीं आज क्या है.
गुड इवनिंग सर. सर आवाज़ ही बहुत धीमे आ रही है.
बोलो ना, देव भाई।
बतियाना पड़ेगा धुरंधर महेंद्र ये जो जितेंद्र है मुझे आना फिर जा फिर वो दुनिया भर का
मेरा उसको इंस्टॉल करना पड़ेगा ना। अरे आप मेरा उस पर इंस्टॉल करके दीजिए बाबा। नहीं, मेरा जा रहा है स्क्रीन। जरा भी शेयर करो।
लेकिन नाम मेरे से जाएगा।
अरे जाने दीजिए, आप बोलिए ना। How is it now?
Still it is fixed. You please click the next slide.
Okay.
So no issue. There is, if you are
That
coming
is
from the
restarting.
another.
I've taken another system, so it's restarting.
All right.
Computer connect हुआ है।
तो बंद हटा दो यहाँ से।
नहीं मैम, इसको कहना है कि 8 बजे ये सबमिट कीजिए, क्योंकि बच्चे देखेंगे और भाग जाएंगे।
अच्छा।
क्योंकि बाकी जो है फिर उनको स्पीकर उनको सुनेंगे नहीं।
चेंज नहीं हो रही।
नहीं क्या प्रॉब्लम आ रही है?
नहीं वो सर सुनते भी नहीं।
ये स्लो है। सारी छुट्टियां हो गई।
Dr. Anjum is working. Dr. Mahulatra is working.
Something moving? Some slides moved. You all have mic.
Professor Malhotra, now we can see your slides, please.
Are they moving?
Yes, yes.
That's what I was asking for.
Yes, it is moving.
चल रही है पीछे, बोलिए।
Are they moving now?
Yes, Professor Malhotra, it is moving.
The slides are moving now.
So this is what I was explaining earlier. I'll try to
Yes.
redo this. Actually, we entered into the zone of complications of buccal capsule, and this has some cuticular striations which earlier Procamellinus did not have. You can see the buccal capsule without cuticular striations. And finally, what I wanted to inform that when we started gathering from a larger area in Goan coast and compared it with the Gangetic areas, some of the worms just coincided, and we did their nucleotide analysis. They were the same. So the question was that from fishes to amphibia, reptiles, birds, mammals, the gradual evolution was taking place simultaneously in the parasites as it was occurring in the vertebrate hosts. So we understand that from fishes, the parasite, the helminths, the cystodes evolved, and then they occurred and enter into the first grade helminths, that was the parasitic trematodes. Unfortunately, this was the paper in Zootexa whose editor left for his heavenly abode, Dr. Norman Dronan. We published this paper in collaboration, and from America, I got the news 3-4 days ago that he is no more. So it's a tribute. The question was, this was plagioporine worms which were occurring in fishes of Goan coast, and when we analyzed these creatures, they were present in the Atlantic Ocean, Pacific Ocean. As many as 21 species of plagioporine Neolborea have been described up till date, and we found this 22nd species from Goa. So the question was, how did it occur several thousand miles away? And let me tell you something of the major events that were occurring in Indian Ocean and Atlantic Ocean that appeared 200 million years ago, and Arabian Sea that connected 100 million years ago. These three oceans had the environmental implications due to which these parasites got a connection from between Indian and American species. And Tethys Ocean was the youngest, 65 million years ago, when it merged into Arabian Sea finally. So what we got in hand was the worms of perciform fishes. They were occurring in the waters connecting Indian Ocean and Antarctica via Tethys Ocean. So this was the final result of the drift, continental drift, that occurred several million years ago. And from Cretaceous era to Mesozoic, this was the fish, perciform fish, which was present in Goan coast when we got this parasite. So this is very well known that the helminths tell us the story of archaic past, and this was one of the beautiful example. These were analyzed not to be paraphyletic in association, but appeared in the Cretaceous period of Mesozoic era. 21 species, I told you they were existing earlier. Simultaneously, it was analyzed that the role of crustaceans in Indian Ocean and Arabian Sea, along with the Atlantic, where -20, -32 degrees temperature was there. But the crucial factor was that Indian Arabian Sea did not have that much of low temperatures, but still these two similar parasites occurred in two far-off continents. Possibly there was some trigger existing already, and that got the push in Indian Ocean and Arabian Sea, where Neolborea was found once again by us after so many years. So that is what I was talking about, that Professor Nilima also mentioned about genetic erosion. And these are the factors which exist in nature, and evolutionary changes involve these kind of environmental implications as well as genetic erosion kind of thing. That is how we get down to newer species. And we now proceed with the other helminths that were being found in our study. Rotundo colored kapuri was the one described only 15 days ago. Procamellinus, paracamellinus, Dr. Anjum will know these are the ones on which she has been working for long. Trypanorhyncha, Eustrongyloides. These two fishes, Jonius, the Sumeri, and Lujanus malabaricus were the ones that existed before tsunami came in Arabian Sea, where we were working. In 2004, when we got into this, okay, can animals predict disaster? This was the brief intervention. Yes, in interviews with scientists and eyewitnesses, nature probes the evidence that some animals may have senses that allow them to predict impending natural disasters long before we can. So now you have many examples in mammals also, and why do these things occur? You must have studied a lot of things about scientific facts and physical theories. Some creatures are able to hear infrasonic sound produced by natural phenomenon like earthquakes, etc., that are inaudible to the human ear. Second reason is animal sensitivities to electromagnetic field variations as well as to shifts in atmospheric pressure. For example, fishes and other organisms in water. But our focus was on parasites. So these helminths, what happened to them when tsunami came in December 2004? You understand this is a major disaster, and what we got physically, earlier paracamellinus was occurring in Lujanus malabaricus, and after tsunami came in December, when only one fish species had paracamellinus, there were tens of species, so the host range became widened after tsunami, and these worms started appearing in several a great variety of fishes. So that was perhaps the barrier which the tsunami like of environmental disaster changed, and the parasites spread out. What is the specificity of paracamellinus? You find the two capsules, the division of the capsule arrow marked is showing that, and the teeth on the right hand side, maybe your left hand side, and these teeth in the buccal capsule, the cuticular striations. These are the paracamellinus characters, and this is what we got now. Look for the trident, the smaller one on one side and the larger one on the second side. This was never found, both tridents together and buccal capsule with striations. You find the yellow lining, cuticular marks inside the buccal capsule and esophagus later on. The scientists of these years have classified this as a separate genus. When we find a buccal capsule with striations, they call it genus Spirocamellinus, but no trident. And what we are finding here is the two tridents on dorsal and ventral side, and combined with buccal capsule with striations. So our trauma is now the characters have started appearing, which were earlier not seen in this worm. And not only this, the characters which were usually found in the mammalian worms, Physelepteran worms with the great amount of papillae. I'll show you how many variety of papillae are there. So these kind of things are fun. These kind of characters now started appearing. That is why we had an idea that there are some environmental changes are triggering these kind of appearances, which were earlier not present. Look at the mouth, the buccal capsule. On one side is male, the other side is female. Again, I repeat, the one with cuticular striations was called paracamellinus, with buccal capsule divided into two that I showed in the earlier diagram. But when you see the female-like oral capsule, buccal capsule, where striations are absent. So the two different genera are now, the characters of the two different genera are now appearing in the same genus in male and female. So that is sexual dimorphism. Earlier it was not seen. This is very, very common worm, and it has been studied 100 years ago. There is hardly anything like striations with present in one species, and in the same striations are absent. So now that was the time when we had to study it or analyze what are the other characters, and ultimately we looked for papillae. Trident is okay, CM structures are there, so there is hardly any doubt. These are the confirmed studies. I must underline that none of these diagrams or photographs are from any book. These are our self-sketched photographs, SEM projections. And look for the papillae on the white one, the right one bottom. These are the papillae on body. Every organ, practically around the mouth, the oral circumference, you find very large papillae for a worm of this kind. And to tell you the quantum of these papillae in Paracamelanus and Procamelanus, hardly totaling 28, hardly. And now what we are getting, these papillae are 148 in numbers. So that is different organs, different functions, and this is the posterior part. Look for the tail end, and this is the terminal part. The bottom left hand side is the terminal part, which is called as mucron. Up till today, mucron has been found as a one single organ having two terminal spikes, or maybe more in different species. So they divided species on the basis of number of spikes. There were group of spikes, there were 2, 4, 5, 10, and they called them different species, different genera. Now look what is happening. The whole mucron, this bottom organ, is divided longitudinally into two pieces. That has never been the report. Never ever this kind of mucron has been reported, and spikes are only two. That means one piece has one spike, the other piece has another spike. And to complicate it further, it has a pore at the bottom. It is very clearly visible, you can see. Normally we had anus as the terminal end of body, but on the upper part you find anus. I'll show you a little further. It's very well marked. See what kind of a bulging is present to show the anus. This bulging, you can read anus also, it is written here. So, and the mucron in the upper picture. So anus and mucron are two distinctly separate organs, and there is no opening after anus in a vertebrate body. That is meant for the terminal end of digestive system. And here what we find is mucron. At the base of the mucron you find MI is written for minute papillae, and beside this is the opening. Mystery is still there. I'm not able to decipher what is this opening about, because mucron doesn't have a tube inside. Anus does have digestive variety, and by the side of this is a ring-like papilla. You find it like a key ring. That is the kind of structures they have started appearing. It's a real mystery that you find these serrations, and serrations with margins as RE जिसे कहते हैं ना, saw blade type of appearance is there. Then buccal teeth are at the bottom. 6 teeth are the normal character. They are more than 6, maybe 12. And these are the circumference of oral. Normally we find heavy muscular lips, but there are no lips in this worm. It's only a white ring you find. This is a minute, fragile circumferential structure, and on this are 14 lips. The word C is written at every single lip. So you can find how many types of complications are appearing in this worm. And the best was TF, the bottom frill. That is a separate structure which is Physalopteridae character, the family Physalopteridae, that is entirely different from Procamelanidae. And what we find here is a collar. So this collar is a specialist organ which would necessitate separation of this species from this family altogether. I believe we will have to go in for erection of a new family, and that is the mystery of complications appearing or evolving in such simplified organism. This is the tail structure, the caudal, papillae, and the LE are there. So this is all about this worm. First thing was the variety in numbers, that is what you call diversity. And what I see at the micro level is within diversity, you find the divisions of characters, and they are innumerable papillae, buccal capsule specialties, which you have seen. Then muscular lips are disappearing. Sometimes they are becoming so simple that only a fragmented ring is present. But specialized kind of papillae are appearing. Then you have looked for the structure of anus that has fortified with nerve rings, white colored nerve rings, which you saw was quite mature, quite stealthy. And these are the worms which scattered, increased in number and variety. So a simple environmental disaster can produce into so many different kinds of things. And to tell you frankly, this parasite has the habit of sucking blood inside the alimentary canal by using its lips attachment with the lips, papillae for attachment, and simple papillae you have seen on the ring also, which I told you, like with the alphabet C. These are for attachment and sucking blood. So this is a dangerous parasite, technically evolving all through, and what we found was becoming dangerous. We were working through, you may not be able to decipher all this, but I tell you, on the one side column are the months. We conducted 2.5 years studies continuously in Gour coast area, and bottom 6 lines you find 6 columns, or rows rather, you find 0. That means this parasite disappeared during the month from May, June. I can't read that, but in the later months of May, June, and these kind of 6 months, there was no parasite seen. Suddenly it disappeared. That is what I told you, that it was appearing in a single fish, but suddenly it disappeared when tsunami came. So this was the period when tsunami came in December 2004, and ultimately when it appeared in great numbers and great number of wide variety of fishes. I call it zoonosis because, when it enters into human system and then causes damage, the pathogeny is linked to zoonosis. The anisakid worm under investigation is a zoonotic organism, as its infections have been recorded in human beings. Over the period of 3 decades, Procamellanus has transformed to coexist firstly with Paracamellanus with trident, and have recently changed to Pronecid. The gross amount of changes which have accompanied all through, I told you the evolution from Procamellanus to Paracamellanus, and now what was happening, which was more dangerous, is that evolution of alimentary system. This character, the arrow shows, it has a ventriculus here. I'll give you a line diagram to understand that. Now you see, Procamellanus, which you saw earlier, has a simple alimentary canal. Then oesophagus in Paracamellanus is rather simple. But when we enter into that area, when great number of papillae are appearing, you have seen those photographs, all sorts of complications appearing. What is happening along with it is there is a ventricular appendix and intestinal cecum evolving together. To tell you frankly, this is another species, genus, whatever you call, this is another family altogether, Anisakidae. Anisakidae is the only one which is causing in the ocean and They were not reported from fresh water for quite longer period of time. We found it in river Ganges now, and they have ventricular appendix, intestinal ceca, and whatnot. You can now understand that a simple worm that belonged to the fish on the left-hand side has now developed credentials to survive in a mammalian character, with a mammalian character inside human beings. So now we have to survey really, because earlier we found these worms in river Ganges from Allahabad and Fatehpur. But now only 2 months ago, 2 years ago, 21, we found it in Gomti. So you can understand that now it is spreading and nobody is paying any attention to this. These are very, very small creatures, but gradually transformed into a mammalian worm. Earlier, it was not. It was simply not able to survive in mammalian systems. You can understand the great amount of physiology and strength it requires to survive those hardy chemicals within the body of a man. It will not survive, but now it has developed those characters of late in 21, 22. This is the purpose of telling you what diversity has to give us. Counting only numbers was never my interest, and that is how I proceeded with nucleotide analysis. You will find, if you can make out, the pronaked is the name I'm going to give this, and family Tridentidae I will separate than Anisaccharidae, because basically it has the characters of Procamelonus and along with it the characters of Anisaccharidae. This is very, very dangerous, and that's how on the nucleotide analysis it is placed. We have submitted it to NCBI, and this says characters of Anisaccharidae. It is in the clade of Anisaccharidae and very far off from the clade of Camelonidae, although it has characters of both families. So for us, it's a very difficult thing to place them now. This is how a taxonomist will become in trouble. Summarily, 20 odd papillae in Procamelonus, I told you, versus 148 papillae which has now appeared in Tridentocameline and ID, new family. Collarate of Physolopteridae. This collar is not of Procamelonus, and this is come from Physolopterus. Buccal capsule of Paracamelonus, tridents 2 of Camelonidae. So it has characters of Physoloptera, it has characters of Paracamelonus, it has characters of Camelonus, then Paracamelonus, then what more? It has dentigerous ridges in buccal capsule, which is a character of Spirocamelonus. People fought over it to call it a new genus, and you find it in one organism. Specially strengthened anus with rigid nerve supply, two-fold mucron, you find longitudinally divided into two with two spikes at the tip, and the multiple minute and sessile papillae on the mucron itself with a pore. I'm yet to find any duct emerging from a tube which opens outside mucron. Anus, everybody understands, but mucron pore, what is it? I don't know. Buccal capsule not split into two like Paracamelonus, so it is more like Procamelonus, but bears 11 to 16 cuticular striations. So that is the new worm which we are finding now, and it's very difficult to place it under any condition. And we find this mammalian characteristics wala worm appearing in a fish. It has reversed the coevolution process, and that is my conclusion. I think given the time we have, I had a very long lecture, but I'll not bore you anymore. I'll stop here because my purpose is served to make you understand, if you follow this, that coevolution is the normal procedure, but here is the worm which has reversed coevolution because it is invasive in features. Thank you, Dr. Shweta.
Dr. Malhotra, you have given very important information. We are not. Hello, Dr. Malhotra. Dr. Malhotra, you have given very important information, and we are not in position to take more questions. I would request participants to put your questions in chat box. I think Dr. Malhotra, can
Are
you
you
listen
getting
to
my
me?
voice, Dr. Shweta?
I am getting you. I am getting you, certainly. Uh, thank you. Thank you. Dr. Anjum, could you please speak?
Yes, sir.
Yeah.
I think yes, Dr. Anjum, could you please conclude?
Yeah. I have concluded.
Okay.
We have
I
we have
am
at least one more two lectures again.
So,
We
Dr.
have
Malhotra, yes, madam. Actually, I was thinking that my voice is not getting to Dr. Malhotra, so I requested Dr. Anzum to pay thanks to Dr. Malhotra, but now I am audible. Perhaps am I?
Yeah, you're.
So,
I could
Dr.
not understand. Yeah. Please go ahead.
My voice was breaking actually. That's why. So thank you, Professor Malhotra. You have given wonderful talk, and we will in touch of you to get more information over the nematodes, and we will try to see you in
Sorry for
physical
those
presence.
interruptions.
It happens many times. It happens. Thank you, Dr. Malhotra. So,
Yes, sir. Thank you, sir.
Dr. Anzum, shall we call Dr. Rajendra Kumar? Dr. Rajendra Kumar, are you here?
Yeah.
Yes.
Good afternoon. I am very much
Sorry,
here.
sir.
Yeah.
Okay. Sir, everybody is facing this connectivity issue. Today, the issue.
Right now, we are facing this challenge. Perhaps our system are not supporting for more hours. Dear participant, we are having another important scientist, and I am presenting his biodata. He is having a wonderful biodata. He is Professor Rajendra Kumar, principal scientist, Division of Genetics, and he is the former Director General of UP Council of Agriculture Research, Lucknow. Professor Rajendra Kumar holds postdoctoral degree in agriculture biotechnology with specialization in genetics, plant breeding, and molecular breeding. Professor Kumar has has multiple organization experience in various capacities in SAUs and ICAR post system and several international organizations. He has made outstanding contribution in the field of agricultural research
इसको बुलाइए.
and education by way of commercial release of 16 varieties of pulses and vegetables, development of 50 technologies, and publication of more than 140 in referred research papers in journals of national and international repute. And he operated 30 research projects, including multinational, collaborative, and the supervision of 24 of students for their thesis work. Professor Kumar has demonstrated his academic leadership capabilities by establishing ultra modern research facilities like phytotron, genetic engineering labs, and initiation of diverse research academic endeavors, including PhD programs in biotechnology. He has expanded activities of UP Council of Agriculture Research by staff strengthening budget enhancement, large number of the projects, sanction and strong project monitoring and evolution mechanism in place. And he is also the founding president of Uttar Pradesh Academy of Agriculture Science. Professor Kumar has proved as a vibrant agriculture think tank for the UP state and has been conferred with numerous prestigious academic recognitions by Indian Science Congress, INSA, NASI, UP government, and latest one, he was awarded DST National Award for Promotion of Science and Technology for 2019. So he has many contribution to his credit, and I would like to welcome Dr. Kumar to deliver his talk. It is now over to Dr. Kumar.
Thank you, Professor Sweta. Am I audible? And what's about my slide? Is it visible?
Yes, it is visible, and your voice is also audible. You are perfect. Please
And
go ahead.
is it slides are rolling out?
Yes, perfectly. It is moving.
Okay. Thank you very much. So, respected chair of the session, Honorable Vice Chancellor of the coveted university, and dignitaries in the audience categories, as well as dear audience. I am going to present my deliverance entitled Unsnarling in vivo and in vitro crab breeding strategies in changing climatic scenario for biodiversity conservation and boosting ecosystem efficacy. As we know now, biological diversity has become a buzzword, and Convention of Biological Diversity defines it as variability among living organisms from all sources, including inter alia, terrestrial, marine, and other aquatic ecosystems, and the ecological complexes of which they are part. In brief, one line we can say all total of gene of a living entity in a given ambient. Thus, biodiversity can be found in three different forms: genetic diversity, species diversity, and ecosystem diversity. Whenever we talk of the genetic diversity, of course, it means gene pool in a living system. Species diversity in terms of the different species or genera across the plant and animal system. Ecosystem, of course, is a complex word because ecosystem contains both all living systems along with given ambient, including non-living materials like light, temperature, water, etc. So and so. The challenges ahead humanity has to face. The right now challenge we are facing: how to feed 9 million people on the globe. Due to escalating population and climate change. As we all know, Indian population is around 1.4 billion right now, which is going to touch a mark of 1.66 billion by 2050. Accordingly, by 2050, global population will be more than 9.6 billion. And the very reason is very fast rate of population growth. Every day, our population is increasing by 2.4 lakh. Means 3 people are taking birth, or 3 children taking birth every second on the globe. So really, it's a big problem before us. Next one is the major challenge is the climate. We all know to feed all these human population, which is escalating day by day, whenever there is a temperature growth, usually this temperature or heat escalation gives a decline of around 5% in the agricultural yields. Not only that, if the rainfall is a deviation from the normal annual rainfall, then we are going to touch around 15% decline or reduction in the agriculture produce. And these all processes are producing a huge quantity of greenhouse gases. Once again, these greenhouse gas emissions are creating a big challenge before the human big science. Indian and world present scenario in terms of biodiversity, particularly in the flora. India represents around 34 globally identified hotspots. Besides, there are around more than 167 important cultivated plant species and more than 320 related wild species to the important cultivated species, number of 167. Besides, India is a home or center of origin to several of the cultivated crop species like rice, pigeon pea, mango, turmeric, ginger, pepper, banana, bitter gourd, okra, coconut, cardamom, jackfruit, sugarcane, bamboo, taro, indigo, sunhip, amaranthus, gooseberries, mango, etc. India domesticated animal wealth is also very rich. It includes not only cattle, rather fish, poultry, etc. Accordingly, when we talk about fauna, the present scenario in India represents 7 to 8% of all recorded species, including 45,000 species of plants and more than 91,000 species of animals, including human beings. Being one of the 17 identified mega diverse countries of the world, India has got 10 biogeographic zones with a home of around 9% of the mammalians, around 40% are the avians, around 8% of reptiles, and around 5% of amphibians, and around 12% of fish species, besides around 12% of the plant species. Global warming or climate change. Now, with such a mega diversity which we represent, that's not confined only to the Indian scenario, rather entire of the Asian countries are very rich as far as biodiversity is concerned. Global warming is the increase in the world average temperature in the form of different greenhouse gases like carbon dioxide, methane, SFCs, PFCs, SFCs, and N2O, etc. Besides these, number of other factors or other minor gases also are emitted in the nature. Millennium Ecosystem Assessment Cell predicts the average global temperature has increased by 0.6 degrees centigrade since 1800 and is predicted to rise by 1.4 to 5.8 degrees centigrade by the year 2200. If we talk of the global rate, so it's going to be per decade increase in the temperature 0.15 to 0.2 degrees centigrade per decade, which that rate is also increasing very high due to the global climatic scenario, particularly in terms of the greenhouse emissions. Besides, the global mean sea level has also risen up to 10 to 20 centimeter, which is about to be 90 centimeter in the days coming ahead. Thus, these global warming and climate change are influencing the flora and fauna in different forms, like early leaf production by the trees, early greening phase of the vegetation, change timing of the egg laying and hatching in case of the animals. Accordingly, short duration of the crop cycles, changes in the migration patterns of the bird, fish, and other animals. Thus, greenhouse gases emission are the major crux bottlenecks for our overall growth, whether we talk of the flora or we talk of the fauna. So we can see in the current, this is the latest data, that when we talk of the land use change and forestry, that emission is due to this part, only one sector, we can see that the CO2 emission is higher in 2020 in comparison to 1990. Accordingly, same condition is also in case of forest and peat fires, land use change emissions, besides fuel gases like total N2O due to agriculture, due to industrial process, due to energy utilization, indirect air wastage, methane waste and other, then methane due to agriculture produce, methane due to energy consumption, methane carbon dioxide due to other non-energy consumes, and carbon dioxide due to international transportation. Besides energy, carbon dioxide. So we can see this graph shows very clearly that in comparison to 1990, the growth rate is escalating every day, and in the 2020 we can see that this is almost double. We can see it was around 35 or 38 now gigatons carbon dioxide. Now we are going to touch 60 gigatons of the carbon dioxide emissions in the nature. So another slide I'm going to discuss energy related greenhouse gas emissions. Like we can see different parts of the fuels like coal, oil, natural gas, waste, industrial process, methane, nitrous oxide, carbon dioxide, etc. Every day, this is a comparison between 2000 to 2031 as per IEA report, very recent report, March 22. We can see that the emission is increasing, and now this is almost once again going to touch the double figure in terms of the gigaton carbon dioxide emissions. Accordingly, we can see the same greenhouse gas emissions or carbon dioxide emissions per capita with a comparison or comparative study amongst the advanced countries or developing countries. In other terms, we can say advanced economies, emerging economies. So let us see that, of course, advanced countries or developed countries, they have tried to reduce their emissions to certain extent. In the right hand side, we can see that there is a comparison in 2000 to 2021. Of course, everywhere in case of all these three lines show that there is a reduction in the greenhouse as well as carbon dioxide emissions in case of developed country. But opposite to that, let us see or make a glance over developing countries, including India. So if we see this bar, we find that of course India has also controlled or limited its greenhouse emissions, but opposite to that, China has increased its greenhouse emissions like anything almost threefold. So that's a challenge. So we have to look into how these greenhouse emissions can be minimized. And it has become not only a regional problem, rather these emissions have become a global problem. That's why very recently, Convention on Biodiversity is going on in the name of COP15 in Montreal, Canada. And it is being chaired by China, of course, hosted by the Canada itself. And there was a slogan that is under the sponsorship of the United Nations. United Nations Environment Minister Cell says that we have to minimize our war against the nature or our war for nature. So that way, that's the slogan. And of course, COP10 Nagoya, Japan Convention 2020 had declared or decided some of the targets point in the name of the Aichi targets. Those targets were 20, but those targets due to COVID and other global pandemic situations could not be implemented. Now, those points are being mooted. That how those achieved points can be implemented, and further how these gas emissions can be reduced. Eh, Under the process of discussion, there is a demand by the developing countries that financial support should be increased. However, developed countries are not mentally prepared to fund more because they are arguing even the already allocated fund is sufficient enough to meet out the target finds. So there is a managerial issue. Once again, still those discussions and debates are going on. Let us see what decisions are coming out of this conclave. Thus, we can see stabilizing atmospheric greenhouse gases levels. The first important one is the biodiversity conservation. That's most important. Second one, the transportation efficiency has to be improved. Accordingly, energy conservation as well as utilization has to be improved. Besides, we have also to focus for the sustainable utilization of the energy resources, and not only that, our resources in terms of the land, water, etc., all those has to be utilized in a sustainable way. Besides, population escalation has also to be stabilized. Thus, we, we see that climate change has a larger impact on the biodiversity. The Millennium Ecosystem Assessment that was done in 2005, it says that loss of the natural habitat are due to greatest threats to the biodiversity. So natural habitation has to be protected. Once again, how to be protected? Once again, these are the debatable and mutable issues and being discussed even in the biodiversity conclave as well. Accordingly, South Africa National Spatial Biodiversity Assessment says that 40% of the terrestrial ecosystem, 57% of the main river ecosystem, and 65% of the marine ecosystem, 44% of the estuaries, and 44% of the offshores, and 59% of the coastal and inshore resources are under threat. So there is a challenge. Not only that, as Professor Nilma Gupta uttered in the morning, that every day around 100 species are on the verge of the extinction. So here we can see that Indian flora under threat categories are around more than 1200. These are under threat as per the report by IUCN 2030. I'm confining my deliberations only confined to the flora, I mean to say plants, because animals, my predecessor speakers have narrated and discussed in detail. So I will not like to repeat all those, so that just I'm skipping. So, but anyway, global biodiversity loss, how it can be minimized? There are very limited options like expanding protected areas, particularly in the in situ strategy, reducing deforestation and urbanization, adoption of integrated crop management, wise and judicious utilization of natural resources, already discussed, adaptation and mitigation to climate change. That is the point we have to focus, improving forest management, and using innovative technologies for the biodiversity conservation. So now our focus will be only for the biodiversity conservation with all those bullet points. So IUCN, UNEP, and WF in 1995 came out with the first global strategy for the conservation of the biodiversity and the defined and management of human use of biodiversity, so that it may yield the greatest sustainable benefit to present generation while maintaining its potential to meet the needs and aspirations of future generation with involvement of 2 points: conservation as well as sustainable utilization or sustainable supply. Now I'm coming to my topic, basically with all these basic facts. conventional crop breeding approaches for the biodiversity conservations. I categorize these crop breeding approaches in three parts. One confined to self-pollinated crops, another confined to cross-pollinated crop, and third one is confined to the vegetatively propagated crops. So in case of the self-pollinated crops, usually we practice these seven approaches like plant introduction, pure line selection, mass selection, pedigree breeding, the bulk breeding, heterosis breeding, multi-line development. I am not going in detail because once again, I think time is a limitation. And moreover, this is a multidisciplinary platform. So that will be injustice if I go in detail with all these specific topics, specialized topics. And in case of self-pollinated crops, particularly utilizing the plant introduction and selections and pedigree method, pure line selections, etc., modified versions, we could be able to develop a large number of varieties in case of pulses and vegetables, like desi chickpea variety Pusa Parvati, then chickpea, Kabli chickpea variety Pusa Subra, then chickpea variety once again Kabli 2024, and Pusa 1108, once again Kabli chickpea, and desi chickpea Pusa 1103, once again Kabli chickpea like Pusa 1105. Then another lentil variety like Sekhar Masoor 3, and Ajad P4, one of the vegetable pea variety. These are the self-pollinated crops varieties which could be able to develop or applying these approaches. Besides cross-pollinated crops, of course, for the cross-pollinated crops, we apply four approaches like recurrent selection, synthetic and composite breeding approach, hybrid varietal development, and backcross breeding. Backcross is a technique which is equally applicable in case of the cross-pollinated as well as in case of the self-pollinated, both. Both ये रेनू जरा ये टेलीफोन हमारा एक आ रहा है, बहुत इंपॉर्टेंट है, इसको उठा के उधर बोल दो कि कहीं मीटिंग में है, बाद में बात करेंगे, आप उधर बात करना ले जा करके. So backcross breeding. Besides, then comes the vegetatively propagated crops like chloral selection and polyploidy breeding. So when we comes to the cross-pollinated crops, we could be able to release three varieties of the vegetables like okra variety Ajad Bhindi 2, okra variety Ajad Bhindi 3, and okra variety Ajad Bhindi 4. Besides, we could also be able to release one variety in case of true vegetatively propagated method, that is a turmeric variety selection 4. So these were the varieties we could be able to release at the national state level in the different release committee, national as well as state release committees, and all these varieties have been notified by the central notification committee for different states as well as some specialized states. Besides, we could also be able to identify some specialty genotypes like Pusa 1108 for the very high antioxidant resource. Besides, ICC 1083 for the low raffinose content. Besides, JG11 as a resource genotype for the nitrate reductase enzymes. And then there is a huge number of genotypes like ICC 1064, ICC 13579, ICP 14638, and ICP 14976. Water-logging tolerant pigeon pea genotypes. Besides BG 1092, ICC 11378, JG 74 at potential resource for the protein and nutritional traits. Besides ICC 13816, ICC 15697, ICC 15610, ICC 15868, ICC 15888, ICC 15996, genotypes for the chickpea diversity breeding. Besides tomato genotype for the pusa sital, for the organic farming and better shelf life. In addition to these specialty genotypes, there are many other genotypes we could be able to identify for the specific purpose to be utilized in our breeding programs. Besides, we could also be able to develop 4 food products. One potato flex energy bars that has already been patented. Only this year we got the patent. Uh, and one chickpea protein isolate from the genotype Pusa 1103 and K850. Besides, we could also be able to develop biscuits, potato-based Kufri Chipsuna, one enriched biscuits, which is very nutritive and useful for the infants as well as older age people. Besides, we also developed husk-based chickpea husk-based biscuits. Once again, these all are the published informations. Then I'm switching over to the biotechnological approaches in biodiversity conservation. In one go, I would like to say biotechnology we don't consider as a separate tool. Rather, these we consider as a supporting tool to the conventional techniques in the plant breeding approaches. So these biotechnological approaches basically started with the recombinant technology development in 1973, and the first molecular marker system was was identified in 1975 by Otis et al. And 1918 onwards, we have started our intensive activities for the utilization of the recombinant technology and commercialize the products. So with the all increased growth surface by genetically improved crops that provides much needed resources in the effort to conserve biodiversity, like improved tools such as cryopreservation for the ex situ conservation, gene shuffling to create more diverse biodiversity, even recreate extinct crop traits, and besides use of DNA markers to provide insight into the dynamics of biodiversity in crop plant restoration ecology. Ex situ conservation, once again, by ex situ, there is always confusion in vivo and in vitro techniques. That's by and large, with a minor deviation, these are the parallel work to in situ and ex situ techniques. So ex situ, we can say that is very much parallel to the in vitro techniques. Whenever and once again, ex situ or in vitro techniques only can be a supplemental tool to the in vivo or in situ technologies or conventional breeding technologies. So collection of species and races for the ex situ conservation has now become a race against time, and a range of methods have been developed to collect pure iron strains, avoid hybridization, and minimize impacts on wild population just by the saving of the time in order to escape the long life long life cycle, as well as for their grow out test. So these ex situ conservation has become a very fascinating word today. Besides ex situ conservation, sites are mentioned as gene bank, seed banks, tissue culture bank, seedling banks, conservation stands, and cryo preservation sites. So these ex situ conservation technologies are equally applicable for the different purposes, like embryo micro propagation can be utilized for the increasing the population. In case of the calcium transplants like orchids and other hardy plants, Besides somatic hybridization, once again for the increasing the population, like calcitrant plants, bamboo, cycads, and other crop plants. Besides coronal propagation of shoot tips, like once again increasing further increasing the population, like ornamental plants, artificial insemination, once again for the population increase, like in case of the tiger, Gaian panda, where due to some or either reasons, fertilization, either fertilization is poor or their conceivement is poor. So artificial insemination, also these in ex situ, even semen or ovum can be conjured through ex situ. Besides embryo accesses cryopreservation, storage of calcitrant species like Curcus, etc. Oak. Like cryopreservation of cell suspension, once again, calcitrant species can be stored like orchids. Besides, in case of breaking the dormancy, these ex situ techniques can be applied, particularly in the seeds of most substrates like several plant species, even in case of the cultivated crop plants, because there are a number of the crop plants where there is a long dormant period. So seed never germinate immediately. They have to be stored for certain period, only they germinate. So for breaking those dormancy, these ex situ conservation techniques can be applied. Besides, germplasm can be a huge quantity of germplasm can be protected through ex situ conservation in a smaller place, like several wild animals, etc. So and so. And several crop seeds like seed storage at low temperature, minus 20 degrees centigrade. For example, we are storing the crop plants at National Bureau of Plant Genetic Resources. Accordingly, in case of animals, National Bureau of Animal Genetic Resources. And once again, when we talk of the storage process, long-term, medium-term, and short-term storage process. Preservation of plants or seed in gene banks or in vitro cell lines or cloned tissue culture clusters under appropriate conditions or long-term storage often seems to be the easiest and more preferred mode. Gene bank collections and in fact many breeders collections are too large for scientists to intentionally investigate and record all traits in the screening process. So these are certain advantage that the large germplasm lines, these are very difficult to maintain for the scientist, either by the in vivo or in situ method. So in vitro in ex situ methodology through cryopreservation, these can be utilized very efficiently, but not only saving the time, rather resources as well as with the economic gain. Advantage and disadvantage of ex situ advantage conservation methods are like several, like seed banks, propagules readily available for use, minimum work, little space, provenance, like variation can be conjured, provide special, but the disadvantage is that these are not applicable in case of the certain recalcitrant plants or seed types. And moreover, where large seed quantity is supposed to be preserved, that is also one of the problem, because due to very high intensive and economic high economic input, large quantity cannot be stored. Accordingly, pollen banks, minimum space required, applicable for plants and are with recalcitrant or orthodox seed, but only half of the genome is conserved so far. Tricellular or pollen storage is problematic. Accordingly, tissue culture banks, minimum space is required, genetic erosion is reduced, and this is the beauty of the tissue culture technique. So there is a lesser degree of the genetic erosion. Besides, short time required to produce the propagules. The disadvantage are sampling is a problem, and besides some clonal variation occurs. If there is no proper transformation, even tissue culture technique cannot be effective in case of the recalcitrant problems. DNA banks are gene banks like minimum age is required. Species with high extinction potentials can be and applicable to all kinds of plants and animals. However, it's a very costly affair and very difficult to maintain, and not the very easy methodologies have been developed so far. So, and very skilled persons are required to maintain all these routine activities. Cryopreservation of OM, sperm, etc., like minimum space is required. All kinds of plants and animals can be stored. However, involves freezing with liquid nitrogen, very costly method, viability of the healthy clone, etc. Sometimes it is questionable that clones are really viable 100%, or their viability is reduced. Crop specific global network of the gene banks like rice, rice for the rice, Erie Philippines. We have the global gene bank. Accordingly, for the wheat, BIR Russia and CNR Italy, maize, CIMIT Mexico, and SSNL USA. In case of millets, ICSAT India and SSNL USA and ICR India. Besides oats, PGR Canada, and in case of sugarcane, ICR India, besides NBGS USA. And in case of potato, CIP Peru and ABRDC Taiwan. Groundnut, ICSAT India, soybean USA, and Physiolus German seed bank. So these are some of the international gene banks. Besides the present status of the national gene bank in case of the plants, we have around 4,79,000 total germplasm available at our National Bureau of Plant Genetic Resources, including the cryobank preservation and in vitro cryobank preservations. So these are some of the glimpses. Besides marker technology, this is the era of the marker technology. Due to some or other reasons, these marker technology have played a very great role as far as plant breeding is concerned. So the first marker system is the restriction fragment leg palmarium. That's very Herculean task, and of course codominant marker. But now this is rarely practiced, only for the academic purpose. We have stopped practicing. And as far as dominant marker is concerned, that was oftenly practiced. This was the RAPD markers. But due to dominancy, it was not results were not very reliable. So that is also the gone technology now. Then AFLP, once again, dominant markers, we don't like to practice. Accordingly, ISSR markers, of course, co-dominant, but due to some other regions, we not very popular. Besides SCAR markers, very specific markers, those RAPD markers, once we have identified some reliable markers, those RAPD markers can be converted to SCAR markers, and these are the co-dominant markers and very robust also. And of course, SSR markers are oftenly used markers nowadays. And as far as me is concerned, for last 1 decade or so, confined only to the SSR markers due to its robustness and reliability. So utilizing these SSR markers technology, we could be able to develop 4 marker assisted back cross generated 4 genotypes during last 4 years span. Like the latest one is the Pusa chickpea Siam. Very recently last month it has been notified by the government of India. Besides Pusa Vijay, then Pusa 400, 4005, and Pusa 20211. These are the four genotypes, varieties, not genotypes. Rather, these are the marker-assisted backcross developed varieties. And these now, with these outcomes, India has also come globally as far as marker-assisted breeding program is concerned, for the chickpea breeding programs. Besides, there are other marker systems like EST, then CAPS, not oftenly practiced, only for the academic purpose. And of course, now high throughput technology like SRP markers, these are getting very popularity, and everyone is trying to practice this because the output is very fast and cheaper also due to oligosaccharides sequencing and all those things. So single nucleotide polymorphism has become the buzzword today. And we are also trying to map the genes for the nitrogen fixation in case of chickpea. As we know, chickpea are almost all leguminous crops who work in the symbiotic fashion, because they work in the association, close association with some certain symbiotic bacteria like microbes, like rhizobium, ejectobacter, etc. And due to these all symbiotic functioning, I mean to say ecosystem, not only we are able to fix the atmospheric nitrogen in the soil, rather we are also able to enhance the crop productivity as well as survival or feeding the these microbes in association with the crop growth. So on all these issues, we are trying to have a in-depth study in case of the chickpea. So we could be able to identify one of the very highest nodules chickpea genotype ICC 9085 with the lowest one 1172. So and as we know that IRI is very rich as far as chickpea germplasm reservoir is concerned. So we have started utilizing the chickpea core germplasm that consists of around 200 genotypes. So out of these genotypes, we could be able to, through the GWAS analysis, we could be able to identify 7 robust SNPs, and those 7 SNPs we are trying to convert in cast markers, which can be utilized for the marker-assisted breeding programs in the chickpea. Besides, this year also under this study we had very multi-locational trials. There are huge number of 7-8 locations are involved across the country, and this year also we generated huge data. Those data are under analysis, and once these are done, we hope that we will be able to identify good number of robust SNPs which can be converted to cast markers and can be utilized in the further breeding programs, specifically for the high nitrogen fixation traits like several of the component traits. Salient finding of so far, because still project has not been completed. So far, the salient output of these projects is like the certain traits like number of nodules, freight weight of the nodules, root fresh weight, root dry weight, nitrogen estimates are one of the very robust traits which can be pick up for the high nitrogen fixing genotypes, and providing a good platform for the anchoring for the beneficial microbes like Azotobacter and Rhizobium, etc. And we could be able to also identify some of the genotypes like BG 3022, ICC 9085, ICC 2083, ICC 7761, ICC 7266, and ICC 1498. These can be utilized in the breeding programs. These data still we have not published, that we could not, I have not reported in the category of the identified genotypes. Accordingly, these ASNP markers can be converted to caste markers, and further we will be able to utilize in the marker assisted selection programs in the future days. On the basis of all these marker systems, we could be able to construct several of the linkage map, and more than thousands of the QTLs we could be able to identify it. These all are published and reported time to time. Like first RAPD map, linkage map, along with two QTLs for the oleic acid was mapped in case of mustard, that has already been published in Genome. Accordingly, a reference map using 1718 ILP markers and 6 QTL in chickpea, that has already been published in Journal of Plant Science. Linkage map and 10 QTLs for the Alternaria blight in linseed, that already published in and new 35 EST ESR markers like in case of vestige, and accordingly other markers in case of chickpea we have already published, and GWAS mapping and QTLs, and in case of sorghum and sugarcane. Besides, so there is a huge number of, I'm not going to describe all those things, but we exploited all these molecular marker technologies, and we have already mapped more than 1,000 QTLs in different crops with a span of around 2 decades of the marker technology conducted by us. Of course, this is the same, just continuity of that slide. I'm not going to discuss all these points. Besides unconventional biotechnological methods like micropropagation, of course, these are not oftenly practiced, but even micropropagation is now and then in few of the labs. Through tissue culture technology, micropropagation is taking place. Besides protoplast culture, then somatic embryogenesis and organogenesis and micrografting, these are the some of the propagation technologies are being practiced in the in vitro or ex situ ambient, utilizing the different techniques either through pollen culture, anther culture, tissue culture, etc. So and so. And cryopreservation, of course, that we are using at the very large scale, particularly our bureaus like NBPGR and NBAPGR, sorry, NBAGR. Then different transformation system, like in case of plant system, most of the are robust system we are utilizing. Agrobacterium tumefaciens mediated transformation is taking place. Of course, technology is upgrading every day. Now there are certain new breeding technologies are emerging at a very fast rate, like epigenetics. Once again, this is epigenetics. Coming days will be the era of the epigenetics. We published a paper in Frontiers only this year. In case on the chickpea epigenetics. Besides speed breeding, of course, to shorten the life cycle of the crop plants, speed breeding is also gaining momentum in in the days to come. Besides editing tools, in epigenetics and editing tools, these two tools are going to be the buzzword of the at least next decade for the utilization as far as crop breeding is concerned. Besides association mapping, already relatively new, but it's being used almost major laboratories across the globe, including us. Besides, there are number of, and not only that, there is a need for the nutrients availability and phenotype evaluation throughout life, and that's why IRI has also developed national phenomics. Of course, we are having the phytotran earlier, but now the IRI has also established phenomics very lately, and these are some of the new upcoming strategies which are going to give a boost up to the breeding technology in the days ahead. So we can see integrated breeding strategies are the need of the time for the biodiversity conservation, not only biodiversity conservation, rather even for the biodiversity creation as well as. So this will be an accelerating process to shorten the life cycle as well. So we can see germplasm evaluation, biparental and multiparental breeding population, marker assisted and selections. Besides, then double haploid approach, then genomic selection, then once again DNA based marker assisted technology applications, and once again field testing and new inbreds oblique varieties. So we can say that breeding can be a tool not only for the biodiversity conservation, rather in case it is integrated properly and utilized properly, it will even will create some new biodiversity, new variants of the biodiversity. So, in more simplified way, we can see that these are the different approaches, and we can see that those for the development of a variety, in case of the self-pollinated crops, earlier we are taking around 14 to 15 years. Now we are taking even 3 to 6 years time. So we can see how this biotechnology is going to play a role as far as crop improvement or plant breeding is concerned. So I'm not going to be in detail. So so now the ecosystem, how we can see how ecosystem is taking place. We all know food web, uh, carnivorous and herbivorous. I'm not going to in detail about those food web cycle, because even from the intermediate onwards, we have seen some, we have been reminding that there is a cycle of the different flora and fauna living systems, just categorizing in the category of herbivorous and carnivorous. So, but here we can see that how these variation is occurring. Net primary product, resource competition, species composition, disturbance patterns, and soil biotic processes are taking place. Then soil decomposition, mineral composition, and leaching, etc. And this is canopy atmospheric in CO2 and temperature. As I talked that for the nitrogen assimilation, we are working. Besides these, some of the vegetation, we are releasing different green gases, high green gases, including carbon dioxide, and the feedback like how plants respirate, trace gases from soil, evapotranspiration, albedo, etc., and carbon cation exchange taking place, whether how they are affecting. So we can say that ecosystem, for the ecosystem functionality, we have to work in a tandem approach. So this whole ecosystem is moving in a synergetic way, and our food web system continues. Besides, so we can, there are different provisioning services like products for obtain. Of course, this is a repetition of the previous slide, but we can say food, fresh water, fuel, wood, fiber, biochemical, genetic resources. Accordingly, we can have some regulatory service like climate regulation. We have to regulate disease regulation, water purification, pollination. Unless until we are going to regulate, probably there will be a crisis or crunch about these resources. Besides cultural services, also like spiritual and religious recreation and ecotourism, aesthetic inspiration, educational sense of place, cultural heritage, and supporting services. So unless until we integrate all these, we cannot create a functional ecosystem, which is the crux for our survival on this universe. So if we are able to have a harmonious ecosystem, certainly we will be moving towards a sustainable food future, and there won't be a crisis, and probably we will be able to meet out the requirement of the escalating population, and uh, to feed the population, and we can not only mitigate the hunger, rather we can fulfill the nutritional requirements of the humanity as a whole. So, message to take home is complete inventory and documentation of all biological resources are required. Databases are required to be prepared. Ground check to know the actual situation and identify the gaps. Those gaps should be fulfilled. Accordingly, genetic diversity, distribution pattern, association pattern, and all those should be studied. Identify rare and endangered gaps. Besides, how these DNA or information can be mutated, can be permutation, reassembling, etc., can be done. So, all these problems which are creating, even in the medical science, also like some clinical treatments, all those can be done. Besides, identification of the problems and solution in the conservation, threatened species, and ecosystem can be addressed. Thus, identification of the problems and solution in the conservation. So these are the requirements of the time, and certainly biodiversity conservation is going to play a big role. And let us look forward what Montreal conclave concludes, what recommendation come, and how we are going to cope with the challenges in the coming days. These are there are a huge number of the contributors and collaborators for the work which we have accomplished and reported in this oration. However, there are other huge number of works. Particularly, I have avoided about the germ plant characterization, all those things, because those are were the very preliminary studies. So I thought, let us escape all those things. Around more than 3,000 germplasm are available with the IRI as far as chickpea is concerned. And we have tried to make the genotyping of all those genotypes, and still it's going on. So with all these, we are thankful to all these collaborators spread over the globe and at the national level. And particularly, I am thankful to Dr. S.D. Upadhyaya, ICSAT, who has been the backbone for by all resource mobilization and motivation, who always supported me, and we could be able to make a good network across the globe, and we certainly delivered. And with all these things, I extend my hearty thanks, and I reiterate the slogan given by Atal Bihari Vajpayee ji, Jai Jawan, Jai Kisan, Jai Vigyan. So we have to integrate the science with the Jai Jawan and Jai Kisan. Only then we can propel the country to encounter the challenges ahead. Thank you very much.
Thank you, sir.
Thank you, Dr. Rajendra Kumar, for a very influential lecture. Many questions are doodling to my mind, but we are already getting behind, and time doesn't permit us to take the questions. If we will have questions, we will forward you and we will get back to you. Thank you, Dr. Rajendra Kumar, once again. And I would like to add Jai Anusandhan.
Thank you.
Thank you. So now this is the time for our next speaker, Dr. Kamal Kanan. And we people are very enthusiastic to listen to you, Dr. Kamal Kanan. Uh, I am just giving a brief of biodata of Dr. Kamal Kanan. He is an expert in taxonomy and ecology of Indian mammals, especially smaller mammals, bats, rodents, and insectivores. He is very particular in identification of species through hair samples, and he is studying the nitty-gritty of the taxonomy, trichotaxonomy of Indian mammalian species. And he had a vast experience in regard of the taxonomy. I would like to place his acknowledgement for the discovery of a new mammalian species, which is after 43 years, and that has been quoted by the Wikipedia, and he deserves for the appreciation for that. And dear participant, he has published several new records from Myanmar, Uttar Pradesh, Southern West Bengal, Chhattisgarh, and he was the co-investigator of the multiple set of the project. He has experience of funnel survey across the protected areas of the country, including Kaziranga, Sundarban, Kanha, Madhubalai, Bandipur, Barnard, and many more. He participated in the expedition of Great Nicobar Biosphere Reserve, Andaman and Nicobar Island. He has experience of delivering many talks in scientific debut. He has published 2 popular articles, 24 journals, and 3 e-publications, and 10 book chapters, and also published 5 books. I gave just a brief of his biodata. We pupils are very much keen to listen to you, Dr. Kamal Kanak. So please deliver your talk.
Good evening,
Thank
madam.
you.
I'm just sharing my slides.
Thank you.
I hope the slides are visible.
It is.
Yes, visible.
And it's moving also, no?
Yes.
So very good evening, everyone. First of all, I'm thankful for this opportunity. I'm grateful to my respected director, Zoological Survey of India, and the faculty members of Dr. Harisingh, Gour University, and the special thanks to the coordinator of today's workshop, Dr. Anjan Rizvi Madam and Sweta Madam. Since we are running late, I'll try to finish it as soon as possible. And today's topic of webinar is role of biodiversity conservation in ecosystem functioning. And the same aspect, I would like to talk about role of mammals in ecosystem functioning. So before we know the benefits or importance of mammals, let us fully and technically understand what are mammals. So we all know that mammals are warm-blooded vertebrates. A mammal can be distinguished by the presence of mammary glands, by the presence of a hair, and there are 3 unique bones, namely incus, stapes, and malleus, which is present in the middle ear. And there is a specialized brain structure called neocortex. So these are the 4 primary characters can tell you an animal as a mammal. With these characters, we can distinguish from the other types of group. So other than this, there are a number of features also different as mammals. Here I have given an example that the lower jaw made up of single bone also differs as mammals. Other than that, there are some behavioral and physiological characters also. Like the 4-chambered heart also differs as mammals. And we all know that mammals are placental. That means the animal can give a birth to live egg, unlike other mammals that basically lay egg. So since the brain and spinal cord are protected by a bony skeleton, mammals are presented within the subphylum called vertebrata. And mammals show the vast variety of extreme the diversity of forms. For an example, the animal like the bumblebee, bat, and shrews, they can weigh only just 2 grams. Just imagine, just 2 grams is also mammal. And there are mammals like blue whale. The ever lived animal of the planet is a blue whale, and it can weigh up to 1,06,000 kilograms. So this shows the extreme morphological diversity in the planet. And if you look at their habitat, ecologically, mammals have colonized almost all continents, including the most inhospitable harsh climate, like in Antarctica also you can see the mammals. And mammals have adapted to live in almost all kind of life. I can tell you some examples like gerbo rodents. So they can see in the desert, a very drier condition also you can see the mammals. And certain mammals like polar bear and brown bear, they have adapted to live in Antarctica and as well as in the ice caps. And certain animals like carnivores and archaeopteryx, they have adapted. They are very much successful in. Sorry.
The slides are not moving.
Okay, I'm yet to move.
Okay. We thought that we
Okay,
have
let me complete this. Yeah. So sorry.
Okay.
Sorry to
No
interrupt.
issues. No issues.
Yeah. So some animals like carnivorous and set of architecture, they are very much successful in living in both what you call this terrestrial and marine habitat ecosystems. And if you see the major greatest diversity of mammals, they have adapted to live in the only in a terrestrial ecosystem. Only 1.6% of mammals they occupy in the marine habitats. Now slides are moving, madam.
No, it's the first slide only. We are seeing only the first slide, title slide. Now the second slide has come.
Now my slides are moving, madam. Are visible?
No, the presentation is
Okay. So I'll just exist and relog in, madam. I think that will work.
Dr. Anzum,
ma'am.
participants usually face the problem of login, slide, and we don't know why it happens. And it happens most of the time. The speakers face problem in login and they have issue. And it was also one of the reason for delaying us.
Yeah, definitely. Actually, students' pack is over. That's why they could not remain joined.
Yes.
That is one of the reasons.
It is certainly it is the reason. And it is about to close the department.
Now it's coming.
Yes.
Yeah.
Yes, please. Dr. Kamal Kanan, please check it whether it is moving or not. Please click the second button so we could be sure. Still we can see your first slide. Yes, now it is perfect. Yes. Yes, please go ahead.
Yes, now we can see.
Please unmute yourself. Dr. Kamal Kanan, please unmute yourself. Dr. Kanan? Yes. Okay.
Please go ahead.
Yeah, so I was talking about the habitat of mammals. So the most greatest diversity of mammals live in the world terrestrial ecosystem, and only 1.6% of mammals they live in the marine habitats. And if you look at their diversity, I mean the adaptation, so they found in all types of habitats. So they have adapted to live in terrestrial, aquatic, arboreal, and they can be flying mammals like volant, and they are the burrow-dwelling mammals like fossorial. So if you see in the Indian scenario, they live from the snowiest Himalaya to deepest ocean. And if you look at the diversity, it can be at any level in the food chain. It can be herbivorous, carnivorous, insectivorous, and so on. And when we look at the classification of the world mammals, the world mammals accounts for over 6,000 species belonging to 1,300 genera of 167 families of 30 orders. Of these 30 orders, 3 orders called Rodentia, Chiroptera, and Soricomorpho occupy the 70% of the global diversity. So these are basically smaller mammals. But if you look at the Indian scenario, out of these 6,000 over species, 7% of mammal species, 446 species of mammals belonging to 48 families of 13 different orders are found in India, of which 413 species are terrestrial form and 33 species are marine form. So, of which 10 species of mammals are endemic to India. So, out of these 46 species, there are 3 genus called Anathana, Latindens, and Nilgiri-tragus. They are monotypic. So, monotypic genus means within that species, only one species is found, no more further species. So, out of these 3 species, there are 2 species Latindens and Nilgiri-tragus is found only in the Western Ghats. So, this is the importance of Indian mammal biodiversity. So, as I said, there are 13 groups of mammals found in India. So, out of these 13 groups, 11 groups like Proboscidea, Puricetactella, Carnivora, Artiodactyla, and etc. They live in the terrestrial form, and there are 2 groups called Sirenia and Cetacea. They are the marine animals. They live in the marine ecosystem. So now let us look at these orders one by one. The first order, Proboscidea, which includes 1 species of elephants. We all know that throughout the world there are 3 species of elephant: African bush elephant, African forest elephant, and we have an Asian elephant. So the elephant is the largest land animal, and the order Proboscidea can be characterized by they have a very large gray and wrinkled skin. They have a modified long snout as a single trunk, and the incisors are modified as tusks, and they have a very massive columnar legs, and they have a very ear flaps also, and of course they have a medium-sized tail with the ending with a bristle tough hairs. And when you look at the characters of Perissodactyla, so Perissodactyla is the O-toed ungulates. So O-toed means you can understand, no? It could be a 1 digit or 3 digit on their legs. So thus makes the characters of Perissodactyla. Throughout the world, there are 3 groups of Perissodactyla: rhinoceros, wild asses, and toppies. Out of these 3 groups, in India we have 2 groups called rhinoceros and asses. Altogether, only we have 3 species. And so when you look at the next group, the carnivora, we all know that carnivores are the most populous group in India, is the 3rd most group in India. And there are 8 groups of carnivores is found in India. So basically they are canids, felids, viverids, mustelids, and so on. And even including red panda also comes under the carnivore groups. So they are basically meat-eating animals. That's why they have a very developed canines, premolars, and molars. The denticles are more developed in order to seize the prey as well as to consume the prey. Same like dentition, they also have a developed claws in order to seize the prey. And the next group is Artiodactyla. So in India, there are 5 groups of Artiodactyla. So Artiodactyla is basically can be characterized by with their even toed. It could be 2 or 4 toes on their legs. There are 5 groups of Artiodactyla in India. They are basically pigs, cervids, it's normal deer, and there is a mouse deer and a musk deer, and cattle like a Tibetan antelope, Gour, and Nilgai. These all comes under the group called Artiodactyla. And they have been characterized by the balance of their weight on the 3rd and 4th toes, rather than which are found 1 or 3 in the O-toed ungulates. So primates are the most group of the mammals in the world wise, but in India it is the 6th group. In India we have 23 species of primates, which includes lorises, monkeys, gibbons, and there are macaques also. So primates can be characterized by they have a forwarding facing eyes, which are surrounded by the orbit eyes, and they have a foreshortened muzzle, and they also have an opposable thumb. So instead of nails, they have instead of claw, they have nails. And in behavior also, they can be categorized from other taxa because they have a ability to stand upright and they can change the varying of degrees also. Lagomorpha, if you look at the characters of Lagomorpha, so once upon a time, lagomorphs, I mean the pika, rabbits, and hares are called lagomorphs. They were considered as a rodent because of their gnawing teeth. So it can be differentiated from the rodent because the lagomorphs, rabbit is having the two pair of ever-growing incisors and the upper jaw. In case of rodent, only one pair is there. So this makes the difference from the rodent. And there are 11 species of lagomorphs found, of which 4 species are hares and 8 species are pikas. And we all know that Foliodot also, mammals, they're commonly called scaly anteaters and pangolins. So out of 8 species of the world, there are 2 species of pangolin called Chinese pangolin and Indian pangolin found in India. So entire life, they do not have any teeth. So if you look at the ontogenic face also, even the adult face also, you can't find any teeth. So instead of teeth, they use their intestine, stomach, keratinized activity to grind the material, plant materials or even many insects also. Scandentia, commonly called tree shrews. So there is a difference between tree shrew and true shrews. So these are not true shrews because tree shrew is completely arboreal. It can be seen only in the trees. And there are 3 species of tree shrew found in India, out of which 2 species are endemic to India. So there are 3 species like northern tree shrew, Nicobar tree shrew, and Madras tree shrew, of which Madras and Nicobar tree shrew is endemic to India. So tree shrew superficially look like a squirrel. Based on the whiskers, it can be identified whether it is a squirrel or it is a tree shrew. And they have been characterized by long snout, and they have a human-like ear flap, ear pinna, and they also have a bushy tail. So these are the characters of tree shrews. And next group is insectivores, and comes under the order called Ulipotipla. There are 34 species of shrews and hedgehogs are found in India. So the shrews sometimes they superficially look like a rodent, so it can be mistaken as a rodent. But shrews have developed dentition. They have almost all types of teeth like canine, premolars, incisors, and molars also, the premolars and molars also. But in case of rodent, they have only incisors and molars. So unlike rodent, shrews have sharp, spiky teeth, and that is the character of shrews. And if you look at the characters of rodents, as I said, they have the ever-growing gnawing teeth. The incisor, one pair of incisors in both and upper and lower jaw. And the molars could be, molar teeth could be vary from 3 to 4 in one side of the jaw. And they're the most populous group in the world, and it is the second largest group in India. And Chiroptera is the most, second largest group in the world and first group in India. And Chiroptera commonly known as bats, and there are more than 130 species found in India. Every year, 1 or 2 new species are emerging, or even 1 or 2 new subspecies are emerging. And bats are the only true flying mammals. Okay, why I'm telling true flying mammals? There are species called like flying squirrels. Actually, they don't fly; they glide from one tree to another tree. So that's why bats are considered as a true flying mammals. And their forearms modified as a wings, and they're helping in flying in the air and equally sharing their habitat with the birds and insects. And let us look at the marine animal Shirenia. So there is only one species found in this order as well as in the family called Dugongidae. It is commonly called dugong or sea cows. So unlike other vertebrae, this animal is having only 6 vertebrae. And another character to distinguish this animal from cetaceans, they do not have any canines, and the incisors are so developed. In case of male, the incisors more protruded, and that makes the difference between from other animals. And when you look at the cetaceans, so cetaceans are basically marine mammals. There are 32 species found, which are basically whales, dolphins, and porpoises. All are carnivorous, except Ganges river dolphin. All live in marine ecosystem. So Ganges river dolphin, we all know that they live in Brahmaputra and Sindhu Valley of 7 states of our country. And cetaceans can be distinguished from the fish or shark because of their lungs, and they have a circulatory warm-blooded system also. And same like other mammals, they also have a hairs which is located in their hind legs. And all cetaceans are carnivorous. Now let us come into the main topic, the role of mammals in ecosystem. So now let us understand the different benefits, importance of mammals in the ecosystem. So we all know that mammals always play the pivotal role for whichever the ecosystem they live in, and they are very much important in terms of maintaining the sustainable balanced ecosystem. And they are the important and significant members in the food chain as well as in the food webs as a predator and as well as grazers. And they can be feed at any various level. As I said, it could be a herbivorous, carnivorous, insectivorous, and so on. And they can provide essential services like they can act as ecosystem landscapers, they can act as a seed disperser, they can be a pollinators, and mammals can be a regulator of pest control also. Since the presence, absence, or abundance of mammals can indicate the health of ecosystem, many mammalian species are considered as an indicator species in the aspect of biodiversity. So now let us understand their mammals' contribution in food chain and food webs. So in the terrestrial ecosystem, plant forms the basis of all communities in terms of both function and structure. So, and these plant communities being altered by the biotic environments like mammals, reptiles, and birds, and other group of invertebrates also. Although mammals insignificant in terms of species wise compared to group like birds and reptiles, and not to mention the protozoans and insects, because of their large size and abundance, they really play the key role in food chain and food webs. So mammalian predators and herbivores influence the ecosystem by consuming their prey, and apex predators like lion, cheetah, tiger, otter species, they affect almost all trophic levels. In fact, they're the good studied group in among the mammal groups, and they play key role in regulating the abundance of their herbivorous prey. So same like secondary consumers, the primary consumers like large herbivores also affect the ecosystem by creating and maintaining the grassland landscapes and by preventing the encroachment of shrubs and trees. So mammals as ecosystem landscapers. So the role of mammals is to basically to alter the vegetation. Therefore, it is altering the nutrient cycling, and therefore there is a change of plant species composition also happening. So some of the greatest impacts of land dynamics is happening because of the dispersal or the migration of larger mammals. So this larger mammals migration is mainly happening when they have been regulated for food rather than the predators. So best example I can say the migratory herds of Asian elephant and migratory herds of Tibetan antelope. When they move from one landscape to another landscape in a long distance, they actually move the nutrients through their defecation and urine, and some poor habitat also they getting nutrients and they getting fertilized because of their nutrients movements. So because of this movement they actually shape the different seascapes, including the cetaceans in the ocean. Different landscapes and different biogeographic zones are changing because of these activities. So, as I said, the mammalian herbivores, carnivores, and plankton feeders are the largest tectine animals, and they often dominate the various ecosystem level of process, including the energy flow, nutrient cycling, and vegetation dynamics because of their energy demand. So, this structuring role as a ecosystem landscapers, not only altering the vegetation, at the same time, it's also benefiting other animals like other group of vertebrates, like smaller mammals, and other group of animals like birds, reptiles, including the invertebrates. So when we protect the larger mammals, we are protecting the larger habitats. So because of this chain sequence, we are protecting other group of animals. Because of larger habitat protection, we are protecting the other remaining community also. So that's why mammals, this role makes the mammals as an umbrella species for their conservation. Just a second. Now let us understand the mammals as ecosystem engineer. So many small to medium-sized mammals, they do acting as the ecosystem engineering. What I mean as an ecosystem engineer, they do the burrowing, or they construct the mound building, or they dig the soil. So due to this activity, they actually disturb the soils. So because of this soil disturbance, there is a chemical composition happening, and because of this burrowing activity, many other numerous vertebrates, invertebrates, they're getting their home because of their this smaller mammals activity. So that's why many burrow dwelling mammals are considered as a keystone species. So this function cannot be replaced by another animal. Now let us understand the pollination services by mammals. So most flowering plants like angiosperms and gymnosperms rely on animals for their pollination service. So animals like birds, reptiles, some insects like bees, butterflies, and mammals, they're making the pollination service, and they have interactions with the plants. So 78% of plant communities in the temperate zone communities and 94% in tropical communities are rely on animals for their pollination. So far, 355 species of mammals have been recorded for their pollination service. Now let us understand the pollination service with one example. So we all know that bats are playing the key role in the pollination service. So some daytime pollinators like birds, insects, they're considered as a daytime pollinators, but bats are considered as a nocturnal pollination, because most of the some plants in the tropical ecosystem, they bloom or they do flowering during the night time. So they rely on bats for their pollination. So like for some species of mango, guava, durian, and other species like a tropical ecosystem, they depend wholly or partly bats for their pollination services. So the significant benefits of plants by the flowering visiting bats include the deposition of large amounts of pollen and transfer of variety of genotypes of pollen. So they act as a carrier, and they act as a disperser of great amount of pollen from one distance to another long distances. Same like birds, even bats also can carry greatest amount of pollen on their face and as well as on their fur. So other than bats, there are other groups of mammals also, like marsupials, lemurs, primates, shrews and elephant shrews, even some rodents also acting as a pollinators. But in India, we have only these three group of primates, rodents, and shrews. Now let us understand their frugivory and seed dispersal action. So frugivory and associated seed dispersal is a mutualistic interactions between the mammal and flowering plants. So in this mutualism, plants provide the fleshy fruits in order to attract the frugivores. So as a mutual benefit, they provide the dispersal of seeds. So unlike pollinators like small bats, these seed dispersal mammals, they can vary from smallest mammal like bat to, and they can vary up to largest elephants. So even including some carnivores, they do seed dispersal, because some carnivores like red panda, they feed on the plant materials and flowers. So there is a study suggests that about 600 species of mammals, they can capable of seed dispersing. They belonging to 10 orders of 14 families. Out of these 10 orders, the order called primates are the most notable contributor for the seed dispersal, because most of the primates, they do actually play critical role in terms of seed dispersal. Mammals like lorises, macaque, langoose, and gibbons, they actually do the seed dispersal in the tropical ecosystem. So frugivores, pteropid bats are also considered as a seed dispersal in the tropical ecosystem. Now let us understand their services in the seed dispersal through scatter hoarding. So scatter hoarding means that the storage of seeds in a hidden place by rodents or any other small animals. So when rodent or any other animal, when they consume seeds directly from the soil, there is a vegetation of there is alter of vegetation, and therefore species composition happening. But when they started storing the seeds, there is a overall effective in seed dispersal. Here you can see the primate has stored their much of their fruits and seeds and their chickpeas, and here the rodent has stored their seeds in different habitats. So overall impact of the scatterhoarding is affecting the seed dispersal in the tropical as well as in the temperate zone communities of ecosystem. Now let us understand the different, regulation. Now let us understand the mammal as a regulator of insect pest population. So mammals like smaller mammals, especially smaller mammals, bats, insectivorous rodents, are playing key role in terms of controlling the insects. So there is a study in United States in the corn industry. Bats can save up to $1 billion per year because of their consuming the pest. Yeah. So this is one of the example. Yeah. So far, you have learned the different roles or benefits of mammals in the ecosystem. Now, let us understand. Let us more understand with one example, because I've given the elephant here as an example, because we all are familiar with the elephant. Elephant can act as a landscape architect because of the large body size. It has to roam around the forest so that there is a clear of forest, and because of this action they are avoiding the overgrowth of certain grasslands, certain plants. At the same time, they are allowing some regeneration of some plants also, so which in turn provides sustenance to other herbivorous animals also. Elephant can be a seed disperser. So we all know that elephant can eat different types of plants, fruits and seeds. And because of the poor digestion, they can defecate 16 to 18 times in a day. So as a result, sometimes the intact seed also comes in the dung. So as a result, this allows the distribution of various plants and seeds from one distance to long distance. So elephant dungs are considered as a nutrition for the habitat as well as a breeding ground for the many invertebrates, including the dung beetles. So as I said, they can defecate 16 to 18 times, and they can defecate up to 100 kgs of dungs in a day. So as a result, this gives the nutrition for the poor habitat, and it gives the breeding ground, and it gives the home for many invertebrates also. So elephant can act as a water providers. During drier condition, elephant access the water holes by digging the soil, and so as a result, other benefits, other wildlife also getting benefits. So because of the massive columnar legs, whenever they move, they make the footprints among the forest landscapes. So when it rains, it stores the water. So many small creatures getting benefit because of this action. And elephant can be a prey to apex predators like lion, tiger, and the larger mammals. So, and sometimes the elephant carcasses provide the food for other animals also. Yeah, because of the umbrella effect of elephant, it is conserving other group of animals also, because elephant conservation is the elephant habitat conservation. So when we conserve the larger habitat of elephant, we are equally or automatically conserving the remaining community. So this makes the elephant as an umbrella species. So here I've given some examples of the contribution of smaller mammals in the ecosystem. So most of the times the conservation priority have been given for the larger mammals like elephant, tiger, and lion, and so on. But most of the times the smaller mammals are neglected, and very least conservation has been provided to the smaller mammals, because most of the time the smaller mammals are considered as a pest. But now when you look at their contribution into the ecosystem, they are the primary producers, and they change the plant community, because 60 to 80% of herbivorous rodents, they can consume the primary products of plants. As a result, there is a changing of plant community composition and as well as the species distribution. And small mammal can help for the decomposition of plant materials. Actually, they influence the rate of decomposition. They will make different pieces of the organic materials, so which helps the decomposition of different material in the soil. And smaller mammal can influence the soil's chemical composition. Soil structure is largely influenced by burrowing activities. So burrowing activities and addition and the incorporation of feces and urine will make the chemical composition, like they incorporate the nitrogen. Therefore, the nutrient cycling is happening and species composition also happening. Especially it's happening in the burrow dwelling animals like shrews and marmots. So small mammal can act as a prey because of their high reproductive rate. Many animals like reptiles, like snakes and from the other group of lizards, they mainly depend on rodent for their energy demands. So small mammal can act as a predators also, because many invertebrates are being controlled by smaller mammal. For example, shrews are the most secondary consumers in the ecosystem by preying the invertebrates and other mammals. So which may have indirect impacts on prey populations and indirect influence on primary production. Here I have given a photograph, picture of a python which is sharing the cohabit of a porcupine burrow. So this burrow was made by the porcupine. At the same time, this burrow being used by the python. So most of the time, the burrow made by the smaller mammals are used by other group of animals. So that's why many mammalian species are considered as a keystone species, as a home builder for other animals also. Now let us understand why we need to conserve mammals. Okay, so we all know that we Homo sapiens, we human beings, are considered as a hyper keystone species because we have transformed the land, water, atmosphere, and the biodiversity of the planet. We are especially successful because we have able to capture the extra energy from mostly from the fossil fuels. As a result, we have a massive infrastructure like agricultural, industrial agriculture, roads, cities, and so on. So our impact so profound, some scientists argue that we have entered into the new geological epoch called Anthropocene. So because of this, what impacts or what anthropogenic activities we are doing, that will let us understand. So we are altering the earth biogeochemical cycles, and because of the use of fertilizers and the pesticide use, we have created oxygen depleted, dead zones across the freshwater and as well as in the marine ecosystem because of the biogeochemical cycles of fossil fuels and burning of agricultural field. We are allowing the planet to get it warm. At the same time, it is acidifying the marine or ocean ecosystem, and at the same time, it is increasing. We are allowing for the increase of carbon dioxide. But this was not the scene during the earlier Pliocene. But now we are facing this problem. So we have converted about 43% of the planet into the agricultural and urban ecosystem, and the remaining landscape we have dissected into roads. So our activities are disregarded the planet and driving into the sixth mass extinction. Because of this, what impacts is happening on mammals? Because about 1/4 of all mammals are now in a danger of extinction, and more than half of population are decline. 25% or 25 species of world largest mega herbivores, they have lost almost 90%, 19% of their historical ranges. And there is a study in recent years that estimated that over 200 mammal species, they lost 30% of their geographic ranges, and 40% of the species had experienced more than 80% of shrinkages during from 1900 years to 2020. And in many parts of the world, many healthy population, like some iconic species like tiger or elephant, they're completely restricted into protected areas that you cannot see in the urban ecosystem because of the anthropogenic activities. On the other hand, the illegal wildlife trade also threatening the mammals. So mammals are the most targeted group in the wildlife because after the drugs trade, the wildlife trade is the most wanted in the international illegal market. Because why mammals are most targeted group? Because mammals are being poached or hunted for their bushmeat consumption and for their derivatives also. So the International Union for Conservation of Nature accessed the 4,556 species, of which 1,169 terrestrial species are facing the threat because of the illegal wildlife trade. So this includes some iconic species like tigers, elephants, rhinoceros, and also as well as some lesser known fauna like pangolins and larger primates also facing the problem because of wildlife threat. So mammals, there is a study in 2003. 50% of wildlife seizures mainly happened from the elephant rhinoceros ivory only. So due to this, there is a negative impact on the biogeochemical cycles as well as in the what you call seascapes also. So let me conclude my topic. So we all learned now that mammals playing a critical role in the ecosystem by providing the broad range of critical functions. So when decline or a decreasing of mammals can have a will lead for the negative impacts on ecosystem. So what actions we are supposed to take? The most critical immediate actions which we are supposed to take will have to mediate the mammals and the ecosystem by doing we'll have to mitigate the habitat loss and over exploitation, and we'll have to address the impending threats of climate change. So we all know that our ecosystem is so complex because it has the smaller mammals like protozoan, and it has the animals like elephants also. So in order to conserve the this integrated system, we'll have to conserve these mammals also. And yeah, so that's all about my topic, and thanks for listening me, and that concludes my presentation.
Thank you, Dr. Kamal Kanan. You have given a wonderful view for the mammals, and your pictures were beautiful, and sometimes it was dangerous also. And I was scared about seeing these snakes and the other dangerous film. Thank you so much. We are having too many questions, but we are
Yes.
not having time to attend this. So see you some other time. Thank you so much.
Thank you, madam. Thank you for this opportunity. Thank you once again.
Dr. Anzum, you are mute. Please unmute yourself. You are muted. You.
Yeah,
Yes,
yeah.
please.
Okay, Dr. Kamal Khan, thank you so much. Like we got by some technical glitches also, and so we are sorry for that. We made you waiting, but
No problem.
nevertheless,
Yeah,
lecture
yeah,
you
it's
gave was
my pleasure.
interesting and really very interesting. We were not aware of many things which we came aware today, like how you have presented it in a very fantastic way. So
Thank you.
congratulations for that, and thank you so much. You
Thank you,
have
madam.
given a
Thank
very
you.
nice presentation. Thank you.
Uh, thank you, Dr. Kamal Karan. Dr. Rizvi,
Yes,
I
ma'am.
think we have to sum up now.
Yes, ma'am.
We are already very late. We don't have much time to say. Yes, please.
So I think so we can conclude that we had 6 lectures today, 6 talks on varied topics and by scientists and professors from different corner of this country. So first lecture was given by Dr. K.A. Subramanian from ZSI so we can this Chennai, and he had given an overview of aquatic insects, like how many types of aquatic insects are there and what is the role they are playing and how to study them, their classification, everything corner of this and was a very enlightening lecture from and so second lecture we had from Botanical Survey of India, Kolkata by Dr. Umesh Tiwari, and he has given our lecture on this Arunachal Pradesh. They have found this plant diversity of Arunachal Pradesh, and he has also like given us a ride of the this Arunachal Pradesh with different vegetations, different kind of forest, and how the diversity of each and every plant is varying at different altitudes, different. He has enlightened us about that. It was very enlightening lecture, and Dr. Sweta, you would like to say something or should I continue?
Yes, you please continue. You please continue.
Okay. So after this, we had another lecture from this plant studies by Dr. Umar Sharif from BSI Coimbatore, and he has highlighted the role Botanical Survey of India is playing in identification and conservation. Besides that, he has also emphasized upon the ethnobotany, that how the ethnic groups are utilizing the plants. So that was also very interesting. And after that, we had lecture by this Professor S.K. Malhotra, sir. I think so, Dr. Shweta, you can speak about those two.
Uh, uh, Professor Malhotra has given the overview about the invasion of the nematode. He also explained about the encyclostoma and many other parasites in the fish and has given a beautiful overview. And that lecture was followed by Dr. Rajinder Kumar, and he has given overview of the different plant varieties and the various mode of agriculture practices that could be helpful in the biodiversity conservation. And he also gave in-depth knowledge about the different varieties that has been developed by the IARI in terms of the biodiversity conservation. And the last but not the least, we had a very interesting lecture about the mammals, and the mammals are the main focus of this webinar because the participants really wanted to know about the mammals, and we were not much aware about their contribution towards the biodiversity. We usually don't speak about the contribution of the mammals to biodiversity, and that was really very interesting lecture that was given by Dr. Kamal Kanan. So that was enough. And we are receiving some requests from the participants for the slides and the some questions, and I have already given message to participants over the mail that if you want to have any question, if you want to ask any question from the speakers, please send us and we will try to get the answer from you, and that will be forwarded. And second thing is that with your kind permission, I would say that if someone wants to add something in the proceeding of the webinar, they can send to us. And when we will prepare our proceeding, we will jointly take the decision whether their statement would be included in the proceeding or not. That I want to say. Yeah, and what is your take on this?
Yes, ma'am. I think so. It is a wonderful idea. We can include the proceedings we are going to make, and we will also include in the proceedings about the what feedback we are getting from the students. That also, or like we'll get an idea what to do next. So that will be interesting.
Yes, that I was thinking. So every participant will have their statement, and I am putting this information in the chat box also. We will send a separate mail, and they can send me if they are not receiving my mail. Sometimes it happens. I am putting my mail for them, so they can send me their statement or any specific requirement from us that could be helpful for their research activity. So I just gave one message in the chat box. Anything else from the participant? If any participant want to submit any statement, yes, they can come forward. I think they are exhausted now. They don't want to stay. It's a long time having. Now they are overloaded. So, what do you say, Dr. Rizvi? Would you like to? No, you are muted. We cannot listen to you. Please, Dr. Rizvi, Dr. Ansu, please unmute. Dr. Ansu, please unmute.
Again, I got. I think so. We should wind up now. So we can thank everyone, all the participants, and also
Professor Sweta Yadav for wonderful coordination and cooperation throughout. So now we'll sign off. Have a good day.
Okay, now it is to be good night, not to be day.
Oh
Day is
yeah,
not
good night.
remaining. But anyway, for the
Yeah.
formal thanks, I would say big thank to my team. I would like to say a few names, especially I am taking it from the seniority from my lab, Dr. Munir Malla, Mr. Azhar Rashid, Mr. Samreen Thakur, Mr. Anupam, Ms. Kiran, Ms. Soumya Bharadwaj, Ms. Nalini Tiwari, Ms. Pooja Tiwari, for their participation and making it successful. And a big applaud for Mr. Vikram, who is behind the scene and he is taking care of all technical setup to this meeting. Even we lost the connection in the middle of the meeting and he arranged the setup to make it continuous. So thank you so much all and my big thank to Dr. Anzum. We will be connected
Okay, dear.
soon.
Future connectivity,
Great.
future.
Of course.
Looking
So
forward to many more like collaborative workshops and training seminars in future. Definitely. We
we had a good experience with work you. Okay,
sure.
thank you. Bye bye.
Thank you. Bye
Bye
bye.
bye.
Good night,
Good night.
everyone.
Good night. So thank you.
Thank you, ma'am.